तो उन्होंने उस पर ट्रायल किया है, एक्सपीरियंस आदमी है, उनको भी हम जरा साथ में जोड़ते हैं।
मेरा डॉक्टर सक्सेना साहब को प्रणाम करता हूं। मैं डॉक्टर पांडे बोल रहा हूं, रानी लक्ष्मीबाई केंद्रीय कृषि विश्वविद्यालय से। सर, मोस्ट वेलकम फॉर टू नेशनल वेबिनार।
नमस्कार
सर सक्सेना साहब। सर, हम लोग थोड़ी देर में कार्यक्रम शुरू कर रहे हैं। सर, आपकी उपस्थिति प्रार्थनीय और आपने हमारा निमंत्रण स्वीकार किया, हम लोग बहुत आभारी हैं। सर, अभी हमारे ऑनरेबल वाइस चांसलर साहब भी सर जुड़ रहे हैं।
धन्यवाद।
दूसरा होगा ये ठीक। ऐसे घुमा के बैठे उधर से, वही ठीक रहेगा। हाँ, वो
नोट मैंने दिया था।
हाँ, मिल गया।
वही है ना, वो शासन का जीवन वो पूरी ये चुप रहना।
कोई पीए में तो होगा ना, उनका कोई पीए वगैरह तो होगा, हम करा दिया यही तो निकल जाएंगे।
डॉक्टर भट्ट,
Should we start?
I think we start. We have also joined, है ना? And Arunachalam sir also joined. So please start.
Ladies and gentlemen, a very good morning and greetings.
Little louder.
Ladies and gentlemen, a very good morning and greetings from Bundelkhand. Respected Vice Chancellor, Professor Abhinav sir, Rani Lakshmibai, Central Agricultural University, guest of honor of this event, Professor A. Arunachalam sir, Director, Center for Agricultural Research Institute, Jhansi. Chief guest of this event, Shri Vinay Kumar Saxena sir, Chairman Khadi and Village Industries Commission, MSME, Government of India. Chairman, panelists and speakers of different technical sections of this webinar. A warm welcome from the family of Rani Lakshmibai Central Agricultural University to our distinguished speakers and the participants from across the country. It is truly an honor and a privilege for College of Horticulture and Forestry to host this webinar. On behalf of the RLBCAU, I, Jahangir Bhatt, extend our heartiest welcome to all of you. Without any doubt, this webinar will be a worthwhile experience as we have many absolutely intellectual, knowledgeable and experienced people on board for today's event. We have an opportunity before us to learn from some of the best experts in this field. I hope at the end we all will have takeaway home messages enriched with a better understanding of nature and livelihood security through Neem. Before we start, let me take this opportunity to acknowledge the presence of our registrar, Dr. Mukesh Srivastava sir, Professor A.R. Sharma sir, Director Research, Professor S.S. Singh sir, Director Extension Education, Professor Anil Kumar sir, Director Education, Professor S.K. Chaturvedi sir, Dean College of Agriculture, Professor S.S. Kushwa sir, University Library, all esteemed heads of departments, scientists, faculty members, students of all, ladies and gentlemen. To start with today's event, may I request Professor A.K. Pandey sir, Dean College of Horticulture and Forestry, and also the patron of this webinar, to welcome the participants with his address. Please, sir.
Thank you, Dr. Jahangir Bhatt Sahab. Most honored, our worthy Vice Chancellor, Professor Arvind Kumar Sahab, Vice Chancellor Rani Lakshmi Bai Central Agriculture University, Jhansi. Today's very renowned speaker and chief guest of the function, Dr. Vinay Kumar Saxena Sahab from KVIC, New Delhi. Our guest of honor, Dr. Arunchalam Sahab, Director, Kafri. All my esteemed colleague, Rani Lakshmi Bai Central Agriculture University, our Dean College of Agriculture, Dr. S.K. Chaturvedi, Director Research, Dr. A.R. Sharma Sahab, Director Education, Dr. Anil Kumar, Director Extension Education, Dr. S.S. Singh, our Registrar, Sri Mukesh Saxena Sahab, Mukesh Ji, all my very distinguished participants and very honored our panelist, chairman of the various sessions. I am looking after my very senior colleague, Dr. A.V. Rai Sahab, Head Indian Institute of Vegetable Research, Varanasi. Participants from Gujarat, Dr. Patel Sahab, who has accepted our request to speak on neem coated urea, and so many, Professor Salil Tiwari from G.V. Pant University. Dear delegates, today we have gathered on very important webinar. Its topic is very important: nurturing the neem for nature and livelihood security. In today's context, our every activities has to address not nutritional security in single manner. We have to pay a very holistic approach for our environmental security also. Neem, I consider it is a rare gift of the nature on planet. A plant full of this your pharmacy, whether it has been to this your Ayurveda to Siddha or Unani to homeopathic, in every therapy, its description has been mentioned. And this plant, though has got its origin into African to Asian continent, has traveled globally, and everywhere it has got its respect because of its immense potential, its immense eco services, its immense different agriculture related practices. And in Indian country, it has got a so immense value that everywhere, right from north to south, east to west, everywhere it is worshipped because of its potential. And in today's context, where we are struggling for sustainability up to this your agriculture, though after the green revolutions, we have got a quantum jump. Globally, we have got a quantum jump. But present day, we are facing the problem of whether it will be sustainable or not. And making to this your sustainable, we have to be a very holistic approach where we have to deep in our locals. And that's why this your our honorable Prime Minister Narendra Modi ji always speak that you have to give your attentions part to this your local. That's why he says part to this local for the global attentions. So friends, in today's webinar, which is focus on to this your neem tree, a beautiful plant for its very bewitching beauty, its all medicinal parts, and it's so hardy nature, that it can survive into any difficult up to these your situations at more than to 37 degrees centigrade to even lowest to this your 7 to 8 degrees centigrade of temperature, different level up to this your pH, different conditions up to these your soils, right from arid to these your coastal regions. So we have to harness its good potential, and that's why today's webinar is focused into different technical sessions. One technical session is the very much focus on to Neem, its immense role in plant health management. Second, its role, as I mentioned about to its all soil amendments properties, and in recent parts its a major usage into soil coated urea, where we have to enhance to this your nitrogen use efficiency. That is the very good work, and country has taken lead into this your neem coated urea productions. Today every our urea is coming, even imported one is also coming to this your coated ones, and enhancing to this your nitrogen use efficiency. It is a very economical for to this your growers point of view, and side by side it has added a number of advantage also in other respect to this your giving very good rhizosphere conditions for to this your different our soil health related different these your microorganisms also. And side by sides it's a different biopesticidal properties. This will be again, it will be addressed in a one technical sessions, where technical sessions Neem has been given as a Neem for plant health management. So I am well confident that in today's all three sessions, one session is dedicated for its genetic resources and its genetic improvements, where learned speaker has been invited. And so I am very much happy that in today's all this your session is will focus on to this your Neem's harnessing its potential. Dear delegates, I am happy to welcome all of you at Rani Lakshmibai Central Agriculture University, and I am very much happy to share that our Chief Patron, Honorable Vice Chancellor Professor Arvind Kumar Sahab, is a renowned personality in the field of agriculture. He is the visionary person, and in every of such type of this your event, what he utters a single words that counts a lot, and that gives the good strength. And he is noted scholar up to this your country, has given a newer height to this your agriculture education system of India, and we are happy that we have got the opportunity to work under such a dynamic leadership. I am further very much happy to accept our invitations Binay Kumar Saxena Sahab, who is a very important persons in KVIC. As I was looking his brief biodata, a very versatile personality who has given in never height to this your KVIC, and apart from that, he has worked in the different scientific organizations where he has paid a good attention for enriching to this your country's bioresources. And apart from that, he is a very known prolific writer also in number of books, and number of books related to this your scientific, and number of related books he has published onto this your literature also, with the very very beautiful titles. So sir, welcome again to this inaugural session of this national webinar on Neem. My guest of honor, Dr. Arunachal Sahab, he is having very ardent love for to this your environment, and at Kafri Central Agroforestry Research Institute, where he is the director, he is taking lead for promoting to this your Neem, and beautiful plantation and beautiful work is going on to this your Kafri. So with these words, once again I welcome to our patron Professor Arvind Kumar Sahab, Honorable Vice Chancellor, and today's chief guest, Dr. Saxena Sahab, guest of honor, Dr. Arunachalam Sahab, all my esteemed delegates, chairman of the different technical sessions, panelists, reporters, and all esteemed participants for today's your webinar. Thanks.
Thank you very much, sir. Thank you for your address. This morning we have with us Professor A. Arunachalam, guest of honor for this event. He is Director CAFI Jhansi. Let me take this opportunity to brief the audience with his vast experience, from academician, researcher to administrator. He is the fellow of NAS and many more national and international organizations and societies. He has been awarded earlier with many honors like fellow DST Bioscost in Restoration Ecology from Boston, United States of America, and many more. His research is evidence of contribution to the science and scientific fraternity. He was earlier at ICR headquarters, New Delhi, before taking up the director position at CAFI Jhansi, and worked there in different capacities as Principal Scientist, NRM, and ADG International Relations. May I now request respected Dr. A. Arunachalam, sir, Director CAFI Jhansi, to address. So please.
Good morning to everyone. Thank you very much, the fraternity of RLBCAU for remembering me on this occasion. Absolutely, it's a talk of the day and need of the hour, and it gives a very soothing effect to all of us to remember one of the pristine forms of plant life form in this planet, that is neem. So I remember in 2006 and 2020, we celebrated the National Science Day with a theme, Nurture Nature for Future. And that was the kind of scenario that we have developed for this webinar today, that nurturing the neem for nature and livelihood security. It's indeed a very appropriate proposition that we are all here and we'll be deliberating on so many issues centered around neem. And we remember neem as a spiritual tree. We remember neem as a tree that gives us enough shade. We remember neem for its oxygen yielding potential. We remember neems for those yellow colored fruits that drop down that we used to gather and sometimes eat when we were all small and young. And we remember the neem in the name of the King Ashoka also, who planted thousands and thousands of neem tree around the roadsides. And it is a tree immensely intertwined with human life, lifestyle, and livelihoods. And that is why the value proposition of this particular species is manifold. And its structure itself gives a kind of message to all of us that have a broad canopy, smaller leaves, strong wood, give more and more to others, allow more and more organisms to come into the canopy zone of itself. So that's the kind of message that we human must take from this particular species. And as far as the appraisal was concerned, Professor Pandey appraised it very rightly. It has all properties. It has all properties. And so we will be deliberating on those independently. But then in today's context, neem becomes very important species because we are all looking forward for chemical-free agriculture, And one of the transition that we are taking using Neem is the Neem coated urea. And so the kind of transition that we are trying to bring into the farmers field needs potential germplasms of Neem, which has good quality acidirecting content. And that is where today's present day research is very important. And for the information of all the August gathering here virtually, Central Agroforestry Research Institute, Jhansi, is maintaining about 276 accessions of Neem in our own farm. So we have the largest collection. In fact, it is pronounced to be the National Field Gene Bank for Neem. And we have already characterized about 170 accessions, and 139 have been already registered with NPPGR. The rest of the work is underway. So a lot of opportunities centered around this. And for Bundelkhand region also, it has relevance. We need this species for our lifestyle development, and also livelihood security. So I'm sure the deliberations will move along in a manner that we are all able to use the double helical model of DNA towards this species. On one of the helix, it could be nature's propaganda. The other could be the human-centered livelihood. So all the best for you for the national webinar on nurturing the Neem for nature and livelihood security. Thank you very much for roping me in, Professor Pandey, and the entire RLBCAU fraternity, and with special respects and high regards to the Honorable Vice Chancellor of RLBCAU. Thank you so much.
Thank you, sir, for your kind and enlightening address. Rani Lakshmibai, Central Agriculture University, is highly thankful to you for taking time out for this event. It will be a great privilege for us to listen to you again for our future direction. Thank you, sir. Ladies and gentlemen, we really get this kind of opportunity to listen to the words of a great, knowledgeable and visionary person. The chief guest of today's event, Shri Vinay Kumar Saxena, currently leading Khadi and Village Industries Commission as chairman and alumnus of Kanpur University with a pilot license. Shri Saxena is a philanthropist in vision and corporate scientist in action. And his vision and action are so eternal that he always gave priority to his immense social commitment, right from the corporate to the NGO sectors. Shri Saxena has always led from the front on any issue related to social development like water resource development, fighting the social malpractices, disaster management, sport, or clean development of the city space. He is the person behind the setup of NCCL in 1991. He has actively pursuing a project named Rainbow Hills to adopt and develop villages across the country under CSR funds. In 2008, he discovered that the statue of the father of the nation, Mahatma Gandhi, was not placed where it was supposed to be placed at Madame Tussauds Wax Museum. He took up the matter with the political leadership in UK and India and pursued the issue until an apology was offered by the museum office, and the Mahatma statue was shifted to the world's leading exhibition hall. Shri Saxena, a member of various distinguished committees, has undertaken several efforts and innovative ideas for the benefit of humanity. Thank you very much, sir. I would like to invite you, sir, to deliver your valuable talk. Sir, can you hear me? Sir, can you hear me?
अभी ज्वाइन करते हैं भट्ट साहब, बस दिख रहे थे डॉक्टर साहब।
अच्छा, मॉडरेटर वो है।
Hello. Hello. Can you hear me?
Yes, sir. Yes, sir.
Yes, sir.
Hello. Thank you. Professor Arvind Kumar ji, Vice Chancellor, Rani Lakshmi Bai Central Agriculture University, Dr. A.K. Pandey, Dean, Rani Lakshmi Bai Central Agriculture University, and other distinguished guests. First of all, I want to convey my sincere thanks to Professor Arvind Kumar ji for inviting me for this national webinar on nurturing the neem for nature and livelihood security. It's really a very, very important seminar. And right now I'm in Varanasi. And because of my busy schedule, actually, I was requesting to postpone this program, but somehow I'm here to attend this program. Thank you very much for again and again calling me. Well, neem is very, very important tree. Neem is a household name actually in India that is used for multi-purposes, essentially because of its antibacterial and antifungal properties. For this reason, neem is also occupies a prominent place in Ayurveda and is known as a natural healer. Neem is also known to be the best natural air purifier. And this assumes great significance when several big cities in the country are grasping for fresh, breathable air. Neem is a common but a prominent tree species in almost all parts of the country, particularly the Bundelkhand region, which has an abundance of neem. Due to its abundance, there is an ample scope of harnessing the commercial potential of neem. It is also crucial for biodiversity conservation. Promoting the plantation of neem on degraded or unused land can be of great benefit to the environment. Neem is one species that can easily be grown in the forests as well as on the farmlands without any special care of or investment. Neem-based agroforestry can also support the rural livelihood and rural economy in a big way. Incidentally, promoting rural livelihood and strengthening the rural economy are also the key objectives of Khadi and Village Industries Commission. Neem seed collections, neem-based manure, neem-based pesticides, neem-based of great advantage to the environment. Use of neem-based pesticides and manure are also safe for honeybees and earthworms, and thereby increasing the crop yield. Collection of neem seeds and manufacturing of neem-based products like non-edible neem oil and varieties of neem soaps have been a major segment of Indian village industries, particularly promoted by Khadi and Village Industries Commission. Neem is extensively used in making neem oil, neem-based soaps, and several cosmetic products owing to its medicinal properties. And I'm very happy to share with you that around 26 varieties of neem soaps are sold by KVIC and are manufactured by KVIC units across the country. There are 26 varieties of. And there are several varieties of neem oil which are used in medicinal purposes. These are also promoted by KVIC. So KVIC is promoting neem-based industry in a big way. Well, the point is how to promote this, how to go ahead about this. rural industry, particularly the village industry, is facing a tough challenge these days. And there is a need to create sustainable employment opportunities in the rural areas. And KVIC is exactly doing the same. गांधी जी का जो विजन था to promote village industry, the same vision is being propagated by the Honorable Prime Minister today. KVIC, through its various resources, is doing various activities. Bundelkhand, particularly the Bundelkhand, I know Bundelkhand very closely because I have spent a lot of time there. Bundelkhand is one such place where there are abundance of neem trees. But the point is they are not properly harnessed, they are not properly used. One thing which I want to propagate is the promotion of beekeeping activities. नीम का जो पेड़ है, उसके जो फ्लोरा और फौना है, उसमें जो पोलन और नेक्टर अवेलेबल है, that is very good for honeybee activities. I request the Honorable Vice Chancellor to promote honeybee activities, beekeeping activities in Bundelkhand region through his university. ये एक क्रांति ला देगी उस रीजन के अंदर. We have already entered into this field very recently. हम लोगों ने वहां पर एक प्रोग्राम किया था 2 महीने पहले, and we have distributed bee boxes to the farmers. But this is a new subject in that region. It's a very new subject in that region. बुंदेलखंड को कहा जाता है it's like arid zone, और एरिड जोन में कुछ हो नहीं सकता. बट द नीम के जो वहां पर पेड़ हैं वो हमारी बी कीपिंग एक्टिविटीज के लिए एक बहुत बड़ा जो है काम कर सकते हैं. वैसे भी आपको मैं कहना चाहूंगा कि दादी माँ के समय से जो है नीम के पेड़ की वैल्यूज को बताया जाता था. अगर किसी के को खुजली जैसी चीज हो गई तो नीम के पत्तों को उबाला जाता था और उसके पानी को बॉडी में लगाया जाता था और उससे जो है उसको ठीक किया जाता था. इसी तरीके से कोई भी चीज अगर जरा सा जुकाम बहुत भी हो गया तो नीम के पत्ते को निचोड़कर उसको सुंघाया जाता था. ये जो दादी माँ के जो नुस्खे थे इसको तो हम लोग यूज करते ही करते थे. लेकिन आज जरूरत है कि हम नीम के पेड़ की जो कमर्शियल वैल्यूज हैं उनको जो है हार्नेस करें. मुझे याद है कि GNFC जो गुजरात नर्मदा फर्टिलाइजर कॉर्पोरेशन जो भरूच में एक बहुत बड़ा प्लांट है, उन्होंने नीम के पेड़ों को और उनकी इकोनॉमी को किस तरीके से यूज किया है, मैं समझता हूँ एक बहुत अच्छा एग्जाम्पल है. नीम के कोटेड फर्टिलाइजर को तो उन्होंने इंट्रोड्यूस किया ही किया, 10,000 महिलाओं को उन्होंने नीम के कलेक्शन के लिए यूज किया और वहां पर उसके सोप को बनवाया गया, उससे और भी सारे प्रोडक्ट्स को बनाया गया जो खादी एंड विलेज इंडस्ट्रीज कमीशन इसको प्रमोट कर रहा है. तो इस तरीके की इकोनॉमी को हम जो है बुंदेलखंड में भी डेवलप कर सकते हैं और ये कोशिश कर सकते हैं कि केवीआईसी के साथ जुड़कर हम वहाँ के रूरल जो लोग हैं, जो गरीब लोग हैं, जो गांव के लोग हैं, उनको रोजगार से भी जोड़ सकें. आज जरूरत हमें है ये सेमिनार करने की इस वजह से भी कि हमें किस तरीके से सस्टेनेबल एम्प्लॉयमेंट क्रिएट करना है. जब तक हम लोकल लोगों को एम्प्लॉयमेंट नहीं क्रिएट कर सकते तब तक मैं समझता हूं कि हम जो ये सेमिनार्स करते हैं या इस तरीके की जो कॉन्फ्रेंसेस करते हैं उसका कोई मतलब नहीं निकलता. हम इस कॉन्फ्रेंस के बाद हमारी ये कोशिश होनी चाहिए कि एक ऐसी स्कीम को बनाए जहां पर हम लोगों को इस नीम के पेड़ से होने वाले फायदे को बताएं और उसके साथ साथ उनको रोजगार से जोड़ सकें. नीम का ऑयल आज जो है बहुत सारी मेडिसिन के लिए यूज किया जा रहा है. आज जरूरत है कि छोटे छोटे यूनिट्स को लगवाया जाए. केवीआईसी की स्कीम है प्राइम मिनिस्टर एम्प्लॉयमेंट जनरेशन प्रोग्राम, जिसमें हम लोग 25 लाख रुपए तक का लोन देते हैं छोटे से यूनिट को लगाने के लिए. 1 लाख रुपए से 25 लाख रुपए तक का यूनिट जो है कोई भी लगा सकता है. तो नीम एक्सट्रैक्शन का जो ऑयल एक्सट्रैक्शन का एक बहुत बड़ा यूनिट लग सकते हैं. पूरे बुंदेलखंड के अंदर इस तरीके की हजारों यूनिट्स लग सकती हैं और केवीआईसी उनको मार्केटिंग का प्लेटफॉर्म भी दे सकता है. तो ये जरूरत है कि हमको इस तरीके की छोटे छोटे यूनिट्स को हम डेवलप करें और लोगों को रोजगार से भी जोड़ें और जो नीम वहां पर बुंदेलखंड में इस वक्त है उसको हम एक प्रॉपर यूटिलाइजेशन के फॉर्म पर लेकर आए. ये जो आज जो वेबिनार हो रहा है, मैं सबसे पहले तो बहुत मुबारकबाद देना चाहता हूँ वाइस प्रेसिडेंट साहब को कि एक बहुत अनोखे सब्जेक्ट के ऊपर जो है इस वेबिनार को आपने ऑर्गेनाइज किया है। ये एक बहुत इंपॉर्टेंट सब्जेक्ट है नेशनल पॉइंट ऑफ व्यू से ऑल्सो, कि नीम के पेड़ सिर्फ बुंदेलखंड में नहीं पूरे देश के अंदर हैं और हर जगह है। नीम की ठंडी हवा तो हमें मिलती ही है, इसमें कोई शक नहीं है। हम अक्सर ये कहा करते हैं कि नीम कड़वा होता है। एक्चुअली नीम कड़वा नहीं होता है, हमें मीठा खाने की आदत होती है, तो खुदगर्जी जो है वो हमारी जुबान की होती है कि उसको मीठा चाहिए। नीम तो जो है वो है, नीम की ठंडी हवा भी है। तो मैं ये कहना चाहूँगा यहाँ पर कि आज जो इस सेमिनार को हम लोग ऑर्गेनाइज कर रहे हैं, ये बहुत सुंदर प्रयास है और उस प्रयास के माध्यम से हमारी ऐसी कोशिश होनी चाहिए कि बुंदेलखंड जो देश का एक बहुत पिछड़ा इलाका है, जहाँ हमें ज़रूरत है लोगों को रोजगार से कनेक्ट करने की, वहाँ के आदिवासियों को, वहाँ के रूरल लोगों को इस वेबिनार के माध्यम से एक ऐसा हम एक प्रोग्राम बनाएं जिससे कि हम वहाँ के लोगों को ज़्यादा से ज़्यादा रोजगार से जोड़ सकें और इस पेड़ को जो वहाँ पर बहुत ज़्यादा उपलब्ध है उसको एक कमर्शियल तरीके से यूज करके आगे बढ़ें। और तभी मैं समझता हूँ कि हमारी सफलता होगी जब हम बुंदेलखंड के उस इलाके को, जो मैंने कहा जैसा कि देश का सबसे पिछड़ा इलाका है, उसको देश की मेन स्ट्रीम से जोड़ने का प्रयास करेंगे। केवीआईसी का जहाँ तक सवाल है वो इस तरीके के प्रयास कर ही रहा है और अगर हनी मिशन से वहाँ पर एग्रीकल्चर यूनिवर्सिटी उन्हें जोड़ना चाहती है तो मैं आपसे ये वादा करता हूँ कि केवीआईसी आपके साथ खड़े होकर इन प्रोग्राम्स को इम्प्लीमेंट करने का प्रयास करेगी। मैं एक बार फिर आप सबको बहुत बहुत धन्यवाद देता हूँ कि आपने मुझे इसमें बुलाया और मुझे सुना, और मैं आशा करता हूं कि हम जो है एक दूसरे के टच में रहेंगे। जब भी मैं झांसी आऊँगा तो मैं वहाँ मुलाकात भी करूँगा और कोशिश करूँगा कि हम किस तरीके से इस चीज़ को आगे बढ़ाएं और वहाँ के लोगों को जो है एक बार समृद्धि के रास्ते पर लेके जाएं, जो मैं ऑनरेबल प्राइम मिनिस्टर का विजन है। बहुत बहुत धन्यवाद।
बहुत बहुत आभार सर, बहुत बहुत धन्यवाद आपने इतना बढ़िया संबोधन के लिए।
Informative address. I, on behalf of my university members, would like to thank you for sparing your time to address us. We owe you a special vote of thanks for honoring this virtual gathering with your presence in your demanding schedule. Thank you, sir. Thank you, and we look forward. Since you said that you are going to visit to our university, that will be our great honor, sir. Thank you, sir.
Mr. Jagir, I want to thank Saxena sir for mentioning GNFC. Sir, GNFC is also putting up his case study to the learned forum. So we will in detail explain our project also. Again, sir, thanks.
Yeah.
Privilege you had mentioned GNFC.
Thank you, sir.
GNFC has done a commendable job actually in utilization of Neem. I know it very, very personally, and the managing director and chairman of GNFC was personally known to me, and we were in constant touch. And with the help of KVIC, we have set up several units there, and now GNFC is supplying soap, oils, etc. to KVIC, and they are creating sustainable employment in that region.
KVIC has helped a lot for it also, sir. Thanks, sir.
I know that. Yeah. Thank you very much.
Thank you, sir. Let us now have the pleasure of listening to the presidential address from the chief patron of this event, Honorable Vice Chancellor, Professor Arun Kumar, sir. Please, sir.
I want to take a leave because I have to rush somewhere.
So, good morning to everyone. Particularly today, I could listen the chairman of KVIC, Saxena Saab, who is a very important person because this KVIC is engaging more than 1.4 crores population. So it's a huge population which is linked with the KVIC with a turnover of more than 52,000 crores. So I know KVIC because my colleague and my class fellow Dr. Yasveer Singh For some time, he was chairman of this KVIC. So we used to discuss what the role of KVIC, and today I am very pleased to see Dr. Saxena over here. And I would like to tell you, he has rightly pointed out about honeybee. We have already started, and with a fund of about 2.5 crores from RKVY, and also we are getting funds from Honeybee Mission. And on the boundary, you will be glad to know, of about 5 kilometers, we have all planted a neem tree so that it gives not only cool air but a good atmosphere to the university. So many initiatives, and I thank him for showing his desire to come to Jhansi. Surely we will welcome him, and we will request him to visit at the earliest to see our activities, and also how best we can take the assistance of Khadi Village Industry Commission, and in providing more and more startups, employment opportunity, and so on. And he is a man who is a very creative person. You must not be knowing that he has taken a bold initiative as far as the marketing strategy concerned, involving various corporates, marketing convergence, MOU with different organizations. Because if you listen to the news, many bulk orders like in Air India, or even PMO office, or even in the health sector, post and telegraph. So many railways, many, now they are the bulk taker of the Khadi products. So this way, and our Honorable Prime Minister, he is so much always propagating about Khadi. So because of this, this industry has really done a commendable job, particularly for building up the artisans in rural India. And he is the president of NCCL also, which have done a great relief work, I know, for earthquake in Gujarat. So lot of initiatives he has taken. I am very happy to see Saxena Saab over here, and surely I know he has got limited time, but it was very nice to hear him this morning, and his presence gave us valuable thoughts and inputs which shall be valuable for our team to carry forward the activities in collaboration with KVIC. So thank you, Saxena Saab, for being with us, and thank you a lot. Thank you very much. We know that this particular today's webinar organized by our College of Horticulture and Forestry. This university was established by an Act of Parliament as the first institution of national importance in 2014, March 2014. On 5th of March, we are celebrating our 8th Foundation Day. And we have been able to establish College of Horticulture, Forestry, and also the Agriculture, and also in the process of establishing College of Veterinary and Animal Sciences and College of Fisheries at Datia in Madhya Pradesh. So Bundelkhand region would certainly be benefited by the presence of this first institution of national importance and the Central Agricultural University in this region. You know that this particular neem is, is a wonder tree, as Dr. Arunachalam had mentioned. It's not only a tree, but it's a boon to our, system. And, this is a tree which, provides us fresh air over a long period of time. If you take the, fully grown neem tree, almost it can survive up to 2 centuries. It's a, you must have seen very, very old tree in different parts of the country, starting from arid ecosystem to humid and perhumid ecosystem. So in various tracts, this neem tree, old neem tree are still available, and it starts fruiting in just, after 5 years, but key productive period comes after 10 years. And it's, products, each and every product, be it bark, be it leaves, be it, which we call neem kernel, or any part, every part of neem is really very, very useful for our health. It has been mentioned quite a lot about the benefits of neem tree by Dr. Pandey and our Saxena Sahab. I will not go into detail, but name anything, and whether it's antibacterial or antifungal activities or antioxidants, take anything. Neem is helpful. You remember when there was a problem of smallpox, we used to have the neem twigs nearby the bed of the patient, and also in the room of the patient. So this helped to protect the chicken pox. So this way, even the boiling of the neem leaves and taking a bath in the when you have the chicken pox, this helps. Even the neem soap, many skin disorders, treating acne and fungal infections, healing burns, and also the many such diseases, even moisturizing skin, dandruff, hair growth, lot of use in the toothpaste, which is being done, and in the leaves, even the you take the neem leaves, it's very good for cure of the diabetic. Even now, many diabetic drugs are made out of these neem products, neem leaves, and particularly for leprosy, eye disorders, even the intestinal worms, this neem leaves work, even the skin ulcers, and also in many cardiovascular diseases, gum disease, liver disease, even you talk about even the fallen leaves, which have, or the fallen leaf kernels, which are also valuable. We used to eat them. Even we used to have, like a cleaning our teeth, this neem twigs, neem which we used to be a very potential source and a remedifier, a source of blood purifier. So all kinds of benefits of the neem are very well known. We have on our university also one of the neem block, and also in nearby Central Agroforestry Research Institute, we have the neem plantation, different species. We are working on it, and we would like to certainly take a lead in propagating this neem in different parts of the Bundelkhand region, have associated with a small scale industry, because this KVIC is directly linked with MSME, and this small and medium scale sector would certainly be benefited by the plantation of the neem and also utilization of these products and byproducts. We talk about immunity, particularly in Corona period. This neem is known, very well known, to boost up our immune system. Even if you take 4 to 5 leaves, particularly the newly grown leaves, daily, it cures many things. Just empty stomach, if you take it, it cures many things. So that is what neem is a wonder plant, and certainly like other tree we call people, or we call banyan tree. This neem has got its very valuable place in our society. Not only in for the various health benefits, you have even in the soil. It also helps in repelling the nematodes. It also helps in because the leaves which are fallen are slightly alkaline in reaction. So if you add to the soil, it neutralizes the acidity in the soil. So that is also a good neutralizer, and that can be used for neutralizing the acidity with the neem cake for many of the harmful you can say organisms. This neem is a very good remedy. Particularly, this has been very well utilized while we talk about the neem-coated urea. And the neem-coated urea, it has really made availability of the urea and slow release. Therefore, the losses which used to occur of nitrogen have considerably been reduced. So this neem coated urea is, making the efficient utilization of our nitrogen. Hardly 30 to 50% nitrogen which we add to the soil is being utilized. Efficiency is very less. So to improve the efficiency, to have the slow release, and to reduce the losses, the neem coated urea has a great significance, which has been taken so prominently by the government of India, and this is becoming now very popular. And now this neem coated urea is widely used for the benefit of the farming community. So this way, this 2 days webinar, which is very useful, you will be able to learn many things. Even we talk about immunity, we talk about turmeric. Turmeric too is also, improves our immune system. So if you take neem plus turmeric plus also you have some honey, so this certainly helps in boosting your immunity system, particularly in corona period. Lot of usage of this honey or neem or turmeric, which you mix together, and it will certainly improve your immune system. So many good things are available, and even with the single plant, you can have even if you have 50 to 60 kg kernel. Initially, it can more than 100 rupees the cost. It depends in different markets and the availability. But even you take 100 rupee or 125 rupee, it certainly gives you a quantum amount per tree, which can be a source of income for the rural community, and also a source of livelihood, and also not source of livelihood, but as it was being talked about, some self-help groups are involved in collecting kernels, selling to the market. This is a product which is always thought of, and if we make collection of the neem kernel, certainly the lot of revenue generation can be had with this. Even in the upliftment of the socio-economic status of the farming community, even on the burns, even in the social forestry system, even on the barren lands, neem could be grown, because this is such a versatile plant which can be grown in different soil conditions, different agroclimatic situations. So this is again I call a very wonderful and very valuable tree. And 2 days webinar in different experts will be telling you, speaking about the different benefits, how you can get the advantage, how you can grow the neem, what may be the impact of it, what are the different species, what are the different products and byproducts, how you can harness the potential of this neem tree. All these shall be talked about. Once again, I congratulate the Dean College of Horticulture and Forestry, Dr. Pandey, Dr. Dobrial, and all his team for organizing this important webinar, particularly on the neem, which is I would call such a subject which people will certainly not think of, that we can have a webinar. And after the conclusion of this webinar, we will try to compile the deliberations and will put on our website, and certainly make its availability to different stakeholders, so that everyone can get the benefit. So thank you very much. Thank you all the participants for joining us. And thank you for being with us this morning. Thank you.
Thanks.
Thank you, sir. Thank you so much for your address.
Thank you, sir.
Your guidance and sharing of knowledge is a constant source of encouragement for us. We look forward to hearing from you again in our future activities. Thank you, sir. As we approach to the close of this inaugural session, I would like to request Professor M.G. Dobrial, sir, the event's organizing secretary and head forestry, College of Horticulture and Forestry, to present a vote of appreciation. Please, sir. Gabriel sir. Gabriel sir,
Please unmute, sir. Gabriel sir, kindly unmute.
Oh, thank you, sir. So, good morning to all. Honorable Vice Chancellor, sir, the President of this webinar, inaugural function. Our Chief Guest, Shri Vinay Kumar Saxena Sahab, Chairman KVIC. Special guest, Dr. Arunachalam Sahab, Director CAFRI. All the Dean, College of Horticulture and Forestry, Dr. Pandey sir, Dean College of Agriculture, Dr. Chaturvedi sahab, Director Research, A.R. Sharma sahab, Director Education, Dr. Anil Kumar sahab, Director Extension Education, Dr. S.S. Singh sir, our other university officers, head of departments, my co-colleagues, and all the participants for this webinar from the different parts of the countries, as well as all the panelists and chairmen of different sessions. I welcome you all, sir. In this formal vote of thanks, first of all, I would like to thank our Honorable Vice Chancellor who readily agreed for this webinar, and he always supportive for this kind of events, and especially Neem is very close to his heart, because he always used to tell, as he already mentioned, that we have around planted along all our boundaries around 500 kilometer this area for the Neem trees, and even in we have the separate plantation in our agroforestry farm too. And besides that, our both the neighboring institutes are also having very good surrounding of the Neems along with in the area. So this was a very good topic to have this webinar. And I also thank our Dean, Dr. Pandey Sahab, who is very much instrumental since last 6 months that we have to organize this webinar. And I'm thankful, sir, your consistent motivation made us to make this day today. And
Thank you,
there are various forums which are doing work on the Neems, but in Bundelkhand it was not prioritized, as our chief guest Vinay Saxena Sahab also mentioned. So thank you, sir, motivating us or making this seminar fruitful today. And I thank our chief guest. No doubt, due to his busy schedule, he able to find the time to address our inaugural address of this webinar. He was very instrumental in promoting the KVIC, and he made this as a good marketing strategy to make these different khadi products. And as his speech also, you might have seen that he is very eager to collaborate also, all the which are want to work in this field of the Neem, especially in Bundelkhand. So I thank Dr. Saxena Sahab who accepted our invitation, as also given his very valuable talk on the promotion of the Neem in this area, especially the mentioning about the honeybee promotion in this area with the Neem trees. Surely, sir, we already, our Honorable Vice Chancellor told that we already have started this project on honeybee under KVI, under RQI, as well as from the National Honeybee Mission. And we will definitely need your support for promoting, especially the marketing of these products in future. So, and we welcome you, sir, in RLBCAU in very near, so that we can have your more blessings in this field. Thank you, sir, again for coming this session and delivering the lecture. I also thank our guest of honor, Dr. Arunachalam sir, who is always with us in all the programs, most of which we host here. And he is, even we not make the consent of him, he will directly put your name. So we are assured, means sir will be there always, any kind of program we are organizing. Thank you, sir. And as sir told that Khafri is having one of the biggest field gene bank of the Neem. It's true. It's very near to us, 276, more than that, germplasm is there. already we will be having one lecture from the Kafri by Dr. Rajarajan, who will be mentioning about all the project of the Neem germ plant in the Kafri, as well as they also have now the mode that georeferencing and all of these germplasm. So, and Kafri also made a very good efforts in promoting the Neem in this Bundelkhand region in different agroforestry approaches. And sir, under Dr. Nachalam sir, dynamic leadership, Kafri is more fast approaching to the farmers, and they are very much to this promotion of agroforestry in Bundelkhand regions, along with this tree species of the Neem also. So I'm thank you, sir, given time and also deliberated among the importance of this Neem and how from the local is become a global product, and how we can also further strengthen it for the livelihood options of this region. Thank you, sir. I also thank all the dean directors of this university for encouraging us to this seminar, organization these seminars, and also giving their valuable inputs. I also thankful to all my colleagues and head of departments of the universities who are also supportive to us, all the faculty members and staff of the RLBCAU. I also thankful to all the panelists, all the chairmen of the different sessions who will be joining in us, Dr. A.B. Rai Saab, Dr. Nachalam Saab, Dr. Sas Biswas Saab, and Dr. Anthony Joseph Raj will be chairing the all 4 sessions. Also the panelists, Dr. R.K. Singh Saab, Dr. Saxena Saab, Dr. R.S. Yadav Saab, Dr. R. Shivana Srinivasan, Dr. Ashok Kumar, Dr. K.P. Mahapatra, Dr. Arvind Bujilwan, and Dr. Salil Tiwari Saab. And also I thank most of them are in this inaugural session. Thank you, sir, spending your valuable time for this inaugural session, and we look for your deliberation in the seminar. Also, all the speakers will be joining us: Dr. S. Satapathy, Dr. Virendra Rana, Dr. G.K. Patel from GNFC. Sir, thank you for joining us, Dr. Dr. Vanilla, Dr. K. Rajarajan, Dr. U.K. Tomar, and Dr. Pandey. And they all will be joining to us in this seminar. Along with that, all the participants join from different parts of the country, and I may see the various scientists also from different ICR institutes and other universities also join. So I thank you all for this inaugural session, and also the this webinar which will be now proceed for the technical session. So, and I wish that all will join hands to make successful this seminar by their participation, as well as their constructive suggestions for the how we can move forward, because its name is a household name since a long time, and we fought for even the patent of the name agency. So it has a diverse product, and also very much importance for the rural livelihoods. So how like all the inaugural session, they mentioned about the importance of the neem-based rural economy by promoting different products, neem herbicides and all neem-based biopesticides, neem oil extraction and all, and various byproducts from the neem, soap industry and others. So how best we can channelize not only the production but also the usable products and linking with the rural economy. So thank everybody. Thank everybody for Dr. Garima. So I thank you, sir, everyone who directly and indirectly supported us for this webinar. And I thank my both organizing secretary, Dr. R.S. Yadav, and Dr. Gaurav Sharma, along with all the team members from the reporters and all the organizing committee, because we are having tomorrow mainly focusing on the farmers, because as the Dr. Minesh Saxena have also told, it's had no meaning unless we link with the farmers, which are the main stakeholders for this. So that's why we have a special session for the farmers, especially for their training as well as for the field visits, so that we can encourage them for the neem. So thank you once again. Thanks all.
Thank Thank you, Dr. Sir. Thank you.
Thank you very much, sir, for the vote of appreciation. Ladies and gentlemen, thank you for your kind attention and patience. It is now time to attend to the wisdom of understanding. Ladies and gentlemen, we will now proceed to technical sessions. From here, I will hand over the microphone over to my colleague, Dr. Pavitra, for the theme area first of the technical session. Madam, please. Dr. Pavitra, can you unmute your mic?
Good morning, all. Welcome to the technical session of webinar on nurturing the Neem for nature and livelihood security. The theme of this session is Neem for plant health management. I, Dr. Pavitra BS, Assistant Professor, will be the moderator of this session. And this session is chaired by Dr. A.B. Rai sir. Dr. A.B. Rai sir is a former HOD, crop protection and principal scientist, entomology at ICR, Indian Institute of Vegetable Research, Varanasi since 2005. He has more than 33 years of experience in teaching and research. After completing PhD in entomology in 1985 from BHU Varanasi, he started his career as Assistant Research Scientist cum Assistant Professor in Gujarat Agriculture University, Navsari in the year 1986, and later he joined to ICR Research Complex for NEH region, Manipur, as Senior Scientist in 1999, and he moved to ICR IIPR Kanpur in the year 2003. His major area of research has been on pest management in crops of agriculture and horticulture importance. Dr. Rai sir handled several in-house and externally funded projects sponsored by ICR, DBT, NICRA, NAIP, UPCIR, and NHM. And he is responsible for developing technologies for pest management. He published more than 200 research papers in journals of national and international repute, 24 full proceeding papers, 9 review papers, 65 popular articles, 7 books, 35 book chapters, 12 technical bulletins, 8 manuals, and guided 7 MSc and 3 PhD students in entomology. He has invited delegate at 8 International Congress of Acarology in the year 1990, conducted by Academy of Science Institute of Parasitology, Brannioska. He honored with many awards like Crop Protection Research Award during 2004-2005 by Agrochemical Manufacturer Association of India, New Delhi, ICR Ganesh Shankar Vidharthi Award, and Dr. Harbhajan Singh Gold Medal Award also. He is Life Fellow of Entomological Society of India, Indian Society of Vegetable Science, Confederation of Horticulture Association of India, Indian Society of Horticulture Research and Development, Society for Recent Development in Agriculture, and Chief Editor of Vegetable Science and Member Editorial Board of Rajbhasha Patrika, Sabji Kiran, Indian Horticulture, New Delhi. Along with academic, he also worked in the administration, like he served as a in-charge plant clinic and chairman of PME cell, transparency officer and nodal officer. Sir, I welcome you to this technical session. And this technical session is the panelists of this technical session are Dr. R.P. Singh sir and Dr. S.P. Saxena sir. Dr. R.P. Singh sir presently working as a professor and head Department of Plant Pathology, GB Pant University and Pant University of Agriculture and Technology, Pantnagar. Dr. R.P. Singh sir, born on 20th January 1967, he completed his BSc Agriculture from ND University of Agriculture and Technology, Faridabad, UP, and MSc and PhD from GB Pant University of Agriculture and Technology, Pantnagar. He appointed as Assistant Professor Plant Protection, GB Pant University of Agriculture and Technology, Pantnagar in the year 2004, and he served as a Senior Scientist at National Research Center for Onion and Garlic at Pune. He has vast experience in teaching as well as research. He taught 8 undergraduate and postgraduate courses guided 6 PhD and 7 MSc students, presently working as local program coordinator, All India Coordinated Research Project on Maize, office in charge of plant clinic. He completed 28 research projects and currently handling 7 ongoing projects in the area of soil-borne plant pathogens, extension plant pathology, fungicides, maize and vegetable pathology. He developed various plant protection training modules for Krishi Vigyan Kendra, worked with resource-poor farmers for technology assessment and refinement, particularly under hill farming system. He developed biointensive integrated pest management modules for various diseases, and he organized 10 advanced training, and he delivered more than 50 radio and TV talks. He published 45 research papers, 4 books, 6 book chapters, 37 papers in proceedings, 13 technical bulletins, 45 popular articles, 54 extension bulletins, and edited 7 proceedings. He is a life member of several professional societies and fellow of Plant Protection Association of India and Indian Society of Plant Pathology. He also a member of selection board of CSIR and many scientific committee of Government of India. Sir, I welcome you, sir. And another panelist of this session is Dr. S.P. Saxena, sir, ex-professor and head and ex-principal investigator, RTVY Project, Department of Plant Protection, Faculty of Horticulture and Forestry, Navsari Agriculture University, Navsari, Gujarat. Dr. S.P. Saxena, sir, born on January 16, 1958, obtained BSc and MSc degree from University of Udaipur and joined Rajasthan Agriculture University as research associate in the year 1982 and later joined to Gujarat Agriculture University as assistant professor in the year 1988 and associate professor in Navsari Agriculture University in the year 2006. He served in many ICR projects sponsored by Government of India, AICRP, subtropical fruits, tropical fruits, and NICRA for more than 18 years. He has served as professor and head entomology and plant protection in the Faculty of Horticulture and Forestry of Navsari Agriculture University for more than 10 years. His total teaching and research and extension experience is more than. He has 2 PhD degrees, like in agriculture entomology, and another one is in forest entomology from Navsari Agriculture University and Forest Research Institute, Dehradun. He also completed MBA degree from Pondicherry University. He is having he completed 19 e-courses and published more than 255 research papers, books, book chapters, booklets, articles, and technical bulletins. And he is a life member of many scientific bodies. He has chaired many international and national seminars, symposium, and webinars, and contributed significantly to horticulture entomology and research on mango in particularly, spanning over 3 decades with emphasis on ecological studies, IPM modules, pest forecasting modules, as well as pest map and pest calendar. In forest entomology, he identified major insect pests of important tree species like tea, sesam, rosewood, Terminalia arjuna, Terminalia tomentosa, bamboo, mahua, and eucalyptus, and their natural enemies in Gujarat. He released 23 technologies for the farming and scientific communities and was also associated with release of mango hybrid Sanpari. He is a recipient of 8 awards, including prestigious Dr. APJ Abdul Kalam Award. He handled the first Atma project at Valsad district as project coordinator, 10 projects as principal investigator, and he also worked as a nodal officer and course director of winter school sponsored by ICR. He is a member of editorial boards of several journals. He has guided 13 scholars in agriculture entomology and 6 in forest entomology. He also organized the first ever National Symposium of Pest and Disease at Navsari Agriculture University, Navsari, Gujarat. Sir, I welcome you to this technical session, sir.
Thank you.
Now I request my colleague Dr. Usha ma'am to introduce our speaker, Dr. S. Satpathy, sir.
Good morning to all. I, Dr. Usha, welcome you all in this seminar So I would like to introduce our speaker, Dr. Satpathy sir. Dr. Satpathy sir is working as a head in Division of Crop Protection in ICR, Central Research Institute for Jute and Allied Fiber, Barakpur. He has more than 25 research experience in IPM of major insect pest of vegetable and fiber crops. Dr. Satyapathy started his career as scientist in ICR, IVRI, Varanasi in 1993. During his research career, he was associated with collaborative research projects supported by DFID UK, World Vegetable Center, Taiwan, DBT, UPCAR, and ICR also. He standardized and promoted innovative low-cost IPM technology like Chinese cabbage as trap crop for DBM, bait application, and male inhalation technology in cucurbit fruit fly, and seed treatment-based preventive control for kharif pests. He was conferred with the International IPM Recognition Award in 2009 during IPM International Symposium in USA for his outstanding contribution as member of the team in the development and promotion of pheromone-based IPM technology in management of brinjal fruit and shoot borer in Southeast Asia. Besides this, Dr. Satyapathy is also the recipient of Outstanding Team Research Award of ICR, Dr. Harbhajan Singh Award, PP Singhal Memorial Award, Best Scientist Award of ICR IVRI, Varanasi. He is Fellow of Plant Protection Association of India, Fellow of Society of Plant Protection Sciences, and Fellow of Indian Society of Vegetable Sciences. The technology, methodology, and concept developed by Dr. Satyapathy have been published as 35 research papers in international journals, 115 research papers in Indian journals, 20 technical bulletins, 2 edited books, 38 book chapters, which are widely referred by the researcher. So now I welcome Dr. Satyapathy sir and invited the Satyapathy sir for share his view on the neem use as a plant protection. Welcome, sir.
Thank you, Dr. Usha. Am I audible?
Yes, sir.
Hello.
Yes, sir.
Am I audible?
Yes.
Yeah.
You are well audible,
Chairman.
sir.
Yeah. Respected Chairman, Dr. A.B. Rai, sir. The panelists, Dr. R.P. Singh, Dr. S.P. Saxena, all the deans of Rani Lakshmibai Central Agricultural University, senior faculty members, all the students and esteemed participants, those who are joining this webinar. Actually, I am going to present your webinar on neem for plant health management, the status and prospects. First of all, I want to congratulate the organizing committee because the theme which they have selected for this webinar, it is very much pertinent to the time as right now the country is thinking to go for organic farming, and our Prime Minister's vision is to go for the natural farming, and that way this neem products, particularly in neem plant health management and other crop husbandry practices, particularly fertilization, the neem products is going to play very pivotal role to support the production and productivity of the horticultural and field crops. So I am going to share my slides. Please wait. Yes, I have to understand. Yeah. Can you see my slide? Hello. Hello.
Yeah, it is visible, sir. It is visible.
Yeah. The first slide I want to just tell you about the alarming pesticide consumption and its consequences throughout the world. If you see that now the whole agriculture is gradually becoming the pesticide centric, because the farmers and all the stakeholders and other the officers, those who are involved in scaling up this IPM, they are depending more on pesticides. And right now the global pesticide use has gone up to almost 2 million tons, and out of which almost 30% are insecticides and fungicides. In India also, the active ingredient of about 56,000 ton is being used in per year. And if you see the top pesticide consuming states are China is the first in the world, and our position is in India is almost 10th. But the main problem in India is that we are not going to care it use cares it use, means we are going, most of the farmers, they go for misuse or overuse of the pesticides for preventing the pests and diseases in their crops. And the consequences is very much clear as in terms of resurgence, whether it may be it is phytotoxicity, hazards, and poisoning. In my photograph, you can see that how much this phytotoxicity is there in a chili plant. Chili plant, its leaves had already converted to tendril like shapes. So these are the consequences of overuse of the pesticides, means phytotoxicity, hazards, and poisoning, and millions of people, they become the victim of the insecticidal poisoning throughout the world because of their exposure during the application of pesticides or during the consumption of the foods like particularly vegetable and fruits. And as far the most alarming thing is that you can see that how many species of insects species, they are becoming resistant, and that is indirectly also increasing the repeated use of pesticides, and that way also the consumption of pesticide is going up globally. So almost 586 species, they have become almost resistant, particularly the white flies and mites. And you can see this, and the main lepidopteran pests also. And this resistance has become almost against 325 insecticides and 5 insecticidal traits. Means in some cases, this particularly against BT, and although the GMO also, this genetically modified crops also, they have become, the insect has become resistance against genetically modified crops also having trait of insect resistance. So as I told that what are the alternate to this under this situation when the government is also thinking, so the in India we are very much lucky that we have a reservoir of neem trees in various states of our country. And the neem, it is known as all of us know that this is a wonder tree, and you can also you can talk it as a village pharmacy, because this neem has very multifarious uses, and from all the plant parts of neem that can be used for different agricultural and medicinal purpose. And you can see all the flowers, leaf, seeds, oil, and bark, they contain very useful components and active ingredients which can be used as a fertilizer, as a pesticides, or they can be used as a, in medicine also. Anti cancerous material can be developed. Insecticidal, they act as antifidant, larvicidal effect, antimicrobial effect. So all sort of this neem tree, not only the only single part, the whole plant parts of the neem, they produced a very effective combined multiferous effect, and which is very useful to fight against the diseases and pests of agricultural crops and the human diseases also. And besides, they are very good soil, they can play a very vital role in reclamation of soil, maintaining the pH and microbial diversity in soil if they are used as a neem cake. That is another way that we can sustain our productivity by reducing the nitrogenous fertilizer and other chemical fertilizers. So that way, this is a complete package, this neem tree, and which play a very important role in subsiding the use of pesticides and chemical fertilizers. If you see the neem as an alternative to pesticides, the their distribution, you can see that particularly in Central Asia, India, and also Central America, they are this Agarica indica species is distributed almost throughout the world, particularly in tropical and to some extent up to the subtropical region of the world. And India, except the sub-Himalayan region, those states, this neem trees are they are present almost throughout the country. The green patches, it shows that distribution of the neem plants in our country. And a rough approximate estimate indicates that almost 1.5 crores neem trees are available in India, and that the seed production potential is almost 4.42 lakh ton, and the oil potential is almost 88,000 tons. So you can think of that when we don't have any type of regulatory mechanism or any type of systematic collection and use of the neem product, particularly the seed kernel. But still we can scale up this production in our country if we go in a very systematic manner to collect the neem seeds and other neem parts of the neem tree, and use and can go for their use in a very effective manner. As far as this throughout the world, if you see this as it is distributed in many countries, like from Iran to Thailand, and the oil content, particularly the azadirachtin content, because there is the main ingredient of the neem which plays the pesticidal role and anti-feral and anti-physiological role on insects and other fungal pathogens. So this azadirachtin content, it varies almost double. You can see this this species from Iran, it contain only 2.75% azadirachtin, whereas in Myanmar it is 6.10. In some of the literature, it shows that it is originated in India-Myanmar border. There is a center of origin. That's why this azadirachtin content in Myanmar and in India, it is almost same, and it is the highest. In Thailand also it is very high, 5.2. In India it is 5.14, and Myanmar it is 6.10. Whether in Iran it is 2.75% only. Side by side, this oil content, it also varies from almost 45 to 50%, and its variation is less. And in India it is almost 47.6%, and Myanmar it is 48. Whereas in the Sri Lankan species, they contain more amount of oil, that is almost 50% oil content has been reported from Sri Lanka. And if we go for, if you see the data on the azadirachtin content in India, actually, in my table it is wrong. I have written it in percentage. It is in milligram, I think milligram per gram. But in my pie diagram, it is written as content. This is correct. And here you can see that in this among the states also, the azadirachtin content, it varies a lot. Particularly in case of states like Odisha, it is very high, 0.45%, and in states like Punjab and in Uttar Pradesh, the content is quite less, and in Karnataka it is almost 0.35%. So what can be summarized from the slides is that this distribution of neem trees, it is almost global distribution is there, but the major concern is that this azadirachtin content and the oil content, it depends on the edaphic condition, means the soil condition, agroclimatic condition, and that regulates the content of azadirachtin and oil content in neem. So if you see that, we know that this neem has a very multifarious effect, and particularly antioxidant effect, and is more prominent among various studies conducted throughout the world. So if you see the sensitivity of the insect in terms of their effective dose, ED50, that is your for feeding inhibition, then you can see that it varies. One way that this neem plant, it varies in terms of azadirachtin and oil content. In another way, the pests, the different orders, the pests belonging to different orders, they also respond differently to the neem. And particularly one study was conducted on effective dose for feeding inhibition. You can see this Lepidopteran, because most of the pests, they belong to Lepidoptera and Hemiptera, whereas the Hymenoptera, most of the natural enemies, they belong to Hymenoptera. But one good thing is that particularly the Lepidopteran, they are more sensitive, because this feeding inhibition is achieved in a quite very low concentration compared to the Hymenopteran, because in the most of the natural enemies, they belong to the Hymenoptera order. That is one thing. And another important thing from another important information from this table, very interesting, that is Orthoptera, because most of our desert locust, they belongs to this Orthoptera. And because the Orthopteran species, they very significantly, means some of the species, they are quite they are very much sensitive for anti-feeding effect, and some they are not at all. They get affected because by this neem product. So that's why here this range is very high. Later on, I will tell you that because of this chemosensory mechanism of the orthopterans, few insects, they never sense this anti-feeding effect and the deterrence effect of the neem. So that it indicates that Orthoptera show a very enormous range, whereas Lepidoptera, they are most sensitive to the neem product as far as this anti-feeding effect is concerned. Then if we just look into that how the insect order-wise susceptible species of insect species, and you can see that this is the major parts, because which are being very much susceptible. So you can see this Lepidopteran. Here also this major group is Lepidopteran, Coleopteran, and here also you can see the Hymenopteran. Very least number of Hymenopteran species, they are sensitive to the insects. Means different species of in a order. Now, as you know that this neem it contains a plethora of active ingredients. But the most important among these ingredients is azadirachtin, which is a complex triterpenoid from neem seeds, and it has a heavy, very anti-feedant and toxic effect on insects. Particularly this neem species, it produces a large number of terpenoids, and besides this azadirachtin, neem seed extract also contain many related triterpenoid like nimbin and salanin. And this natural mixture of azadirachtin is neem in neem, because whenever we see that what is the theory of pests becoming resistant against insecticide, because of the use of single type of chemical group against against the same type of insect like mites or whitefly, then those chemicals, they put selection pressure to great extent, and the population becomes resistant, which takes more time, because you will not believe that now the reports are there that one Myzus persicii population, that is showing resistance against neem after 7th or 9th generation. That I will tell in later on. But here we can see that why this neem takes more time, or neem is lesser susceptible to become insecticide resistant than chemical insecticides, because it contains a good number of it is a mixture of different your active ingredients which act differently on the pest, and that's why it takes more time for development of resistance against the neem product. So as I already told that neem oil, it has medicinal application, agricultural application, and different other your terpenoids which are available in neem. They act on differently, like antimicrobial, anti-feedant, and other effects on human being and insects. The most important effect of azadirachtin on insect is the growth regulation, because it has shown that this physiological effect which is imparted by the neem, particularly this main constitution, that is azadirachtin, it is more consistent than the anti-feedant effect, because this anti-feedant effect, it is regulated by the chemosensory mechanism of the insects, and which varies from order to order. That's why this antioxidant effect is less consistent than your consistent than the other physiological effects like growth regulatory effect. So this all these neem different components of neem, they interfere with growth, particularly the molting, reproduction, cellular processes, and it also disrupt the endocrine system. And that way these insects, they fail to go for molting, and their total life cycle is disrupted, and the population, it goes down. So if we saw the effect of this insect growth regulation effect of azadirachtin on this effect of azadirachtin on insect growth regulation, then you can see that it can affect the pest in different way, particularly it interfere in molting, it causes mortality also, and it abnormal molt also. So so here it indicates that as this dose increases, then this abnormal molting, proportion of abnormal molting in the population, it increases. So here it is being used from 0.00015 to 0.015 microgram per gram of azadirachtin. It was applied on the grass weed bug. So this is just to show that how this growth regulatory effect acts on different insects by azadirachtin. This overall effect of azadirachtin, if you see that this primary antioxidancy, it occurs in mouth parts and other chemoreceptors, as I was discussing in case of locusts. So it is deterrent, and it is deterrent cell stimulation, sugar cell inhibition, and in gut also it affects the peristalsis movement of the inside the gut, and reduce also this enzyme production of the digestive enzymes produced by the insect. And in cuticle, it is we all know, it is well established this alternative to ecdysteroids and juvenile hormone titers by blockage of the morphogenetic peptides causing molting defects. And it also causes sterility. So the insect, they fail to produce the viable eggs. And the cellular process, it block the cell division, loss of muscle tone. That's why the insects, they fail to to move their mouth parts and cause the damage, a blockage of digestive enzyme, and the protein synthesis inhibition in various tissues also occurs because of this azadirachtin. So there are various compounds of azadirachtin and other alkaloids which are present in neem, and neem extraction is very important because this insecticidal property, it depends a lot on the method used for the neem extraction. Particularly these in this figure, the major neem extraction procedures have been given. So if we go for those type of procedures, then this retention of the active ingredient is more, and this product which is developed from those type of extraction procedure, they are become more effective against the target pests. So here, if you the neem seed kernel powder, aqueous extract, you can see this range of different azadirachtin components. Particularly azadirachtin A is the major component, and another is azadirachtin. But this effect of azadirachtin A is more compared to the other components which are present in the neem. So as you know that although in market there are many finished products which are available by different companies which are marketed and used by the farmers, but in farmer who the homemade NSKE, it is very predominantly researched and used by the farmers, because in most of the literature you will find this mention of neem seed kernel extract 4% or 5% or 3%. So this is the very crude way of preparing the neem seed kernel. Neem seed kernel in the household itself. As in my distribution, I have told that almost in all states. Huh? Okay. Because this it is predominantly used in village level also. So here you can see that this neem seed kernel extract preparation is a very simple method, because these neem trees are distributed in many states of our India, particularly UP. In UP, the number of neem trees is highest after Tamil Nadu. So in village areas, these neem trees they are predominantly available, and the kernel and seeds they are also available. So the first thing is that after getting the neem seeds from the tree, those should be dried properly under shade, not in sun. And then after drying, they may be, they are to be crushed to very small particles, and those powders they can be kept soaked overnight. These crushed neem seed kernel, five for if you are going to prepare 5%, then we'll have to dip 500 gram in 10 liter of water, and by binding it inside a muslin cloth bag, and that bag is to be soaked overnight for 24 hours. And just after shaking, you can remove those that bag containing those crushed neem seed kernel, so that the smaller particles they will not flow into the water, and then it will be useful to make a clean neem seed kernel extract. And for precaution again, that can be filtered, so that these larger particles there can be removed, and to avoid the choking of the nozzle of the sprayer. After that, this spreader and sticker can be used. Many household spreader stickers are can be used, but nowadays commercial spreader stickers are available in the market, those which are very of low cost, that can be added, and then this solution can be very effectively applied as a insecticide in the against the target pests. So this is the way that how this neem seed kernel extract can be prepared. But actually, this insecticidal use of neem products is it started in the late 60s, when one German scientist group they started working on the insecticidal properties of neem. But I'm happy to share that our IARI they played very important role in 1962 in which they developed they reported the active ingredient, and also they could successfully apply this neem seed kernel extract against locust control in Delhi. And now it is almost worldwide, more than it is used against more than 500 pest species are being controlled through neem have been researched on 500 pest species. And the multiple effect against insects, repellent, antipherent, oviposition, as I already told that it act. So that's why many scientists they have tried different neem products in the field itself. And you can see how the major pest, major crop and rice, and they are these important insects like brown plant hopper, yellow stem borer, gandhi bug, seed blight, and bacterial leaf blight. Those can be controlled by application of nimbicidine, multi-neem spray of neem oil, NSKE, and soil application at the rate of 25 kg per hectare. The another, there are many examples of the pest control by application of neems. You can see in maize also, this corn earworm, it can be effectively used by the aqueous neem extract. The most importantly, very recently introduced this fall armyworm in different parts of our maize growing states. You can see that neem has shown to effect to some extent, like application of neem oil at the rate of 4 ml per hectare, and also the seed cake at 50 kg per hectare. These are effective in reducing the borers and other fall armyworm in maize. And in against wheat aphid also, this one product marketed as endoneem, it has this is a concentrate of 1500 ppm azadirachtin that has been found to be very effective, and neem oil also. And particularly in vegetable, I want to say that very safe. This one good thing is that we always want to go for the good agricultural practices in vegetables to reduce the residues of the pesticides. And particularly in case of vegetable, you can go for neem seed kernel extract up to 5% without any phytotoxicity. Some sort of phytotoxicity have been seen in case of okra if we go for more than 3%. Otherwise, particularly these crucifers and other solanaceous vegetables, they are very safe in terms of their phytotoxicity of the neem product and also NSK. And in cabbage, very safely we can go up to the NSK 5%, multineem 3.5 ml to control the DBM, particularly in the primordial stage of the crop. And in case of cauliflower also, early cauliflower, this Sporoptera is a problem. There also the farmer can go for the spray of neem oil and NSK. Actually, what I want to say that most of the cases we are applying in neem oil, the scheduling, the application timing, decision-making procedure, just like insecticides. But if we want to get more effect of the neem products, we have to go for smart application of neem products on the basis of very strong monitoring and it as a part of the integrated pest management that we'll discuss in short later on. And in case of eggplant also, this Leucinodes urbalis neem seed kernel extract and the neem based product. Actually, we used the many plants like your coriander and dill to repel the moth of brinjal shoot and food borer to lay egg on the plants. And it has been seen that if neem is used along with the repellent crop, then the efficacy of both neem and the repellent crop it becomes additive effect, and the extent of control mechanism is also improved. So in case of root knot nematode, particularly we are most of the cases we are going for now the protected cultivation, particularly in our hills in Uttarakhand, in northeastern states, and also in Sikkim. So in Himachal, we are going for floriculture and production of vegetables in your poly houses or net houses. So they are this when we use the same soil for multiple times, particularly this prevalence of root knot nematode has become very much prevalent. And this in nursery bed, if you go for the application of neem product and neem cake also, it prevents the root knot nematode in a very effective manner. And in one study, leaf miner can be controlled by if it used in a ratio of 1:1 with abamectin, then it is very effectively it controls the leaf miner. In okra also, fruit and shoot borer and root knot nematode. These can be controlled by the various neem products like NSK or multineem. In Chile, actually, one of our ICR institutes, that is ICR, Indian Institute of Horticulture Research, Bangalore, they have developed some neem product like one minute.
हेलो सर अभी गोवा को फोन किए थे उस समय नहीं आया था, अभी एक मीटिंग में होता तो वेबिनार में 10 मिनट में खाली हो जाऊंगा जी सर। हाँ मैं गोवा को बोल के रखा हूँ सर, चाय बना के जी सर घुमा के चाय पिला देंगे। ओके हाँ ओके सर ओके हाँ सर हाँ सर हाँ ठीक है मैं प्रेजेंटेशन में हूँ सर जी जी हाँ हाँ सर मैं प्रेजेंटेशन कर रहा था 5 मिनट में खाली ओके हाँ सर हाँ
Hello, sorry, sorry, Chairman sir. And am I audible? Hello.
Yes, sir. Yes, audible, sir.
Yeah. Yes, sir. Yes, sir. Actually, as I was telling that Indian Institute of Horticulture Research, Bangalore, and they have developed one neem soap, and that neem soap is very much effective against chili thrips and other sucking pest and other horticultural crops in vegetables. So there are many examples which I don't want to go in detail, but the main thing that how we can go for use of these neem products against various target pests and crops, that to increase its efficacy, that I'll discuss later. And now coming into the other part, because selectivity, how much these neem products they are selective against the parasitoid and predators, because this in any crop ecosystem, they have a very good niche of parasitoid and predators, which combinedly take care of our the insect population and keep them below the economic threshold level. So there are many studies which have been conducted, and I have listed some of the very important parasitoids, particularly this trichogramma, bracken, and diadegma semiclosum and cotesia. These are also very much prevalent in crucifers and tetrasticus. You can see these are the very important group of parasitoids, and various concentration of neem and other products has been tested against these. And you can find here that most of the parasitoids which are marked green, those have been found to be have a safe effect, a positive effect, means they are least affected when they are tested against these concentrations against them. This indicates that most of the parasitoids, they are have least negative effect of these neem products. Now coming into the predators and entomopathogens, because these are also part of the various biological control system. And Cytohinus lividipanis, as we know, this is a very effective predator of brown plant hopper in paddy ecosystem. And other like Chilonomus, Chrysopala, and Lycosa, this spider. So these are the very important predators. You can see most of the predators which are prevalent in major crop ecosystem, they are not affected by these neem products. So this is these studies indicate that unlike insecticides, most of the neem products, if they are used in proper concentration, then they are mostly safe to the predators. And coming to the entomopathogens, all the three, they are the fungal entomopathogens, the metarhizium, verticillium, and beauveria. Except metarhizium, these neem products has been found to have positive effect on verticillium, licani, and beauveria bassiana. I want to mention that positive means they don't encourage, later they don't affect their population. So this should be taken in that way, that they are safe, and negative means they affect their population in adverse way. And particularly the entomopathogenic nematode, they have mixed effect of the both negative and positive effect of various neem products. Now as we know that there are these neem products have been researched in a very for last 50-60 years, and now we know that there are certain limitations that hinder their effective large scale use. And what are those limitations? As we know that low stability, like pyrethrum and other botanicals, they are very much sensitive to the UV exposure. So this low stability under field condition mainly due to the high rate of photodegradation, it reduces their effective insecticidal activity in field condition. Then the short residence time and very slow killing rates, unlike insecticides, so which have very strong knockdown effect on the insects. And these genetic factors, as I already mentioned, that if you see the neem azadirachtin and oil content in Punjab neem population, then it is very less compared to the other parts of the country. And however, the environmental factors and the type of extraction methods can lead to significant difference in their composition. And the initial report of resistance, as I was mentioning in my earlier in one slide, that this aphid Myzus persicki, it has shown after a ninth generation continuous exposure to neem, that it is showing a bit of resistance against the neem product. Azadirachtin is less stable to heat, light, and water, that I have already mentioned. So what are the precautions that if we use in neem product, then we have to avoid the photo degradation. For that, there are many sunscreen like para-aminobenzoic acid. Those can be added to reduce the adverse effect of the UV light. And the azadirachtin is more effective when formulated as oil than that contains other natural products of the plant. And particularly as in the left side table, I already mentioned that this the way these neem products are being extracted, it regulates the efficacy of the neem product. So that way this methanolic and ethanolic extracts, they are their active ingredient are better than the water, and their integrated use with the IPM components and improved nano formulation. This why I am telling this integrated use with IPM components. I will mention that if particularly we are going for mat application, because male annihilation technique you might be knowing, which is used for the control of cucurbitaceous fruit fly and mango fruit fly also. So if we go for male annihilation technique and bait application technique, along with that, if we go for the application of neem products in the plant, then it enhance the index of attraction to the bait points, and that way it increase the control efficiency of the product. And the most important another thing is that this improved nano formulation, because formulation is a major problem in case of neem to increase its efficiency under field condition. Now there are many studies, many theses they are coming up on nano formulation of neem. So this nano formulation, as you know, they are advantageous in a many way. So these are the things that this biotechnological interference, it can increase it has great potential, which can improve the formulation technology of neem. And this azadirachtin can be loaded into both organic nanoparticles and as well as an inorganic nanoparticles, particularly for mosquito control also. This neem nano pesticides have been worked out, and the improvement of physicochemical stability, reduction in degradability, and the effect of these natural product can be enhanced through nanotechnology. And these nanocapsules provide slow control and sustained release of active compounds. It is the most important thing. And it also reduces the loss of volatile compounds. Their photo degradability can be improved. And as the literature shows that this nano encapsulation can be done, there are many attempts have been done. Various neem components have been taken up, and various carriers have been used for the nano formulation. And it shows that particularly with silica also, silica itself also, it act as it act against the insects, particularly this lepidopteran. And you can see that there are many good your reports that it in case of this polycarpolactone using carrier, this nano formulation, it almost only 100% larval mortality against Plutella. Against this tomato tuta absoluta also, this efficacy have been improved. And that way this nano formulation are the future of the neem products in coming days. And conclusion, you can see that as I have shown, that adoption of alternative strategies with special emphasis on IPM. And over the years, numerous research has validated the pesticide activity of neem. So it's upscaling in rural areas, particularly in farmers level, it is very important, so that the farmers can go for the integrated pest management by using very low cost products which they can prepare in their household. And another important thing is amalgamation of nanoscience, as I already discussed, that in incorporating the organic nanocapsules to provide dual benefit of controlled delivery and biodegradability and improved application technology. This is very important, application technology in terms of appliances so that can produce very small spray droplets, as well as the scheduling and its integration with other insecticidal approach which we are going to adopt in case of IPM. That is also very important. So if we think of neem products in a very combined way with all these things, what I hope that in future we can improve the efficiency of neem, and we can convince and bring it to the farmers level in a more effective manner. Thank you. Thank you.
Thanks.
Thank you very much, sir. for your elaborative lecture on Neem in plant health management. You covered many aspects like composition of Neem, mode of action of Neem, and the target group of insects against Neem pesticides. And you also mentioned about the compatibility with the parasitoids, predators, and entomopathogens, and precautions to increase the efficacy of Neem and advantages of Neem in nano formulations. So it will be useful for both faculties as well as the students, those who want to work in this area. So thank you very much, sir. Now I request Dr. Usha ma'am to introduce one of our other faculty, Dr. Virendra S. Rana, sir.
Thank you so much, Dr. Pavitra. Now I would like to introduce our next speaker, Dr. V.S. Rana, sir.
Usha, Dr. Usha.
Please.
Just the remarks from the presentation for panelists and chairman can proceed.
Okay, sir. Okay, sir.
Hello.
Dr. Pavitra.
Sir.
Remarks from the chairman and panelists, and then proceed for next. No?
Okay, sir. Okay, sir. Sir, now I request Dr. Rai, sir, to give his remark regarding this lecture.
Okay, thank you. First of all, I thank the organizer, particularly the Vice Chancellor of the esteemed university, as well as the Dean, faculty of this A.K. Pandey, sir, for providing me the opportunity to chair the session. So now, if you go by the presentation of Dr. Satpathy, his presentation deserves appreciations. He has covered a lot of things on Neem products and their compatibility and their safety in the control of the pest populations. So now, if you see, it is compatible with the this entomopathogens, or even now you can see here in the recent studies that has been made, particularly this neem oil, if it is mixed with the entomopathogens, it not only increase the, this is also having a synergistic effect, and it is causing, that has been proved particularly in case of the sucking pests which are being controlled in the vegetables particularly. And the LT50 times also gets reduced because of the mixture of half of the oils, neem oil, that is, if it is 5%, 1:1 ratio, even the load of the entomopathogens can also be reduced to half. So for that, that will be a safer use of even the pesticide load also. Now you see, in case of imidacloprid, if it is neem oil is mixed, so it also enhances the efficacy of this this neem oil along with the imidacloprid. That too, if you are reducing to the half to the imidacloprid. Now, if you see, despite many promising properties of neem that has been elucidated, still there are limitations that hinders the effective large-scale use of neem. So now one of the main problems that is facing the commercial development of neem is that is a lack of industrial interest, largely due to the difficulty of patenting natural products, as well as shortage of scientific evidence to support the claim regarding the benefit of this uses of this neem. As a result, this product is not widely publicized in the farming communities. So for that, the very good cover Satpathy has presented. But the point is that we have to, although he is talking about the nano formulations and all, nano particles, this nano encapsulation of neem oil that has been doing well against the pest and diseases, but with the cautions that their environmental effect or the adverse effect on the environment and their dissipation, etc., can be taken into considerations. So while considering this neem, particularly this azadirachtin. Now you see azadirachtin 1.2% EC. that is, there is no restriction. If it goes beyond that, 1.2% formulations, if it is available, then it is a 1 to 2% means that is a 20,000 ppm. 1% that is a 10,000 ppm. So it can be used without restriction even at the harvesting stage of the crop. And now, while this lauded in most of the world, particularly in case of neem and all. So recently, currently their neem oil particularly, their use has been banned in Canada. Particularly in this report was 25th July 2021. And that is also neem oil that has been banned because it is having the adverse effect on the organ of human beings. So that is also there. But then what Satpati was telling, unless you have to have, everything is bad if it is not being used properly. Beyond the proper limit, everything is bad. So with the caution that whenever neem oil means when to use it, so the time of this one, when to use, suppose, say, for example, neem oil, it is phytotoxic also. If it is a recently planted or seedlings and all, if you are putting, so it will be toxic to that. So that has to be avoided. Means while applying this neem oil or the, particularly in case of neem seed kernel extract. So although he has pointed out that there is a development of resistance in Myzus persicae, particularly in case of aphid. So now you see, so earlier the people used to assume that since the neem seed is having a hundreds of biological active compounds, so that because of this, the people used to assume that there will not be a development of the resistance. So because of this photosensitivity, volatilization, and less, I mean, this is short lifetime. So because of that, that you have to use the neem seed kernel extract or even the neem oil frequently. So there is a chances that there may be a development of resistance. So now, as per record, this Myzus persicae has come out to be a resistance against the neem products. So there may be a chances with the caution that this formulations and other things should be taken care of. So as to avoid the frequent use or the frequent interval use of the neem products. So these are some of the points. So actually we have to highlight what are the gaps and obstacles which are hindrance in the potential use of neem for the pest management. So these are the gaps. I mean, particularly this formulations development. It has been pointed out by the Satpati, Dr. Satpati, so that the formulation has to be given much more importance to have a efficacy of that products, and then persistence, as well as that you can get a longer duration for the effectiveness of the neem product in the plant system. Thank you. Thank you very much.
Thank you very much, sir, for sharing your deep knowledge about the neem. So now I request our panelists to share their view about this nature.
Hello. Hello.
Yes.
Sir.
Yes. Hello.
Hello. Saxena, sir. Sir.
Hello.
Meanwhile, RP Singh sir.
Can you hear me?
Yes, sir, please.
Honorable Dr. Rai, the chairman of the session, and my fellow panelist, Dr. Saxena, Dr. Satpathy, Dr. Rana, and other dignitaries on the panel, and dear participants. Dr. Satpathy has covered a very wide area, right from the need of the botanicals to the limitations and the possible suggestions how its use can be promoted. 2-3 things basically I would like to focus. Particularly if you see the first aspect that if you want to promote any biopesticide or particularly the botanicals in our country, it has to be registered with the Central Insecticide Board under the Insecticide Act 1968. And as far as this act is concerned, so there is no difference between the pesticide and botanicals or biopesticides. So as on date, there is no separate clause for registration of the botanicals or biopesticides. So there is one legal aspect. If we have to promote the botanicals and particularly the neem products, so there must be a separate clause or there must be a separate law for that registration and promotion. This is the number one aspect. Second one, if you see the government support, in particularly if you take the states like Uttarakhand, where state government is providing 50% subsidy, be it a pesticide or biopesticide or botanicals, it is equal. So if you have to promote the integrated pest management or if you have to promote the sustainable use of the pest management, so then there should be a on the botanicals or particularly the neem products and the biopesticide. This is the number one. This is that I wanted to highlight here. So policy issue is very, very number one point. Second one, as Dr. Satpathy has pointed out, that was new to me at least, that even if we are taking the seed kernel grown over various states like Odisha and UP, there will be great difference in the content of the IL as well as the agent active. So this is not our area, but I feel that this is the one very, very important aspect that should be dealt in the genetic session that is going to be discussed in the afternoon, where the genetic issues will be, genetics or breeding issues will be dealt with. How? Because if a particular industry is situated in Punjab or UP or in the North India, it has to
म्यूट हो गया आरपी सर म्यूट हो गया yourself muted sir
okay आवाज आ रही मेरी am I audible
yes sir
the issue that Dr. Satpathy has pointed out, that with respect to the variation in the IL content and azadirachtin content in the seeds that is produced across the state, as he said that the seed that is grown in or harvested in UP and Punjab has the less concentration as compared to the from South or Odisha. So that is again, that is the one issue that must be dealt with our breeders. This issue is very, very important because the local, because any cottage industry or even any industry has to procure its raw material from the nearby area. Okay, so if any industry is present in UP or Punjab, it is not possible for them to bring the material or procure the material from Odisha because it will have the large transportation cost. So I feel this is the very important issue that is to be highlighted, and this is to be discussed in the afternoon session, that is with respect to the genetic improvement. Second point is that is very, very important, and that Dr. Satpathy has also mentioned also, but I would like to put more emphasis on that one, that is the integrated use of the neem products, be it a neem oil or NSKE or the neem cake, along with the other management tactics for pest management. And for that, compatibility is very, very important. And as we have done at Pantnagar, and Dr. Satpathy has also mentioned that it is compatible with the Beveria, and it is compatible with the Bertram lectini. But there are a large number of parasite and parasitoids and the entomopathogens are there. As he has rightly pointed out, that it is incompatible with the metarhizium. So that compatibility issue is need further investigation, not only with respect to the entomopathogens, but almost all the bioagents, be it a biofertilizer like Azotobacter or Rhizobium or others, that is being now promoted at the part of the sustainable agriculture or in the form of natural farming. So compatibility issues is very, very important aspect, and it should be dealt with respect to the be it a fungal antagonist, bacterial antagonist, or biofertilizer besides the entomopathogens. That is to be dealt. Second one, with respect to the stability. Again, it is very, very important aspect. I'm not talking about the other things, because if you have to talk about the sustainable terms, then NSKE and neem cake has the greater role as compared to the neem oil or its commercial formulation. But still we don't have any information how long we can store the neem seeds without any further deterioration, and what will be the fate of the neem cakes if you are going to store for a longer duration. So deterioration in the content or deterioration in the quality of these raw products is very, very important aspect, and that is again, I feel this is the one gray area where not much work has been done. Third one, when we talk about the quality control, basically we are talking quality control in respect of the commercial product that is being sold in the market. But with respect to the raw material, or even like neem oil cake, many times we are purchasing the neem oil cake from the market, but its quality is very poor, and many times it is molds. So this is again very, very important aspect. You have to promote the neem. So these quality issues need to be discussed in detail, or is to be worked out, and there's must be specification for these also. And the last point that again I want to highlight, and that is field application or field use. If you see the literature, lot of information is available on the efficacy of these neem products under the in vitro condition. But in spite of that, why it is not being used at the field scale? Why its field performance is not good? Why farmers are not preferring? So this limitation is to be addressed. And what I feel that field testing needs much more precise formulation of our research program or research planning. Just I would like to point one aspect, particularly with respect to the plant diseases. If you see, there is a lot of paper that have been published in many research journals with respect to the use of the neem leaf extract with most of the fungi. There are so many. It is not only Alternaria, Coletotrichum, Fusarium. There are several fungi that has been shown that they are sensitive to the leaf extract. But whenever we are going at the field scale, so either its concentration needs very high. Many times it is more than 25%. And if you are going for the higher concentration, then it is whether this much raw material is available, whether it is going to be safe with respect to the phytotoxicity. So that is our major concern. Second one, what is its nature? How it is going to provide the protection? Mostly these neem products are basically protective in nature. So if you are applying prior to infection or just after the infection has taken place, then it is going to provide the good performance. But its curative action is absent. So if you are going to apply the these pesticides or these neem products, once the disease is established already in the plant, then it is ineffective. So this thing is to be promoted at our individual, particularly the farmers and particularly the extension specialists, those who are promoting or those who are responsible for promoting these bioproducts. They need to be educated that if you are using it as a protectant, if you are using it as a protective fungicides, then it is equally good, but its curative action is not there. So don't use it once the disease pressure is very high or once your pathogen is already established. And last one that is very important, that is the toxicity. Many times we have seen the toxicity, particularly when we are going to promote the use of oil cakes, particularly in the vegetables and during the nurseries. So that is again very, very important aspect, because the different crops has the different sensitivity level. In one crop or in one vegetable nursery, it can tolerate the high dose, but in another at the same dose you will get the toxicity symptoms. So again, this is the very tricky one, and again it has to be worked out in detail. What are the crops for which what should be the dose? It can't be a blanket dose that you can go for 1 to 2 quintal per acre, or it is the 2 to 3 kg per meter square in the nursery bed. So it should be a crop specific recommendation, keeping in mind the sensitivity of that particular crop to the neem products. Thank you so much.
Thank you very much, sir, for sharing your remarks. And you have cleared many of our doubts, like why farmers are not preferring these neem pesticides and all. So actually these questions, we are also having same questions. So thank you very much, sir. And now I request one of our another panelist, Dr. SP Saxena, sir, to share his view.
First of all, I'm thankful to the organizers of the webinar and authorities of RLBCAU for inviting me as a panelist. The talk delivered by Dr. Satpathy was very diverse, and it touched almost all the aspects of neem application, its chemistry, its biochemical components, its toxicity, but I have to say some of the points. First, in one of his slides, he showed comparative efficacy of different insect orders or susceptibility of different insect orders against the neem or azadirachtin or neem components. Here, what I want to say is, in case of Isoptera, the total number of fauna or the species diversity is very less as compared to the other insect orders. So we can't say that the members of the Isoptera or Isopterans are less susceptible against the neem and or azadirachtin or its products. Firstly, secondly, those sucking pests, insect pests, which are carriers of virus diseases or which transmit virus disease, the efficacy of neem or neem products is reportedly less. I am emphasizing those insect pests which are carriers of virus, not all the sucking pests. And thirdly, the residual toxicity. If we compare the residual toxicity in foliar applications, it never exceeds 7 days, 8 days, or in soil application. Whereas in case of seed treatment, the comparative residual toxicity of neem products never exceeds 15 days. This has been we have tested under the field conditions also. And then lastly, in some of the agroforestry crops or social forestry crops, what we have studied, for example, the poplar, we have introduced this poplar in Gujarat, particularly in the North Gujarat, and the eucalyptus in the South Gujarat. The neem products have been found effective, particularly in the on poplar under the North Gujarat condition, where the soil is sandy loam and the temperature remains very high. So we can't say against the Isopterans this neem products are less effective. This much I want to say. Thank you very much.
Thank you very much, sir, for sharing your view. Now I request Dr. Usha ma'am to introduce for another speaker, Dr. Virendra S. Rana, sir.
Thank you, Dr. Pavitra.
I think Mr. Chairman sir wanted to have some comment, I think.
One thing I would like to mention here. Hello?
Sir.
Audible? So one thing I would like to mention here that of course many of the things Dr. R.P. Singh has pointed out correctly, but as far as biopesticide and their formulations registered for use in India, so if you go in the CIBRC website, you will find even apart from the biopesticide, the NPB, BT, apart from that, this azadirachtin, particularly the certain formulations, that is 0.03, 0.2%, like that, it is even in the pyrethrum also that has been registered in CIBRC. It's okay for the homemade products for Neem. Really, it is difficult to maintain this one. As far as field application of Neem is concerned, so now you see in the there was one project, DFID project, World Vegetable Centers. So for the pest management lucinodes and all, that we are using this neem seed kernel extract at the rate of 5%. For the pest management in okra, that is being largely used by the farmers in the vicinity of our institute as well as some villages also, and they are getting good control of this lucinodes in particularly in that thing. So these are the points of what I want to make it. So the even in our vegetable, particularly in case of this one okra. And then this one cowpea and other things that we have made it. I mean, neem oil mixed with entomopathogens, particularly against the sucking pest, the Licanicilium lacani, that is Verticillium lacani. It was giving a synergistic impact, even if both are used at a half concentration. I mean, the instead of Licanicilium 6 gram per hectare, 5 to 6 gram per liter of water, you can reduce this to 2.5 gram, and then you can mix it with this neem oil. So it will have a enhancement in the potency of this combination. So in the field also it is being used, particularly in case of vegetables. And that is recommendations is also that is there for, and it is published in the research paper also. That is not the case for research paper publication in this. And the one thing you have rightly pointed out, even though despite the properties, promising properties of neem, it is not being that is being implemented on a farmer's field at a larger scale. So that I one thing tell you that industrial interest, they are not putting much interest. Because of that, they are finding it difficult to get it patented, that is a natural products. And apart from that, there are still lack of, or the shortage of lack of scientific evidence in support of the efficacy. Although the things are, there has been a World Neem Conference or the Neem newsletter that is also coming up. And if you go by the international conference and other things in the world over, that is also this negative point with this neem oil that has recently been come out for Canada and because of the potential misuse of that is of the side effects of the potential misuse of the neem oil. So these are the things are there, but CIBRC, particularly if you go the biopesticides which are registered in CIBRC, that is all there. But the formulations which has been registered, that is listed in there. And also Insecticide Act 1968, they are also included in that Insecticidal Act 1968. So this was all little bit. Thank you very much for a nice presentation that is covered by Satpathy and a lot of discussion. This all our colleagues have given important points that should be taken into consideration for future planning of or the research planning in the field of neem. And since it is grown throughout the year. Now, one thing I would like to mention here, the Satpati has mentioned 1.5 crores plantation of neem which is available. So in one of the calculations we have calculated on an average of 25 kg of neem seed per tree available on an average. So although it may be more also. So I could consider if we apply at the rate of 5% neem seed kernel extract. So actually in one time application per hectare, so the 5 tree yield will be, I mean, it is necessary for 5 tree yield on an average to have one application per hectare at the rate of 5% NSK. So this is too much. For that we have, there is a need to promote and for a large scale, this honeybee also that is coming in a picture, that this unilateral flow of neem, that will be a therapeutic value for that. So there is a need for the large scale or the concerted efforts needed to promote the more plantation in the neem, particularly throughout the country. Also, it is highly suitable. Our country is highly suitable for the cultivation. Thank you. Thank you very much.
Thank you very much, sir. Ushama, okay, please introduce.
Now I would like to introduce our next speaker, Dr. V.S. Rana, sir. Dr. V.S. Rana, sir, is an organic chemist and currently working as principal scientist in the Division of Agriculture Chemicals, IRI, Pusa, New Delhi. Before joining IRI, New Delhi in July 2013, he has worked in the Chemistry Division of Forest Research Institute, ICFRI, Dehradun, Institute of Bioresources and Sustainable Development, Imphal, Manipur, ICR Doctorate of Medicinal and Aromatic Plant Research, Anand, Gujarat. He has been working in the area of natural product chemistry since last 24 years, especially on isolation, identification, and structure elucidation of secondary metabolites from medicinal and aromatic plants using spectroscopy method for their utilization for healthcare and crop protection. During this period, Dr. Rana has isolated and identified many new, rare, and bioactive compounds from medicinal and aromatic plants, and some of them were found to have potent therapeutic and pest control properties. He has developed the separation process of number of bioactive compounds and has published more than 104 research papers in international and national journals and 35 book chapters. He has worked in about 11 research and development projects in-house and external funded as PI, including Indo-Spanish project and co-PI as a 5 project. His group has also identified natural fumigants for the control of nematodes in brinjal, tomato, and rice, the substitutes of synthetic nematicides. He is life member of Indian Science Congress Association, Indian Society of Mass Spectrometry, Essential Oil Association of India, National Environmental Science Academy, Medicinal and Aromatic Plant Association of India, Society of Pesticide Science India, Society of Conservation and Development of Medicinal Plants. He has worked with Medicinal and Aromatic Plants Association of India. I welcome you, sir, and invite you to share your views on neem for the plant health management.
Thank you, Dr. Usha, for your kind introduction. Good afternoon to everyone. Respected Chairman, sir, and panelists, sir. I thanks Dr. Pandey and Dr. Dobrial for choosing me for this particular session. And I also thank them for making neem as a main focus point of this webinar. Because as you know that neem is used in medicinal purpose, but it's one of the important ingredient for the biopesticide. If you see that many of the biopesticides known in the world, neem is grown in India in the highest amount. If you see the pyrethrum, it is very rarely grows in India. If you see the dairies, very few amount, but this is the only one of the plant grows in the large number of India. And I fully agree with the chairman, sir, comment, as well as Dr. R.P. Singh, sir, comment that we have a neem in tremendous amount, but we are not able to exploit it. We are not able to produce the product and give to the farmers. Many of the farmers are not aware about the neem products. And there are the many reasons they have also pointed out. And certainly this need to be addressed at the policy level, at the product level, at the market level. So I was asked to give one small presentation on this. So I chosen the topic, the neem as a source of a pest control agent under this theme. And I would like to just this actually, I'm not going to teach any agriculture, but just to remind that it's a very oldest practice we are following since 10,000 years ago. So since then, we are continuously. Hello.
Yes, yes.
Am I audible?
Yes, yes, you are audible.
Okay, okay. So since then, we continue to have a challenge of a food security as well as the ecosystem security. And here what we are trying to ensure the sufficient food for the increasing populations with available resources in changing pattern of biotic and abiotic factors, produce a sustainable, safer and nutritious food items, keep our food competitor, especially the other animals at the threshold level to protect the edible material and also biodiversity, to protect the nature from the environmental pollutant and climate change. And the major focus, what we are trying to see, that the identification of environmentally benign pest control agent. Because if you see, we have a very, very less pest control agent available with us. And if you see the biopesticide registered in the CIBRC, only the neem is only one of, and not only neem, only azadirachtin is only the one product registered in the CBRC. So we don't have any options except for the neem. So you see that since generations, we are continuously addressing these issues of plant insect pest-borne diseases, and they are the main competitor. And second, the changing climate and natural calamities is another problem for us. So here, in order to maintain the food security, produce the large amount of the edible materials, we are using the pesticides. In the two sides, everybody know that we are using the synthetic pesticides and natural pesticides. The natural pesticides, we have a very limited amount, but synthetic pesticides is highly dominated in the markets. And if you see that because of the synthetic pesticides, we are able to produce a large amount of the food grains and vegetables and the fruits. So, but they have their own advantage and disadvantage. They are synthetic pesticides are highly effective, multitasking. Normally they are hazardous, they are considered less biodegradable, if you see, and extensive and continuous use of these pesticides cause the environmental health hazards. And again, what we are reporting, they induce pest resistance also. But on the other side, if you see the natural pesticides, we consider that they are less effective normally, but they are very target specific, non-toxic and biodegradable, environmentally safe. And we normally, whatever report is published, they show that there is no pest resistance against the biopesticides because normally they are the mixture of multiple secondary metabolites. So because of those reasons, they are not able to use the resistance. So if you see, I'm just telling to go back to the history. From 3000 BC, we are using the pest control agent, and the sulfur was one of the oldest ingredient we use for the pest control. Then we started using oil and ash and other material to control the pests. Then then we again shifted to the tobacco infusion for controlling of different insects in 80s. Then sulfur and the copper compounds, we started using these for the crop protections. But in the modern year, after 1930, the chemists, they started identifying the alternative, because before that, normally we were focusing only on the nature-based product, whether they are inorganic and organic. But later on, after the discovery of a DDT, then tremendous change has been made in the discovery and development of the synthetic pesticides. And now you can see that many generation of insecticide has been developed, whether they are the pyrethroid, whether they are organophosphate, whether they are nicotinoids, and many more compounds. So we were continuously focusing on discovering these pesticides. But what is happening that they are causing a lot of problems to the end user as well as the environment. So now people are again focusing towards the product of a natural origin. So here there is a need of a search of effective, economically, environmentally safer alternative of a pest control agent. And that is the main source from the plant biodiversity. And these bioresources, which we say that plant biodiversity or microbial biodiversity or fungal, whatever it is, so they are the basic source of a pest control agent. So among those, if you see all the literatures, around 1,500 plant has been identified that they are the most effective one. But among those, only 24 plant has been considered as the crop of the cream of the crop. So out of these, neem is also one of them. And if you see in the Indian scenario, in India, we have a large amount of the neem, as I told earlier, and other speaker also told, that if you see how these plants are contributing in the development of a pest control agent, if you see the contribution of a naturopaths in a global pesticide discovery and the development, if you see the naturopaths are basically the unparalleled source of structural diversity. We can have a different structures, and those structures can be converted into the different type of products. They have a very unique mode of actions. They provide a very unique class of pesticide. And if you see the history, the discovery of carbamate was started from the identifications of a compound from the calabar bean, Physicostemma venenosum, then nicotinoids from the Nicotina tobacum, this is basically tobacco, and pyrethroid group from pyrethrum. So you can see out of the 5 generations of the pesticide, 3 was the concept of 3 generation was originated from the plant only. So you can see how these biodiversity, these plants are very important for the discovery of pesticides. So if you see among these, the pesticide, what we are using, the 70% pesticides are the natural or naturopaths derivative. Even you can see the microbes or the ivermectin and spinosades and so on and so forth. Other compounds are also there from the natural sources. So if you see, as we were discussing, that the manufacturing and consumptions of a biopesticide in India, if you see in 30 countries, 225 microbial pesticides are manufactured, and the total maximum amount of a pesticide is consumed in the Western US, Mexico, and Canada, around 45%. If you talk about the Asia, the market for Asia, 5% of the biopesticides sold in the USA, it's around 200 biopesticides are sold in the US. And if you talk of the European Union, it's 60 biopesticides are available in the European market. If you see in India, the biopesticide registered in India based on the 12 type of the products, and they are around 970, and they mostly on the microbial base, except the azadirachtin. So others are all, whether trichoderma, whether any other species of bacillus and other species, they all are from the microbial. So the public sector is normally producing a 70% of biopesticide in India. So here there's a point that we must encourage the private sector to produce the biopesticide in India. So even we say that in India we have a lot of biodiversity, but if you see the registration of a biopesticide in India, It's only the one biopesticide we were able to register, and that is only the azadirachtin, even not the neem oil. So this is the status, and this is because of some of the stringent policies, because the synthetic pesticides and the biopesticides are having the similar policy of registration. So as Dr. R. Singh has told, that there is a change in the policy level also, so that we can easily register the product, because whatever criteria they have given, it is the same for them, whether it is for the biopesticides or whether it is for the synthetic pesticides. Yeah, certainly there is a point that if you have a single molecule, say if it is only the azadirachtin, so certainly there will be the same. But when it is the mixture of a number of compounds, in that case we must have a different policy for the registration. Then if you see the market of the synthetic pesticides, 95%, biopesticide only the 5%, and India only use 3% of the biopesticides, and I told majority of them of the microbial base. They are the normally biocontrol agents. So, but if you see the overall projection of the biopesticide markets increasing at the rate of a 17% up to this 2016. So, and that's region of increasing of this market could be the increased R&D cost and registration cost of the synthetic pesticides, increased regulatory criteria on synthetics, shift of consumers towards the natural one due to toxic nature of the synthetic pesticides, and another go for the organic food. This is also another where we are trying to have the non-pesticide food type of food. And the second, the ease of application of biopesticides is very, very good. So it's very safer to the end user also. So these are the some of, there may be many other reasons, but these are the some of the major reasons that the market of the biopesticide is increasing. But the issue is we don't have sufficient. So based on these, we have divided 3 types of biopesticides, one with a extract base, one the biocontrol agent, and plant incorporated proteins, that's basically like a BT. So if we talk about the Azadirachta indica, Dr. Satyapati has already given a very detailed information of the use of Azadirachta in crop protections. But some of the point what I would like to mention, that the whole study, the whole focus is around the Azadirachtin only, because this is the one of the important crop protection agents which is obtained from the Azadirachta. The good thing is here in this particular plant is it's a perennial tree. Once you plant it, there is no issue. It will be continuously giving fruit till the plant exists. It grows almost all over the India in all type of the soil. These are the very good thing, and it's very, very popular for neem oil, neem based biopesticides, and the other advantage that we are using the neem coated urea for the field applications for increasing the productivity. So if you see the importance of it, because the leaf, if you go through the literature, the majority of the most of the work or the major work has been focused on the neem oil only, and leaf was not found as the best source because thorough study has not been done. There are maybe the other reason, because the molecule present in the Azadirachta is very, very complex, very, very complex, and identification of this molecule is really was a very, very tough. So I'll go little bit on it. So, and these basically the seeds, they are the rich source of oil, neem and the nimbin and other limonoid group of compounds are there, and this is the one of the major source of Azadirachtin. This is the only the natural source of Azadirachtin here, because when I discuss the points with the peoples, that they say, I many times say why the neem oil is active. The people say that it is because of Azadirachtin, but many times I say no, and you people will be surprised why I'm saying no, because the azadirachtin is a polar component. It is not the non-polar component. When we go for the solvent extractions, the very trace amount of azadirachtin may be present because it contains little bit amount of water. When you go for the expressed oil, again there may be a little bit amount of the azadirachtin, but it will be in the very less amount because it will be not at all soluble in that solvent, because it is a polar component. So it is a polar component. It is soluble in ethyl acetate, it is soluble in the acetone, it is soluble in methanol, it is soluble in alcohol. So the point here is, I want to clarify here that the oil contains other limonoids like nimbin, nimbinin and nimbidin and many other components. So, but little bit amount of azadirachtin may be there. If you see the formulations available in the market, they are basically containing a rich amount of azadirachtin. So this is the one point I wanted to say, that yes, oil is also having an efficacy against the insect, and it is because of the activity of the other than the azadirachtin, but certainly very less amount of azadirachtin may be there because of some of the moisture level or some of the extraction process is there. But if you see here, this seed consists of the seed kernels 45% and the cell is 55%. So this seed kernel, this oil yield is basically based on the seed. Other residue of a seed kernel after extraction of the oil can be used for the animal feed after little bit amendment in that the residue, because it may be having little bit bitterness. So that bitterness can be removed using the chemical process, and that can be used for the animal feed. So you get the oil here, and the cell also can be utilized for the other purpose. If you see, we have given a lot of information, but we are not able to exploit. If you see the initial concept that this particular neem leaf has strong inhibiting antioxidant activity against the locust, was given by the Dr. R. Chopra. I think he has published this information in the Punjab. He was working in the Agriculture Ministry in the Punjab, and they published this information in 2019, 128. Later on, from the IRI itself, because see, whatever we chemists do on identification of bioactive compounds, we normally 99% of the work we do based on the traditional knowledge, what traditional knowledge is saying. So here, the further another information, Dr. Pradhan and his team in 1962 and 63, they demonstrated the strong antioxidant property of a crude extract of neem seed kernel against the locust. So, but I have gone through the lot of literature, and I found that this is the one of the best product of a natural origin which can work against the locust as far as the antioxidant is concerned. Because people say in the literature, they are telling that even 0.05% concentration, if spray in the field, locust will never eat the plant. So like last year, we face a lot of, day before last year, we face a lot of problem of a locust in 2020. So if this neem kernel could have made available and can be sprayed, so we can protect the crop. And this is also an option with us because they have done this work at the field trial also. So there is the scope of utilizing of a seed kernel against the locust. Then other compounds, what at the time because I'm just telling the history how they have identified the active components. So based on these informations, people have identified the 3 antioxidant compounds against the other species. And the selenin, it was identified in 64, the milletrinol, it was identified 69, the azadirachtin was identified in 1968 and 71, because at the time what happens when I'll just show you how actually the discovery of this was done, azadirachtin, because it's really a very interesting story. You see that based on those informations of as an antioxidant, because azadirachtin was first time identified as an antioxidant, and based on this activity, the further work was done. if you see the Butterworth and the Morgan, they isolated the azadirachtin, and they were working in the University of Calais in UK. And at that time, it was not identified, and they have given this name based on the Azadirachta indica. So what was the problem during that identification of azadirachtin? One is the large number of functional groups. If you see here, the molecule, it's a large number of functional groups are available. So identifying those during that time, the convey, because no spectroscopic techniques was available, no structure was very, very complex, because many functional groups like ester, alcoholic, ether, epoxy, and many other type of group were available with the compound. There were 16 stereogenic centers. It's a highly chiral compounds. The sensitivity of azadirachtin to acid and bases was a problem in structural analysis. And another one, unstable in the photosensitivity of moiety. So these were the basic problem during that time. But people throughout the world, they were continuously working, and especially the Butterworth team. So, but during in 1972, they using, because at the time mass spectrometry was available in the market, and they little bit they started studying it, and they identify some of the key fragment. But even though at the time they were not able to identify it, if you see, the isolation was done in 1968, and continuous identification was carried out. And then JNU in 1975, the first structural assignment using an NMR was done. Even at that time also, they were not able to again identify the component. The name was going like azadirachtin. But we were not having a complete structure of the molecule. Then in Karas, at 1985, they proposed the structures based on the NMR data. And during the with the same period, like when in 1986, using the X-ray diffraction methods brought in, they have both of them, they have confirmed the structure, and they proposed the structure of azadirachtin. And this is the story from the identification. Took around 17 years to identify this component from its first isolations. So it was really a very, very complex molecule, and most structural. If you see that why the name the azadirachtin is active, the most critical structural element for the bioactivity epoxy group present at the 13 and carbon 14. So if you remove this particular group, the compound becomes inactive. And another story, because the azadirachtin is only sourced from the seed, but during that period, after the establishment of the structures, the chemists throughout the world, more than 100 chemists, they were working behind the synthesis of the azadirachtin. And many teams tried, but they didn't succeed. But later on, the total synthesis of the azadirachtin took 22 years. You can see, and this was the team led by the Stephen Lay at Cambridge University. And they published this synthesis 2007 with the title, The Synthesis of Azadirachtin, a long but successful journey, this journey. So, but if you talk about why the synthetic, now you will say that why now the synthetic azadirachtin is not available in the market. It cannot be produced because the cost of the synthesis of this product is very, very high. And that's why now we have only a natural source of the azadirachtin is the neem seed only, not at all other. But certainly they have attempted it. They took a lot of years to synthesize this component, but it is not commercially viable. So again, it is the nature is only the source of this particular compound. If you talk about the stability of this molecule, because the whole thing is going behind this azadirachtin only, so it's really very, very unstable when exposed to light, heat, and water. Within 100 hours, it goes down. The activity goes down and goes down, but there is no much data that how much time, what is the rate of a degradation of this one. So they further need to be focused. And another of point here is that what is the solution then? If azadirachtin is unstable, then how to make it stable, and how to make it stable, and how to get the compounds having a similar activity. So what solution has been reported is to convert the azadirachtin into dihydroazadirachtin. So there is a double bond, so that has to be hydrogenated, and this was published in 1984. And both of them is having a similar antifidant activity. So this, now we have the option that we can go for dihydroazadirachtin, but a lot of work further need to be carried out on this particular product, because the concentration of azadirachtin reported from the Indian plants, if you see the Indian literature, 0.2% to 0.6%, but if you see the world literature, people are saying that it is even up to the 6.5%. So why this much of variability of azadirachtin is there in the seed? That certainly need to, and Dr. R.P. Singh sir has also commented on this, because these type of study need to be done, because even when you harvest it and keep it in the store, there are a lot of biochemical changes are going on during the storage also. If you see in the market, if you go to the market and you purchase the seed, many of the shop, they are having a decaying seed, people are getting it. So certainly if you don't get the healthy mature seed, you will certainly found the lesser amount of the azadirachtin purchased in the seed. So the another issue here, but panelist has also pointed out that in my term, I can say the standardization of azadirachtin formulation is really very, very tough, because the availability of azadirachtin in the seed is a different. So here we can do one thing, we can have a batch. We can have a batch, and batch by batch can be analyzed. So normally, sometime what happens, the standardization can be done using the one of the bioactive components. But if suppose you start that, then we'll purchase the azadirachtin and then we'll standardize the formulations. It's not at all possible. If you see the sigma, 0.5 mg will be costing 13 or 14,000 rupees. So how you can standardize that particular extract? But yes, if you have some high azadirachtin yielding plant, from that plant, if you standardize that harvesting stage, the optimum amount of azadirachtin, then in that case you can standardize. But certainly it will vary again with the season, a lot of environmental and harvesting, so many parameters are there which basically affect the yield of the azadirachtin. Because we have found that we have carried out some of the study even on the same plants, within the month also the chemical components, if you separate the components within the month also, there's a lot of variability within the month with the same plant also. So it is totally biosynthetically or genetically, you can say it's a control. So really that is the one of the limitation in the naturopaths chemistry. The standardizations of the product is very difficult, and if the active ingredient is costly, then it becomes almost impossible to standardize that product. So that is the drawback here. But if you see the toxicity is very less. Oh, sorry, this is kg actually, 3.54 gram per kg in red. So toxicity is very less. Nobody is going to spray the plant, because this much gram of azadirachtin will not at all available with any formulations. And when you spray, nothing will be there. So toxicity level as far as the initial data is concerned, but now only some of the informations are now coming that it is, but it is not that much of a toxic for the especially to the mammals. So from this we are very safe. If you see, because I am not going to tell a lot of things, because already Dr. Satpathy already informed you that for how many purpose, against the whom it is used. So, but there are the something that neem seed has a very good potentials as a crop protectant agent, but there is a need to have a more work on the neem leaves, because it has not been much exploited. If you see the market product, neem leaf, except in the Ayurveda and except in as a mulch and manure, it is not used commercially. So this neem, because they sustainable also, like seed, we can utilize the neems for the production purpose, and we have to do it. So a lot of this type of work has already been. So other usefulness you see in it's having a very importance for the environment, purify the environment, it remove the harmful elements. So if you see the old belief that if neem grow inside the house and keep the surrounding air clean, so having a very good impact. It provide very good dense and excellent shades. In summer month, if you see the temperature under the neem is -10 degrees compared to the surrounding. It can be used for the restorations of the degraded soil and reclamation of lands, and many other uses are there for the even neem was used to reduce the spread of a Sahara desert in the country like Somalia, from Somalia to Mauritania. The medicinal important, everybody know the lot of medicinal importance are there. In Ayurveda, it is used since 5,000 years back for the medicinal purpose. As for the seed is seed oil can be used for both purpose, for insecticidal purpose, or even if it is produced in the large amount, it can be used for the production of a biodiesel, because you can remove the bioactive components from the oil, and other part can be used for the biodiesel purpose. So this is the basically. Then if you see the wood can be used, especially a source of cellulose, alpha cellulose, which we are basically using in the cotton. So this also can be used. Seed cake also can be used. Seed cake, if you see that seed coat can be used as a source of a carbohydrate and the crude proteins. So if we purify the seed cake, we can produce the animal feed. The seed cell can be used for the production of activated carbon and preparation of the hardwood. So a lot of uses are there. Here at the IRI in India was one of the organizations who has done a very good work on the, especially on the formulations aspects. As some of the panelists was telling, we had a process of as a direct rich concentration making methods, and that was transferred to the 3 firm. We have a neem emulsified concentrate with a minimum 300 ppm as a directant, and the formulation was made based on the BI specifications, and again the technology was transferred to the firm and they are commercializing it. Then again we made a polymeric seed coat based on the neem bioactive, and this technology was transferred to the NRDC for commercializations. Here previous speaker Dr. Satapathy was also telling about the nano product. Because if you see the normally neem oil is what in the laboratory we found that normal neem oil is not much effective against these 2 fungi. But when we make a nano formulation of this one, you can see the neem nano oil emulsion was very effective against Rhizoctonus solani and Sclerotium rolfsii. So this particular type of the product we are also testing in the field, and we publish this particular product also. This is the another possibility that neem oil can be converted into the nano formulations, and that can be directly used in the field crop. Another very important use, everybody knows that neem oil is used for the coating of the urea, because the nitrogen loss during the nitrification is very high. So this was the one of the very good use of the neem oil, which is used for the coating of a urea and to increase the productivity also. And when you spray it, people say that very less insect populations are seen in the field, and this is the farmer observations that the meanage of the nilgai has reduced significantly in the paddy crop where you are spraying the neem coated urea. The incidence of a leaf folder and stem borer in a paddy crop is reduced. This is the farmers' observations. Incidence of white ant reduced with the less neem coated urea crop. And if you see what they have, the NFL, basically they were the people who developed this neem coated urea. They were telling that the 0.5 liter of a neem oil is required for the 1 ton of a urea to just coat it. And they have added the cost of this one. And neem oil, if you see, it's cost around 75 rupees per liter. So that was added with the cost of the urea. So, but my point is, if we really focus with, on this one, so my one suggestion is that we must have a mission mode program of neem plantations to provide the sufficient raw material for the neem based product for the crop protection. This is actually, as Vice Chancellors are already told, they have taken a very big program. I personally feel this is the one of the crop which we can focus so that these seed or leaf are available to the farmer at the cheap rate. So we have a technology at the institute. We can give to the other people also, so that the large scale production of the neem-based biopesticides is there, or that oil can be used for the neem-coated urea. So this is all about my presentations, and thank you very much for each one of you. Thank you, sir.
Thank you, Roshan.
Thank you very much, sir, for your very informative lecture on neem. You have mentioned about the natural biopesticides, history, manufacturing, and the chemistry of azadirachtin, and the major constituents which is present in the leaf, neem seed, and on the neem seed kernel extract also. And you elaborated about the chemistry, particularly the chemistry of azadirachtin. You elaborated. It's very informative, sir. And thank you very much, sir. And moving forward, because of shortage of time, I request our chairman, sir, and panelists to give brief review about this lecture. I request Dr. Rai, sir, to give his comments.
Okay, thank you. Dr. Rana has covered nicely what to do for the promotion of this role of neem in the crop protections. Besides this historical background about the this identification, synthesis, total synthesis and all, he has highlighted nicely. It was really a good presentation, and we should stick. Finally, he has told also that we should start the plantation of neem tree. That has to be promoted on a larger scale, so that the availability of neem should be easily available to the farmers for their use in crop protections. So these are some of the points. Only thing is that another point I would like to mention here, that since because of this photosensitiveness and volatilization, that we should also work on how to extend the shelf life of azadirachtin or the neem products. That should be given importance so as to have a long persistence and for long-term control of the pest on the crops. Thank you. Thank you very much.
Thank you, sir.
Thank you very much, sir. Now I request our panelist, Dr. R.P. Singh, sir, to give his remark.
Dr. Rana has covered very well. I have made to brief you just one or two comments. What if you see, as we say that it is a wonder tree, every part it contains very valuable bioactive compounds, and then can be used for various purposes, right from cosmetics to the human medicine to the pest management. We have information that it has the insecticidal property, fungicidal property, bactericidal property, and nematicidal property, and many more. But what is important here, mostly if you see the total work, almost more than 90% work is only confined to its insecticidal properties only. Even out of insecticidal property, if you see that the commercial product that we have in the market, it is only one product, that is the azadirachtin based. Whereas we know that there are more than 100 biologically active compounds that is well characterized. So I have one question in my mind that Rana can clarify. Why it is only the azadirachtin that is being focused mainly so far? Why we have not concentrating on other compounds that is neem that you have already listed? So what is problem in their exploitation? This is the number one. Second one, if you see, the more work should be focused on its other properties like fungicidal properties, bactericidal properties, and nematicidal properties. As far as its current use, mostly it is for the insect management and for the nematode management. As far as its use in plant disease management is concerned, it is very, very minimal. So this product, almost all the neem products have certain specific areas where it has the advantage over pesticides, like synthetic pesticide, like organic farming. We have no other alternative. So for disease management, neem products can be exploited, but still lot of work has not been done. This is the number one. Second one, the crops like vegetables are any high value crops where the toxic residue of the seed are concerned, it can be exploited. Protected cultivation is another area. Okay. And this is the one area where the bactericidal diseases, as we know that we have only one antibiotic that is registered in India, that is the streptocycline. But if you see, the neem product has the very good bactericidal properties that can be exploited. And as I have limited knowledge, hardly you will get any reference where it has been tested against any bacterial diseases for plant disease management. Hardly you will get any reference. At least I have not seen so far any reference. All the references that is there, this is only for the fungal diseases, be it alternaria or the colectotrichum, or it is the fusarium, or it is Dr. Saab has already mentioned, that is the rhizoctonia and sclerotinia. Okay. Second one more property it is there, that it also has the antiviral properties. It is known to induce the host resistance. It is reported in the literature, but still we don't have exploited it, or we have not understood, or we have not tried it for its antiviral property or host defense inducing capabilities. So these are the certain gray areas that can be exploited and that can be worked on. So basically I wanted a comment from Dr. Rana, because I don't have any knowledge why our total exercise or total work is confined to generating only, not for other bioactive molecules that have the lot of potential. Thank you so much.
Sir, it's really a very valid question. If you say to me as a chemist, so if I see the literature and I found that a lot of components has already been identified, as a chemist, I will certainly not focus on those, because when I do the chemistry, I'm not getting to get anything, right, sir? As far as the product is concerned, for the farmer purpose, certainly it is having a very huge impact, and that specifically we go for the quantification of the azadirachtin. People say that the bioactivity of the seed is based on the azadirachtin, so the commerce is also focused on that only. So this is the one of the reasons that people have not worked much on selenin, nimbidine, and other limonoids present in it. But I personally feel they are equally important as we are focusing on the azadirachtin. So this part has to be taken, because as I told earlier, that the seed contains high amount of selenin. It is having very good antioxidant activity and very less amount of the azadirachtin in the oil. So certainly your point is valid, and these other limonoids need to be exploited, and we have given some work to our students also for the PhD purpose. So in this, during this period, sir, we'll certainly, because 2 point what you have suggested, bacterial, antiviral, these need to be addressed, because I agree with you, there is no reference on this, and we'll certainly see that how actually it works and can we exploit it for the antibacterial purpose. So this was the only reason that people has not worked on the other lemonoid except the azadirachtin because of a commercial interest. So even industries are also focusing that if azadirachtin is high, then we'll go, otherwise we'll not go. But certainly we'll have to change this perception also, that other lemonoids are also there and they are also equally important as azadirachtin, sir. So this is the basically, sir, my view on this. Really very thankful to you. You have given a very good suggestions, and those certainly will take in our R&D also in future purpose.
Thank you very much, sir. Now I request Dr. S.P. Saxena, sir, to give his remark.
It has been a very good talk by Dr. Rana, but only one thing I want to add is there must be some research to be carried out to increase the residual toxicity of any neem product. That is the azadirachtin product, because if we don't release, if you can't increase its shelf life, then no neem product can compete with any commercial formulation in the market.
Sir,
because
actually,
because in any form they are not comparable. If the shelf life is less, that means you need more applications. Firstly, second, you need more application means more total cost of the finished product. And if you are preparing at farm level, then the farmer has no time that to prepare the product many times during the crop cycle. So if you want to compete with the commercial formulation in the market, if you want to increase the organic farming, then the shelf life has to be increased. This is what I want to say.
Yeah, I fully agree with you, sir. Because the previous my senior colleagues, they have worked on the formulations, and some of the study they have already done, and these informations they have given to the farm to whom they have supplied the technology, transfer the technology. So this information is there, but it is not open to the public. So
okay.
Yeah, it is
if the work is going on, it's nice.
Yeah, it is already there, sir. And I didn't explain much also because already transferred to the industry. So
if the research is in pipeline, it cannot be disclosed until unless it is released.
Yeah, but it is there, sir. Yeah.
Okay. Thank you.
More work, certainly, I agree with your suggestions. More work is needed to be done so that we can increase the shelf life. It is not that within 6 months you should use it and within a year you can use it. It can be for 2 years or 3 years. That much work is also needed.
Actually, when you apply as a foliar application, then the problem persists more.
Yes,
if you apply in the form of a seed treatment, or basal application in the form of cake, that problem is not so aggravated. But if you apply in the form of soil application or a foliar application, the shelf life is very, very less.
Certainly, sir. So what we are also recommending is spray this product late evening, so that there is no photo reductions and like this. So there are some of the point guideline they have already given to the industry people also.
Okay.
So based on that, they are doing it. But I feel that more and more company need to be done, should participate in the production. Because if you see, sir, we are sounding a lot of neem tree are growing, seeds are falling on the earth and nobody is picking the field. There is no mechanism to collect those seeds. So if those mechanisms are also there, that we can pick up the seeds and collect and store it. So that much, even I personally feel that the seed coat, the unripened the seed coat, that pulp, that is all there must be having some of the very important ingredient, and that also can be utilized because we are showing it.
Due to these reasons, there are not too many formulators in this country.
Right, sir.
Although neem is our recipe, even then there are very few formulators, and out of the total formulators,
right.
out of the total formulators, mostly are the agro-based industries, only except 1 or 2. like the AID, Perry, or 1 or 2 big players. Most of them are the agro-based industries.
Right, sir.
No MNC is coming forward to manufacture or formulate the Neem products.
So the issue what Dr. R.P. Singh sir has raised, that is the registration. That is the big issue. We have to somehow, because if you see that Ayurvedic product, you can make and eat it, and you can put in the tea and you can sell it out. But this same plant you cannot sell as the biopesticide product. So we have to have some mechanism that biopesticide production and marketing is promoted in the country. And certainly government is focusing on natural farming, so you'll have no alternative now.
And lastly, one question I want to ask, not question, in the form of query. The third concept of farming has come, that is the natural farming.
Right.
Okay. Where do you see the neem and its products in the natural farming? Where do you see?
Sir, I see bright future, but raw material availability will be again the constraint, because we can use it, and critically I'm not very much aware about the what are the ingredients need to be added in the natural farming, because we say organic farming and again it's come to natural farming. So what are the components need to be added? So some guideline must be there. I'm not aware about that. So but yeah, but natural, if we add those non-toxic components to natural farming, which is normally we are eating also, so there should not be any problem. But I will have to see those guidelines, what are the ingredients to be added.
Okay.
Yeah, that's true, sir.
Thank you, Dr. Rana.
Thank you, sir. सर हम ही लोग बचे हैं शायद, और तो निकल गए लग रहा है।
चेयरमैन सर प्लीज कंक्लूड।
खाली 6 ही लोग हैं।
Sir, there is I think technical right now with the participants, particularly from the university. Anyway, for the concluding remarks for this session, so that we can proceed for the next session.
Yeah.
Okay. So this session we consider it as concluded.
I think sir, आप ही सर्वे आंसर आप conclude कर लीजिए।
नहीं ठीक। The only thing is that okay, in conclusion I could see that from the presentation of our 2 learned fellow, so that there are 2-3 things has to be taken care of. That is the promotion of neem on a larger scale for making the availability. That is one thing. Second is that we have to increase the shelf-life so as to have a toxicity for a longer period, so that it can be used effectively. And the third point is, apart from major directing, that we should have to have a consideration of other alkaloids which are having different kind of action against the different entities which are harmful to us. So these 3 things that has to be taken. And now I could see in one of the papers that nimbine and nimidine, so that the last one is having antiviral effect, that in some papers that is mentioned from abroad. This Neem oil and the pest management that is published in 2016. So he has mentioned this nimidine that is having a antiviral property. So these three things we are concluding that, and really I appreciate all the presentations I could find is a good presentation. I mean, covering most of the things, and it was really very interesting and thought-provoking lectures given by our two speakers. And to the panelists also, they have interacted a lot, and Dr. R.P. Singh has given a thoughtful suggestions for working on these applied aspects that has to be taken. Except for these two, what he has rightly pointed out, this Rana sir, this azadirachtin and pyrethrum. These two is having the this thing. Apart from that, many of the biocontrol or this molecular organism that has been registered. So it's really very good. And once again, I thank to organizer for this making a very good topic for consideration that will take care in the natural farming. And this Neem, finally, I want to say that what is the full form of Neem? Neem that has been implemented by Government of India. The Neem, that is the National Employability Enhancement Mission. So that can be utilized for that. If you are going to promote this, so a lot of employment will be generated for that. So rightly, this Government of India has launched a Neem, I mean the National Employability Enhancement Missions. With this, I thank to all the participants and then the panelists and the speakers for their presence and the interaction to come out with the good ideas for future planning and the research and development aspects of the role of Neem in the development of plant protection. Thank you. Thank you very much.
Thank you, sir. नई information के लिए बहुत बहुत धन्यवाद, sir. I was not knowing. Full form. Great, sir. Great to see you, sir. Really.
Thank you very much, sir. Now I request a participant to ask a few questions. Is there any question from participants?
Participant है ही नहीं है.
अब तो conclude हो गया.
Okay, no problem. We thank them, and if they have any question, already share the links of all the panelists as well as the resource persons, so they can directly contact also. We are running short of time, so we move ahead. है ना?
Okay. Okay. Thank you.
Okay, sir. Thank you.
Thank you, Dr. Rana.
Thank you, sir.
Thank you, the chairman, sir, and panelists and the speakers, sirs. Now I request Dr. Vinod Kumar, sir, to continue the technical session. Thank you.
Thank you, Dr. Vikram. I am not able to my desktop. There is some problem. From my mobile. So thank you, Dr. Good afternoon. Thank you for being here this afternoon. Kumar, Assistant Professor, RLBCAU. Welcome you to the second technical session of the National Webinar on Neem. The theme of this session is Neem for Soil Health Management. For this technical session, we are having a special guest, sir, but due to his prior engagements, we won't be able to chair this session. So I would like to request Dr. R.S. Yadav, sir, to act as chairman as well as panelist for this session. Please, sir, with due regards. So the panelist for this session are Dr. R.S. Yadav, sir. He is a principal scientist and head ICR in this.
Any problem, Doctor?
Gabriel sir, internet is properly working now. You can join through your desktop.
It's working. I think Vinod is lost somewhere.
Yeah, I am not able to join to my desktop right now.
No, it's working now. Anyway,
No, yeah, university internet is no working condition. So mobile पे join मत कीजिए.
Just wait.
Dr. Vinod sir, no need to join through mobile. Okay, you can access. Otherwise, voice will be breaking now. No need to join through mobile hotspot. Now Wi-Fi is working.
Wait. I'm joining. It's not working. That's why I said. So, the problem in internet. Just wait for a few minutes. It's not working. Yeah. Dr. Tanu, it is not working. वो बंद कर मैं मैसेज क्या है उसको बंद करो इसको बंद तो जरा आप देखो इसको करो मैं इसी से बोल रहा हूँ हेलो am I audible
Yes, sir. Yes, sir.
Yes, sir. You are audible, sir.
Yes, sir. Yes. The problem is with the internet, so I have joined from my mobile only. So we need to say so. For this session, Dr. A. Nasam sir supposed to be chairman, but because of his prior engagements and some another work, he won't be able to join this session. So I request Dr. R.S. Yadav sir to act as a chairman as well as panelist with his kind permission. So the panelists, Dr. I would like to introduce a brief introduction about Dr. R.S. Yadav sir. He is currently working as a principal scientist and head ICAR, Indian Institute of Soil and Water Conservation Research at Tatia. He obtained his MSc Agriculture Soil Science from Rajasthan College of Agriculture, Udaipur, and PhD from Shri Karan Narendra Dev College of Agriculture, Jobner, Bikaner. He served as AARO in the government of Rajasthan in the year of 1996, then as assistant professor at RAU Bikaner 1996 to 96, scientist at Modipuram from December 96 to June 1997, and as a scientist at ICR Kafri Jhansi and principal scientist at ICR IIFSR Modipuram from 2011 to 2016. His area of specialization is soil agroforestry research, watershed management, farming system, organic agriculture, assessment of carbon emission and mitigation. He has also been awarded by various organizations for his excellent work in the field, and he has received many awards and distinctions such as Vasantro Nayak Award 2010 for outstanding research applications in agriculture, Best Research Paper Award 2008, 10 and 12 by ICR Kafri, Jhansi, and also Best Teamwork Award for 2007 and 10 by ICR Kafri. He has also published a good number of research articles, which includes 31 research papers, 47 scientific articles, and also written 2 books. He has guided 3 master level students and 1 PhD student. Thank you, sir, for your presence. Our another eminent panelist for this session is Dr. R. Srinivasan, sir. He is currently working as Principal Scientist, Agriculture Microbiology in ICR, Indian Grassland and Forest Research Institute, Jhansi. He has obtained his BSc Agriculture from TNAU Coimbatore, MSc in Agriculture Microbiology from University of Agriculture Sciences, Harvard, and PhD in Microbiology from ICR, Indian Agriculture Research Institute, New Delhi. He served as Assistant Professor in College of Dairy and Food Science Technology, Maharana Pratap University of Agriculture and Technology, Udaipur from 2007 to 12. And joined ICR IGFRI Jhansi as Senior Scientist in 2012. His area of specialization is agricultural and soil microbiology and working on stress-tolerant bioformulations. The PGPR for abiotic stress tolerance in plants, microbes for soil health, and nanonutrient applications in agriculture. He has over 50 publications, research papers, chapters, articles to his credit. He is a life member of 9 professional societies, recipient of 6 awards. He has also guided 8 masters and 1 PhD. I welcome both of you to this
Hello, am I audible, sir?
Yes.
Yes, thank you.
Yes,
very good afternoon to respected chairman of the session, panelists, speakers, and all the dignitaries here to attend this webinar. I would like to introduce the speaker of the technical session on the theme 2, that is Neem
for soil health management of today's webinar, Dr. G.K. Patel. Dr. G.K. Patel is from Gujarat Narmada Valley Fertilizers and Chemicals, Vadodara. Sir is an agriculture graduate and has a MBA in marketing. Sir is working with GNFC since 1986. So he has a very vast experience of about 36 years. Sir has worked in various capacities in the states of Uttar Pradesh, Gujarat, and our national capital, Delhi. Presently, Sir is working as a Chief Marketing Manager in Gujarat. Sir is attached with GNFC Neem project, a responsible act for social transformation since 2015. Sir is a key member in devising concept and successful execution of the Neem project in Gujarat and Uttar Pradesh. Dr. G.K. Patel has toured extensively in Madhya Pradesh, Maharashtra, Karnataka, Gujarat for the Neem project. Sir has attended various seminars and international seminars at Dhaka, Bangladesh and Mumbai, organized by Neem Foundation. Brief introduction. I again invite Dr. G.K. Patel to deliver his talk for today's webinar. I request Dr. Shailendra to start video as requested by Dr. G.K. Patel, sent by Sir. Dr. Shailendra,
डॉक्टर काले सर इस समय इंटरनेट नहीं होने की वजह से ना लैपटॉप से शेयर नहीं हो पाएगा वीडियो 12 चलाती है
ओके
थोड़ी देर बाद
काले इफ कैन बी देयर ओनली डायरेक्टली वी आर शेयरिंग नो इश्यू वी आर शेयरिंग
नाइट्रोजन का मतलब नाइट्रोजन का मतलब आप रहू के वैसा नहीं यूरिया ने हलवा ना
I request other participants to please mute yourself while the speaker is speaking. Thank you.
सागरी का पाए माफी दी सागरी का ऊपर ना आती है ना तो देश से जाता पाए माने जो विचार पाए में होता नहीं ऊपर से अपन चल तो पाए मारे तो
वीडियो नहीं वीडियो तो ना अरे YouTube જોન બાપા ના આવડતું
तो बताओ
Yes, sir. Sir, video is visible, sir?
Yes, visible.
जो आकाश वाला म्यूट वे कर यार
अरे इसमें आवाज क्यों नहीं आ रही इसको नीचे ऑडियो ऑन पड़ता है एक बार
सर साउंड नहीं आ रहा है
साउंड
बानून बाजू पूछो
प्लीज शेयर करें। उसमें नीचे ऑडियो आएगा, जब शेयर का ऑप्शन आता है तो लेफ्ट लोअर कॉर्नर में ऑप्शन आता है, वहां पर क्लिक करेंगे सर, तब आएगा।
हेलो,
Voice is not coming.
It is diode of some.
It's side है ना, तो सेलिंग का है, आप शेयर कर रहे हैं उसको तो साइड से वो Dr. Kale, better go for slide without voice.
Am I audible?
Yes, sir. Yes, sir.
Yes, sir. You are audible, sir. You start your presentation, sir.
Yeah. Thank you. Thanks for inviting GNFC for the NEEM presentation. I would especially like to thank Dr. Ajay Pandey sir and Manmohan Dovarial sir. On behalf of hundreds of GNFC officers and staff who has worked passionately for the NEEM project, I am presenting this initiative of GNFC. Friends, in the morning session, inaugural session, Dr. Saxena sir has also told that research is there, theoretical things are there, but the commercialization of NEEM is required. And since way back, since 2015, GNFC has started it. And I would like to share some of the experience of the GNFC. Mr. PK Singh, please mute. PK Singh, please mute. Thank you. As earlier told by a few presenters, neem-coated urea has the benefits. It increases the efficiency due to slow release of nitrogen, and so nitrogen is available for longer duration. Due to neem coating, urea becomes less hygroscopic and it prevents lump formation also. And due to this coating, ICAR studies that reduction of 10% in consumption. So naturally, that's why Government of India has introduced 45 kg bag of urea. And azadirachtin of neem prevents crops from some disease and pests also. So these are the benefits of neem coated urea. Friends, we will go through the total consumption pattern of urea and requirement of urea nationally. Nationally, urea consumption is 335 lakh metric ton. Out of that, 244 lakh metric ton is indigenously produced, and 90 lakh, around 90 lakh is imported.
तो आपका कब है?
So, value of imported urea, considering recent hike of international urea prices, is 17,400 crore. In dollar term, we can say it's 2.3 billion US. Major manufacturers of urea are IFCO, CRIPCO, RCF, GNFC, GSFC, Chambal, Indogulf, NFCL, SFCL, SFC and NFL. Many more are there. Overall, 16, 17 plants are there. So, after doing neem coating of urea, these are the benefits, socio-economic benefits. Financially, due to less 10% less consumption of urea, saving of 15 per bag to farmers. And that's why government has reduced the bag capacity from 50 kg to 45 kg. Also, reduction in import of urea, and naturally subsidy reduction is there, and our precious foreign exchange outgo is saved due to neem coating of urea. Total foreign exchange saving is 17,400 crore due to this neem coating of urea. And also, due to neem coating, diversion of urea to the industrial use is somewhat controlled, and thereby subsidy loss is averted, and more urea is made available to farmers. As per rough estimates, in Gujarat, only 75,000 to 1 lakh technical grade urea requirement for the industry. And naturally, for industry use, technical grade urea cost very high, because in urea, for example, in recent scenario, we can say that urea price is, international price is more than $650 per metric ton. So it costs around above 50,000 per metric ton, and we are selling urea to farmers at 6,000 per metric ton. So naturally, in open market, urea price is very high without subsidy, so diversion issues are there. From May 2015, Government of India has made it mandatory to coat 100% urea by neem oil, and neem oil is used for coating of urea. I will go through some details also. With the efforts of GNFC, these things has happened. We are proud of it. GOI notified on March 2015 and make it mandatory to produce 75% of total production to neem coated urea. Then within 2 months, GOI notified 100% coating of neem urea, and notification came in May 2015. And on August 15, from Lal Killa, Honorable Prime Minister also declared that we had decided to do the 100% neem coating of urea. That was in the video, but somehow it was not audible. Then we observed that people are using not pure neem oil. So new guidelines were issued in October 2015 that with neem oil specification, and GOI notified these specifications in FCO on February 2017. Also, GOI advised to all urea manufacturers to procure neem oil from genuine neem oil manufacturers only. These all things were to increase use of pure neem oil. And last but not least, during 2018 March, Agriculture Department had notified. Recently, our last panel were discussing about the usage of nimbin and selenin also. So to get the benefit of these nimbin and selenin, this azadirachtin, nimbin, and selenin content was to be tested from urea coating also. And this is in pipeline. It is under consideration. Final gazette notification is yet to come. This is a simple process of spraying of neem oil on urea. And our last presenter has told that 500 ml per metric ton is requirement, but actual requirement is 800 ml per metric ton. For 1 ton of urea coating, 800 ml neem oil is required for the spraying. A simple nozzle spray on the belt where urea moves, it is being done, and complete urea coating is being done. As far as GNFC is concerned, GNFC was the first company to produce 100% neem coated urea since January 2015. Means before notification of GOI, we had started coating of neem urea. And nowadays, we can also say that GNFC is the only company which uses self-produced by backward integration method. We do the neem seed collection, we expel the neem oil, we extract the neem oil, and we use pure neem oil for our urea coating. And if all companies may go by this, it will be a great neem revolution in India, because total neem oil requirement for coating of 335 metric ton urea. So total neem oil required will be 26,000 metric ton. And for producing 26,000 metric ton neem oil, 4 lakh metric ton neem seeds are required. And it will generate income to more than 40 lakh people. And it can become 1,000 to 2,000 crore industry and will provide additional income to rural citizens. It will also create growth of expeller and extraction units also. And neem has a great export potential. Nowadays also neem oil and neem cake being exported to European countries also. And use of neem cake as organic fertilizer will enhance soil fertility, will maintain soil status also. So neem revolution can be very much helpful to our country also. Regarding our project, I would like to highlight some facts of Gujarat region. We had worked in Amethi, Sultanpur and Raebareli also. So some experience of UP is also there that we can also discuss. But in morning presentation, it was shown that at India level, there are 5.5 crore neem trees. That is wrong. Actually, our government of Gujarat's forest department has done the neem tree census. They have done the tree census of all trees. And from that census, I have the official data also. Only Gujarat is having 5.03 crore neem tree population. And other than Gujarat, UP is having larger than neem tree population. Those data were shown in the morning slide, where of way back of 1981 of NEEM Foundation booklet, and this data has been revised. But I would like to tell that like Gujarat, if other states will do the neem tree census, then we can came out with neem tree population at all India level. But I can say that other than Gujarat, major states having more and more neem trees other than Gujarat are Karnataka, Maharashtra, Madhya Pradesh, Andhra Pradesh, Telangana, Uttar Pradesh, and of course Rajasthan. These states are having a good population of neem trees. So neem tree population is not the problem. We can fulfill our required resources by tapping the 50% potential of neem trees can also. And only Gujarat is having 1.12 lakh metric ton per year neem seed potential is there. Generally, neem tree start yielding of seed after 5 years, and day by day year by year, its yield per plant increases, and tree having good trunk size, and after 10-15 years can give up to 50 kg per year also. This has been studied well, and this has been experienced well. On the basis of our GNFC team's experience, we are sharing this data. Simply district-wise, because when we started our project, we studied well, and we studied about the district-wise neem tree population. So major district is shown in the slide, but person from the other than Gujarat might not be having that district knowledge. But some districts are having around 40 lakh neem trees per district. In case of UP also, I can share that when we visited Amethi, Raebareli, Sultanpur area, local office were telling that hardly 50,000 trees might be there. But when we surveyed in detail up to 800 villages, and we found out that more than 5 lakh trees were there in that area also. Some of the team members had joined also today also. So, and from that resources, within 1 year, we collected about 700 metric ton neem seeds from Amethi, Sultanpur, Raebareli area also. So, there are many. There is a huge population of neem trees in UP also. Major objectives derived for our neem project were for reduction of urea usage and increase in efficiency by neem coating of urea, supplementing income to rural poor and women empowerment, because generally neem seed collection is being done by women only. Saving in foreign exchange. Naturally, when person will earn from the neem tree, they will plant neem tree only in their courtyard, in their backyard, and social forestry can be also taken up. And promotion and usage of neem cake and neem pesticide in organic farming, and preventing misuse of urea and to prevent the diversion of urea. These were the objectives of our neem project. Our primary model was like that. Seed collection from farmers and farm labor, women self-help groups, run by Livelihood Promotion Company, Gujarat Livelihood Promotion Company, GLPC. Because we are in the fertilizer business, so we are well related to cooperatives and fertilizer retailers. So they are also our channel partners. Gujarat Agro Center in charge, and Gujarat is having White Revolution. So there are many milk cooperatives, and they are also connected to at village level. So by using this channel, we had developed village level collection center. In shorter term, we are telling them VLCC. And from village level collection center, with the help of our strong network of our own network of 51 Narmada Khedut Sahay Kendras, we are telling them NKSKs. We are collecting the neem seed, and we are having our own expeller and extraction unit at Bharuch, and we are storing intermediary at open warehouses and sending material to processing unit at Bharuch. So this is the model developed by GNFC. Again, after collecting this, in-housing crushing and at expeller and extraction is being done, and from that we are doing the urea coating through neem oil. We had developed Narmada neem pesticide of 300 ppm and 1500 ppm. I would like to show the packing. It is with me, and our neem pesticide in shorter duration is very popular. Annually, we are selling more than 50,000 liter neem pesticide in Gujarat only. And we are hopeful that we will go nationally and we will increase our turnover also. Also from neem oil, we had developed neem FMCG products also. We are having neem soap also. We had developed neem soap. Generally in the market, neem soaps are having standard brands. Neem soaps are having only 1 to 1.5% neem oil only. We had developed neem soap having 21% neem oil. I think it is only soap in the world having such high content of neem oil, and it is successfully produced, and we are selling sizable quantity of neem soaps in different versions. Neem soap with aloe vera, neem soap with tulsi, and these all natural extracts are being collected from our Kamboj center, and all natural ingredients we are using. We have got the EcoCert certification also as an organic product also. We have developed neem hand wash, neem oil is there, neem shampoo is there. These all products are having 5% neem oil in their content. And also we are producing neem cake for farmers as well as for garden manure. For garden manure, we are also having pure neem cake in 1 and 2 kg packing. And for farmers, we are having 25 kg packing and 50 kg packing. And in the morning, one respected panelist has said that neem cake quality is an issue. But in case of GNFC, we had accepted the challenge, we had taken up this challenge, and we are producing neem cake with less than 10% ash content. Our 90% material is pure neem cake. Sometime due to faulty storage, due to its self-combustive nature, it gets fire also. But we are not bargaining in quality, and we are producing pure neem cake also. This is the neem seed cycle. Those who are in learning phase, because neem is a typical fruit. It is of self-combustive nature. And initially, when neem fruit falls from the tree, it's a more than 60% moisture level. So its collection starts in June and July till rainfall comes. Because when heavy rainfall came, all the fruits gets wiped out in the flood. So we start purchase from our VLCC level with 20 to 40% moisture level. We supply to our service provider partner and our company go down. And from there, we move our material to our company go down. And when neem fruit moisture level goes to 10 to 12%, we start expelling and extraction. But one or two things I would like to mention, that due to its self-combustive nature, it is to be stored in jute bag only, because enough airspace is required for that, and it can be stored in open shade, with shade but in open place, so some wind and some air can go there. In closed godown, we can't store neem fruits. So these are some limitations for storage and this packing also, but we are coping it out. These are some photographs of our field promotional activity. Because in Gujarat, this concept was very new, and UP also this concept was very new. So from March onward, we start our activity before seed collection activities get started. We do the meetings with government stakeholders and livelihood promotional company stakeholders. Our strong network of more than 100 officers go to the field. They explain about neem seed collection, how to collect, how to clean it, how to control the foreign matter, what will be the moisture level before supplying, and this complete promotional and collection activities being supervised by our fertilizer officers. These are the data of neem seed collection. You can see that during 2017-18, we had collected 23,000 metric ton neem seed. And that was the 50% of Gujarat's potential. That year, Gujarat State's total seed collection was above 50,000. So this is the potential of Gujarat State, and same way other states like Maharashtra, Karnataka, MP, UP, all are having same potential. But only one thing is required, how to explore it. GNFC has explored. IFCO is doing some small work, but other companies required to come forward. From very first year, we had collected more than 10,000 metric ton neem seed. and what the rates were given, what the purchase value in crores. So overall, more than 70 crore rupees have gone to the rural economy due to our project. These are the data from other states also. We had collected some quantities also, and data from UP is also there. This is data of 567 metric ton during 2018. This is our official collection. One local fertilizer plant was our partner to process the material, but due to limited capacity, we have to collect the material for other purpose also. Otherwise, we can have collected more than 1,000 metric ton from Amethi, Sultanpur and Raebareli area. So these are the activities being done at different states also. Some of the challenges and some of the parameters I would like to repeat again. Neem seed moisture content around 30%, but while crushing it requires only 10 to 15%. Sand and foreign matters should be less than 10%. Otherwise, it affects the machinery and it affects the quality of neem oil and neem cake also. Challenges while purchase of neem seed, because this is the direct collection from fields, gochar land, roadside, backyard, etc. And people are doing by two methods. In MP, etc., people are doing neem seed collection by hand picking. But in major region, it is being done by brooming only. So foreign matter and sand content varies at different areas. Also, moisture level is also varies in different areas. In middle Gujarat, people came with neem seed having moisture level up to 45%. And in Bhabar and Banaskantha area and in North Gujarat area, people came with moisture level of 20%. In MP also, neem seed with 15 to 20% moisture level is being collected. We had tried the use of, we had tried to convince the collectors to use the sieve so foreign matters can be avoided, but we had done it with little success. I would like to say that because people are adamant, because they are earning a small amount, so they are not techno savvy also, and they are not they do not want to increase their labor also. We had tried to use moisture meters to measure the moisture in the markets also, but it is also having little success due to self-combustion nature of neem seed. Storage at open space with cover shade and jute bag is required, that I have already told you. Journey of our neem project. Our neem project was inaugurated by Honorable CM on 2015 at Ahmedabad. Then we had launched our GNFC Neem website in 2015 only. Very quick step taken by our managing director that time. And due to his passion and his team passion, we are able to do it. Very first year, our neem seed collection was 9,151 metric ton from Gujarat only, and from other state, we totally collected 10,000 metric ton. Honorable PM praised the GNFC Neem project on 2nd September 2016 in a private channel interview, and that added more passion and more motivation to us. And then we decided to diversify our neem project also with forward integration. Marketing of neem products, we started from 2016. We established our own neem complex, and Honorable Prime Minister had done the inauguration on 2017. Then our neem project was viewed seriously by many agencies, and our assessment was done by UNDP, and they published socio-economic impact report of the project. We had participated in many award presentations and PM award presentations also, and our project has got many national and international awards like Porter Prize, Golden Peacock Award, India CSR Award, Scotch Award. And also DOF has mentioned our project and asked all companies to follow GNFC model, Department of Fertilizer. These are some glimpses of our awards and our inaugural ceremony by Honorable Prime Minister. Due to our Neem project, before 2015, Neem seed market was very scattered and unorganized, and it was exploiting rural people. Limited collection centers, only 50 to 20,000 metric ton Neem seed collection was there, and people were getting only 2 to 3 rupees per kg. So they were less. They were collecting very less also. But due to our Neem project and our promotional activity, Neem seed market started to become organized due to assured purchases and supply chain at the village level. I would like to tell that more than 2,000 village level collection centers were established in Gujarat during that time. Empowerment of rural people and women by better pricing and enrollment of self-help groups and cooperatives, our approximate centers at 2,200. And collection crossed 40,000 metric ton at Gujarat level, double collection within 2 years, and rates people were getting were 8 to 12 rupees per kg. Last year, people had got the rate of 12 to 15 rupees per kg also. So now Neem seed collection become an industry also. Assessment of our Neem project was done by UNDP, and this project was assessed in 4 districts, Gandhinagar, Bharuch, Patan and Banaskantha. And following were the key findings. 36% increase in average income of rural women was reported. 18% increase in number of women who owns livestock. Reduction in migration, I will elaborate. Enhanced decision-making role by women. 13% women reported reduction in domestic violence. Asset creation by women, and transformed some of the seed collection transformed to rural entrepreneurs. I would like to tell that this seed collection activity is being done during May, June and July. That time, no agriculture activities are there. So in rural, people are unemployed. And due to this, our project, people are getting daily income of rupees 100 to 500 till 30 to 45 days. And this has added in their confidence, in their livelihood also. Because due to unemployment, sometimes people have to do the migration. This migration has been stopped. Women is empowered, so domestic violence has been stopped. And from their income, they started some small business like selling of vegetable, hen, marga palan, like getting some loan and purchase one cow or a buffalo for a livestock also. And 1 or 2 ladies become entrepreneur also. I would like to mention one lady, she is from Bharuch, and she collected 80 metric ton neem seed through her village level women. And she started own papad making unit, and she started supplied papads to GNFC canteen, to a business also. So this type of such type of entrepreneurs also developed due to our Neem project. Again, Neem oil requirement, oil is used in Ayurvedic medicine. It is being exported. Organic fertilizer also used. And Gujarat level, 3,000 metric ton Neem oil is required. It has been already told, so I am not going in detail. It is also discussed, but I would like to describe some Neem seed and Neem fruit related information. Generally, GNFC collects Neem fruit, and Neem fruit is having 12 to 16% Neem oil. If extraction is being done, 1.5 to 2% Neem oil goes to de-oil cake, and we realize 10 to 10.5% Neem oil after extraction. If only expelling is being done, we got only 2 to 4% Neem oil, and whole 8 to 10% Neem oil goes to oil cake, and which is a very great source of Neem oil in case of soil application. If upper pulp is removed and Neem seed is crushed, it gives approximate 25% Neem oil, and from neem seed, upper shell is removed, and we got neem kernel. It gives 45 to 50% neem oil. This is for some basic or primary understanding. Active ingredients, as told in the morning, neem oil is having more than 100 active neem salts. We can describe it as azadictine, melantriol, selenin, nimbin, nimidin, etc. And it can control more than 200 species of insects, pests, bacterial, virus, fungus, etc. Some of that are there. Some SHG and NGO storage. As I had already told one lady, Amishaben Patel, she had collected with the help of 100 members, 86 metric ton neem seed. Reliance Foundation also has collected 55 metric tons. So at the institutional level, we can do it and we can get the success also. Somebody's mic is unmute. Please mute. Please mute. Again, Government of Gujarat's agencies are doing hand holding, and one convergence model is being developed. Recently, we had done the MOU with Gujarat Livelihood Promotion Company, and their pilot project is being started in Patan district. In Gochal land and in government land, GLPC with the help of Panchayat and Forest Department will develop Limda 1, Neem 1. And they will add this activity in Manrega, and they will provide wages to persons for tree plantation, tree maintenance, and seed collection also. So this will add on also, and they on this in this Neem 1, they will plant medicinal plant like Giloi also. So it will give also additional income to them. This is pilot project, and GNFC has GNFC is also providing them a know-how and required information for this project, and we are supporting them. And recently we had done the MOU with GLPC, our Honorable MD, and Principal Secretary Rural Development was present there, and MOU was signed between GNFC and GLPC also. And we hope that this will go a long way, although it is in initial stage. But the persons from Bundelkhand are joined in a different capacities. They can also start this type of pilot Limda 1 projects. So social forestry, in addition to social forestry, with the help of Manrega, people will get additional wages by plantation, neem plantation also. We are selling neem cake also, and these are the benefits of a neem cake. And in a short duration, our neem cake is the hot cake. It becomes very popular among Gujarat farmers. Whichever the production we are getting, it is instantly sold out. There is no inventory at our state level also. It's a pure organic manure. It's a pest repellent also in all type of crops and soil. It prolongs the availability of nitrogen to short duration and long duration crops also. Although urea is neem coated, 100% urea is neem coated, but if you want to use in paddy like crops, crops like paddy, people can mix urea with neem cake before 24 hours and spray the urea with neem cake. It will give excellent results also. It protects plant roots from nematodes, soil grubs, and white ants. Recently, some doctors, some research scientists has also supported it. It had very good result in white grubs and white ants. Because nematode control is very tricky thing. But neem cake is the natural resource that it can be controlled well. It is organic plant food. It increases nitrogen use efficiency. It increases soil fertility, and directly it improves the soil health also. We had also neem pesticide, and neem pesticide is having EcoCert certificate, pure organic. Whichever the stickers and solvents we are using, they are also bioproducts, and our products is 100% environment friendly. It is having no residual toxic effects, it also says friendly insects like dahlia and our earthworms, etc. It is compatible with the chemical pesticide also. It is effective on more than 200 species. It is already told. It can be used in integrated pest management and organic farming also. How it affects in the morning session, it was told, but we had simple 2, 3, 4 things we are telling. It is anti-repellent. So due to smell, pests goes away. It is anti-fidant, because when we spray the neem oil on the canopy, whole canopy becomes sour taste. So pest will not able to eat anything. So it is anti-fidant. It is reported that this bad smell also inhibits the signal of male and female wild reproduction. So due to inhibition of mating, further generation is also stopped. It is ovipositional deterrent also, and it due to continuous use of it and exposure to the pest, its size becomes very tiny. It's what you can spinal growth is very gets limited, and day by day the pest becomes very tiny, and it's not become healthy. So its next generation ability also decreases also, and it deters the this egg larva and this formation. The 4 cycles is there. It breaks that cycle also. Our Neem FMCG product, I had already shown to you, but it is made from the virgin neem seed oil, not from extracted oil. It is from the virgin neem seed oil. It is blend medicinally beneficial vegetable oils are also blended in this, and 80%, 85% ingredients are from natural sources. Neem hand wash is there, and I would like to mention that recently we had supplied material to government hospitals in the form of hand wash, and recently we had supplied 35,000 kits of neem hand wash and neem soap to all government primary schools.
Hello.
These are the other products: neem face wash and neem shampoo that I had already discussed. Neem shampoo is the very good, very useful for dandruff control. And last but not least, the neem tree is truly an agro-scientific celebrity, and it has been declared the tree of 21st century by United Nations Environment Program. Thank you. Thank you for listening me. Thank you for patience. If any questions, I will answer. Again, thank you, Dr. A.K. Pandey, sir, for inviting.
Thank you, Dr. Patel Saab. You have given a very excellent talk on neem and especially the role of GNFC. You have played a very pivotal role in harnessing the potentials and enhancing the livelihood security of the rural people near to your plants and other states. And you have made a wonderful number of the products that is really very important and very, very practical approach to for the plant and for the environment and for the people. Thanks, sir.
Thank you, sir. Thank you, sir, for a wonderful and informative presentation on Neem project, which is an initiative of GNFC. And you highlighted the benefits of the neem-coated urea, socioeconomic aspects of neem urea, and also talked about the neem revolution in India and how this project is preventing the misuse of area, reduction in urea usage, and increase in efficiency, and also the promotion and usage of neem pesticides in organic farming, and also supplementing the income of the rural poor and especially the women empowerment. And how you also said how this project has channelized and organized the neem seed market and assured the purchase and the supply chain empowerment of the rural people by providing them job opportunity and entrepreneurship development through this project is also possible. And you have also given information on the different products from the neem like neem-based biopesticides, other products like soap, hand washes, face washes, etc., which indeed increase the consumption of neem in the market, and rightly quoted that this tree said that the tree for 21st century is neem. So thank you very much, sir.
That's how I would like to request that the internet connectivity is good. Can you please rerun the video from your side?
Yes, sir. I will.
If possible.
Yes,
People will
if possible,
enjoy.
sir. Yeah, I was off to please, sir. Dr. Shailendra, if you can please play the video sir has shared with us. Dr. Shailendra, are you there? Sir, I will surely use this video after the next presenter, sir.
No
there
issue,
was some
no
technical
issue.
issue.
I am waiting for
Surely I will play that video, sir. Some technical issue is there. Thank you, sir. Now, I would like to request my colleague, Dr. Garima Gupta, to please introduce and invite our next speaker for this session, our respected Dean, sir, Dr. A.K. Pandey, sir, Dean, College of Horticulture and Forestry. Dr. Garima, please.
Sir.
Good afternoon to all and thank you, Dr. Vinod, sir. It's a great pleasure for me to introduce Dr. A.K. Pandey, sir. A.K. Pandey, sir, he is currently Dean in College of Horticulture and Forestry in RLBCAU, Jhansi. Before joining here, Dr. Pandey, sir, served as Dean College of Horticulture and Forestry, Pasighat, Arunachal Pradesh. Before entering in academic services, Dr. Pandey, sir, worked as principal scientist at the ICAR, Indian Institute of Vegetable Research, Varanasi. He is ARS scientist of 1985 batch. Dr. Pandey, sir, has published more than 80 research papers and almost 167 popular articles and review articles, and has authored almost 12 books. Dr. A.K. Pandey, sir, has been conferred several awards for his distinguished academic contribution. Honorable President of India has conferred on him the prestigious Rajiv Gandhi Gyan Vigyan Puruskar for his book Dalhani Sabjiyan during 2012. DRDO also honored him with the first prize Rajbhasha Puruskar for his book Bharat Mein Sabji Utpadan Evam Vikas Ki Takniki. His other two books, Patti Wali Sabjiyan and Nayi Sabjiyan, have also been awarded by the DRDO. Dr. Pandey sir's contribution toward utilizing the underutilized vegetable crops in addressing the nutritional security is considered one of the most notable achievements in enriching the food's diversity and inviting the attention of researchers to lesser known vegetables of the country. He is fellow of Indian Society of Vegetable Science, Varanasi, and Society of Biological Sciences and Rural Development, Allahabad. So with the introduction, I would like to invite respected Dr. A.K. Pandey, sir, for his talk. So kindly, sir, give your presentation.
So, very good afternoon to all my esteemed delegates, respected chairman of the session, panelists, and reporters. We are listening right from morning about the potential of the neem and its wonderful applications in the different aspects. Most of this your presentations part has been eased out by Dr. Patel Sahab. He has given a very beautiful talk on this neem coated urea. I have much appreciation, and the way this your GNFC has knitted the programs very efficiently, that is that deserve to worth of appreciations. Dear delegates, we are every day we talk about to these your chemical hazards, and how these your chemical hazards are causing a problem to our health, and major source of these your hazards are coming from to these your food stuffs. So one very talk about to this your organic natural farming. In last budgets, we have listened about how these your natural farming can be taken and how these your efforts are being made and how this your planning has been set on the bank of different rivers. So how this situations has happened and what is the scenario, what we did wrong, all these things I would like to within 20 minutes I will summarize. लाइट नहीं मूव हो रही भाई.
और सर लेफ्ट राइट करिए सर, लेफ्ट राइट.
So we are all aware about that how this your production has increased. No doubt after independence we have got a quantum jump in the productions. Though in 2021 I am here have mentioned about to this your 303 million metric tons. Actual figure goes more than 308 million metric tons.
Slide is not moving, sir.
Raghavendra, this slide is, sir, visible. Slide is visible, sir.
स्लाइड इज विजुअल.
This one is
विजुअल.
visible.
इसको स्लाइड मोड में करिए सर, स्लाइड मोड
स्लाइड
में.
मोड में डाल दिए सर इसको.
स्लाइड मोड में डालने की करें.
नीचे सर वो है ना.
फिर से उसको फिर से कर लो.
राघवेंद्र रीशेयर कर दीजिए, रीशेयर कर दीजिए.
यस यस. नमस्कार नमस्कार. यस यस प्लीज.
रीशेयर कर.
ओके डन सर, हो गया.
Yeah, it's fine.
हो गया
फाइन.
So, sir, just I said that as this population is increasing, and populations I am saying this are 139 crores of these populations. So, upper this your figure itself indicates that our challenges are what it was. Again, it is this your burgeoning population is taking place, and cultivable land are day by day is sinking. There is a very fast growth of urbanizations, so that our challenges still remain. And looking that on that account, how we have got to this your surplus of the food security, and in all food grains to wheat, rice, multiple times, more than 15 times, and in poultry productions more than 52 times, like that. That is all very golden achievements we have made. But look back, just around to this your 60 when this your green revolutions came to this your country and globally, and our visionary scientist Dr. Swaminathan, right from the 4th January to 1968, almost 44 years ago, how he looked to this your green revolutions, this your impact, and he clearly mentioned that it is okay, that has given a very good jump. He appreciated a lot of to this your work of to this your Dr. Borlaug, how this your gene revolution has given to this your green revolutions. But same times he pointed out that how will be these crops are very fertilizer responsive, very water responsive, and on that account we are, if loading to this your pesticide fertilizer and depleting to this your our water resources, water bodies, aquafair, then what will be the situation for the futures. So today we all should appreciate to Dr. Swaminathan's that voice, and after losing much, almost 3 and 4 decades, we are every day we are going to talk about these our health issues, our different aspects of this year, how to cope up the situations. Though we have got this your food security, but still these your situations, this picture indicates that in Asian countries, almost 381 million population is having the problem of this your food security, and other aspects are also not good. 54% of our children are stunted, and almost 69% of our children are vested, and 45% are having into this real of obesity. Earlier we were considering to this your obesity is the cause of to this your overnutrition, but recent researches and number of these your findings indicates that obesity is one of sort of disease, and major cause of this your obesity is a cause of to this your malnutrition, and very well to this your deficiency of to this your micronutrient malnutrition. So we can say that still we have got to this your food security, but nutritional security is still a dream. Here I have mentioned about all these your health related or malnutrition related to these your indicators. In India, 37.9% children are malnourished, and almost 50% of our ladies are anemic. Half of these your country's women population is anemic. There is a problem of this your iron deficiency. And obesity, so this global scenario looks that developed countries, almost more than 50% populations of these your even developed countries are in the real of or in the this your class of this your obesity. As I mentioned that obesity doesn't come alone. Obese persons are having the problem of hypertension, high blood pressures, diabetes, even during to this your COVID period, obese persons were very prone to this your COVID problems also. And further, this slides clearly indicate that micronutrient malnutrition is the very much problems, where countries only 50% population, whatever to this your recommended dietary allowances has been prescribed or to this your micronutrients, that is compensated. It means 50% populations is due to this your micronutrients, this your availability, and that is the hidden loss. I was interested to this your collect the update informations about to this your micronutrient loss, but what I published this your long back, only that information was available on this your internet, and that time I mentioned about more than 27,000 crores of loss on the account of micronutrient malnutrition. That is the hidden loss. So that is one issue. During COVID period, we realized the importance of zinc. I could not collect to this your latest data about to this your zinc related micronutrient malnutrition, but 30%, more than 30%, 2005 data indicate that the 30% population is having the deficiency of to this your zinc. Zinc, as all we have realized, zinc is considered as it's a this your immunity role, different physiological role, different it's a this your participation in different type of this your enzyme, how that is a zinc is considered as a gatekeeper of to this your immune systems. So that is one problems. Food security, as I said, that is still that is a change. Further, there is a problem of to this your malnutrition related micronutrient malnutrition. And in that aspects, we are facing the problem of to this your excessive or how to this your feed to this your plant, and feeding the plant fertilizer have become a only source. NPK, that is on top of to this your list, and how this figure that is going 2019-20, 191,000 metric tons we have nitrogen, phosphatic fertilizer 76, and potassic fertilizer. If we get any data right from to this your 1950 to 55, that time average application of NPK per hectare, I could see only 500 grams. And presently we have got to this your 133 kg of to this your NPK. So it will not be absurd to say that present days of farming is a total chemical farming. Whatever we are raising the crop only through the support of to this your chemical fertilizers. Second part go how there is the balance between to this your NPK. A natural balance by these your fertilizer experts, soil scientists, that has given is to this your 4:2:1. And national average, how much that is going? That is going to this your 7, 3, and 1. And how worst in the very prolific states where we say that we are having to these your grain bowls in Punjab and Haryana, that is the 31:4, 4:1. And in Haryana, 27:6. So how that is there imbalances into this your NPK fertilizers. Further, how we are nutrient deficient other way, how there is a gap between to this your addition and to this your its exploitations and gap. In different region wise, here I have shown to this your addition and removal, and how we are having to this your difference all over India. That is addition is the 241 lakh tons up to this your fertilizers, and removal by this your all crops, that is the 281 lakh tons, and deficit that goes to this your 66.89 lakh tons. So this is the chemical story, and further how there is the challenge. This is part of this your all over learned participants. How that is the deficiency, as we are taking it multiple times up to this year our productions. So almost 63% of our soil are very low into this your nitrogen content, and 42% in this your phosphorus content, and potassium 13, and sulfur into this your 40%. That is the deficient. If 40% deficient, certainly that will give a diminishing effect out to this your harmful effect to this your our productions out to this your pulses and to this your acid crops. Further, you look to this your how this your micronutrient deficiency into this your our soils. 49% of our soil is the deficient into this your zinc, and almost 32 to 33% into this your boron. Boron happens to be the very important elements for to this your nourishing the plants, for to this your fruit setting, and particularly for to this your horticultural crops, for its fruit signing, fruit setting, etc. And if this your soil is the deficient in zinc, automatically whatever we are getting to this your produce, that is a deficient into zinc. That's why today in the most of the medicine coming into this your market, that is being to this your aided from outsides. Even Government of India is also planning part to this your zinc fortification as per line up to this your iodine fortifications, and shortly that program will be launched. That is the condition of our soil. Morning, our Honorable Vice Chancellor, he this year emphasized about to this year poor nutrient use efficiency up to this year different, and especially part to this year nitrogen, where almost nutrient use efficiency is the 30 to 50%. Thanks a lot part to this year recent efforts, and particularly efforts taken by to this year GNFC coming with this year neem coated urea to enhance the nitrogen use efficiency. Friends, very shortly, And for to this your students' sake, this slide is the very important. We all know that the 16 type of these your elements are required by to these plants. Out of 16, that is the 3 CHO from to this your air and waters, and rest from to this your soil, and few are there are the major, few are to this your micronutrient that you all know. And if we go for to this your 1 ton up to this your yield up to this your wheat, it means 10 quintals up to this your wheat. So you require these much up to this your nutrient, that is this your removed by to this your 1 ton yield up to this your wheat, that is the nitrogen to 28 kg, phosphorus 4.4 kg, and micronutrients such as zinc 100 gram, this your copper 20 gram, and iron 100 grams. My point is that how to this your compensate micronutrient with a very novel approach up to this your green manuring. Here 1 ton if you are taking to this your green manure from to this your suspenia to this your chlorella, how that is going to this your supplement. I am least bothered about to this your how this your green manuring is supplementing to this your micronutrient, but it is very nicely supplementing, though that is in small quantity zinc, iron, magnesium, copper, but certainly that is giving to this your benefit to this your soil. And such benefits we are not getting from to this your chemical fertilizers. So we are having a very green option for to this your green manuring, that is second point here I want to say that once we are using a plenty up to this your chemical fertilizers, chemicals, in brief I say, how we are going to invite the problems up to this your different heavy metals, and heavy metals such as to this your cadmium to this your chromium, copper, lead, zinc, etc. And fortunately presently we don't have to this your much of this your problems related to this your heavy metals. Otherwise heavy metals problem is also very much, and especially into this your near the industrial areas, some of these your metropolitan cities, and much of these problems we are getting in the our leafy vegetables and some of these your root crops. That is a very challengeable task also. Coming how this your we have to, today's topic is the nurturing the neem for nature and to this your our livelihood security. Our every efforts must be to this your for low emission up to this your carbon dioxide and save this your energy. And we all know that almost 24.2% energy is utilized into these your industries, and out of 3.6% that is utilized into this your fertilizers making. Where we are going for to these your nitrogenous fertilizers, almost how that we are going to enhance to this your increase the temperature, and how we are releasing to this your different greenhouse gases. That is one of these your concerns. So if their option, or if that nitrogen is taken from this your, or some nutrients are taken from to this your our other sources, certainly that will be this your one of the most good efforts. Organic farming, very simple definitions, that is any production system that's aware to this your chemicals, that is the fertilizer, pesticides, growth regulators, like that. This definition has been coined by to this your Thomas Lumpkin. Earlier he was a DG up to this your AVRDC, and from there he moved to this your CIMITS DG, and a very comprehensive and very good definitions has been given by to this your USDA reports. Organic farming is the concept of a farm as an organism in which all the components, soil, minerals, organic matters, microorganisms, insect, plants, animals, and the humans interact to create a coherent self-regulating. In fact, this USDA reports that gives a very holistic definitions that we should not look our Mother Earth just as a production systems. we should be very holistic and we should be very poised thoughts that there are in the rhizosphere, not only there are the nutrients, there are a lot of numerous microorganisms are there, and that microorganisms, number of arthropods are there, and number of the beneficial insects are there. They are supporting to this our production systems. Unfortunately, we don't care, as we have started to this using recklessly or wanton way of use of to this chemical fertilizers. Most of to this field, if you visit, you will not get a single population of to this earthworm or like that. So that is the main problems. So if we are going to follow to this organic production system, certainly that will reduce to this our production cost, that will be long lasting, and secondly that will reduce number of to this hazards related to this. Pesticide is the major culprit. And how in the India we are going to this use to this pesticides. 62,193, that is the technical grade of to these pesticides we are using. Once we see these pesticides, that includes all insecticide, fungicides, and to these herbicides. Globally, these herbicides that is used to this more 56% by to this that average. But in India, we use less herbicide. That figures goes only to this 16%. In our major concern part to this pesticide is the major, our chunk goes far to this insecticide to this fungicides. It means we have surpassed to this global average. In India, almost 51% insecticides and 33% to this fungicide. It means 85% we are using only to this insecticide and fungicides. That as comparison to these herbicides are more deaded compounds. That is the scenario. So coming to this organic farming, that looks a very novel options, and globally that has got a good this year recognitions, because that practice is avoiding to this year chemical applications. And how this year globally we have got to this year area under organic farming. Presently we are having 74.93 million hectare area. And but how these your different regions this your share is that I have one, and further I will be saying to this. In India, we are having this your 2.7 million hectare area, but of the total cultivables area, we are having a very minimal area, that is the 1.5%, but that produce has got a very good up to this your export value, that is almost to this your 186 million up to this your export that is going on. Globally, because most of to this your concentrations on this your organic farming, we have paid 2000 onwards. So still our organic land in proportion to this your total agricultural land, that is the very minimal, only this your 1.6% areas. And for the students' sake, I will inform that Liechtenstein is the small country, very small country, up to this your European continent, and that has converted its 41% up to this your land under area organic, and followed by to this your Austria, that is also European country, 26.5%. So these are the two of these your top into this your as proportion up to this or their agricultural land to this your organic land. How this market up to this your and how this your liking or preference up to this your organic has increased in the world. In 2000, there was only 15 million hectare area. And by 2020, that is the current picture that has been given by to this your iPhone's websites, 74.9. So this indicates that there is the good concern, good liking. People are converting their land for to this your organic protocol or organic area. And same way how this your this has given to this your market price. In 2000, its market was only 18 billion, and by 2020 that has reached to this your 129 billion US dollars. Certainly this is having to this your immense scope of this your organic farming. Further, I will say one concern that most of this produce is going only into these developed countries. It indicates that the developed countries are more health conscious. That's why you look to this 45% and 44%, almost 80-90% that organic produce are going into this your developed countries, European and to this your American market, because they are having to this your price to this or pay the premium price. And same way to this year by 2015, this year per capita, this year consumption, in 2000 it was only 18 euro dollars, and in 2015 that has gone to this year 84 euro dollars. Different components of to this year organic farming, vermicompost, green leaf, crop rotations, that's what the different options you can add part to this your nurture to this your organic farmers. In past, we have paid a good attention to this your vermicomposting. Vermicomposting, no doubt, that has given a very good that is a very good aspects where we can go part to this your very small unit and we can produce. And major benefit of to this your vermicomposting is that it is having significant superiority over to this your conventional compost. And very important thing that it's this your very balanced CN ratio. And if there is the very balanced CN ratio, certainly that nourishes to this your soils and to this your different microorganisms. Second part of this your vermicompost is that it's a vermivash. Vermivash is having wonderful tonic. Even I have used during my to this your experimentations that has given a very good results onto this your fruit setting up to this your tomato due to its this your very hormonal actions as it is shown into these your pictures. Friends, one very important thing that's we are getting a very means a garbage, and in the city garbage also. And for that sake, I appreciate to this your Honorable Prime Minister that has given in 2014-15 how to this your city compost you can make. And for that purpose, 30 crores of this rupees was also allocated for that. And I'm happy to share that in all 3 years, with the government of India has taken initiatives, a good number of this year our city waste has been created, and that is a very useful effort. And this also support to
सर म्यूट हो गया आपका।
No, slide is not visible.
पंडित साहब लग रहा आउट हो गया।
Seems connection is not there. Slide.
क्लास ले रहे
Slide is not visible.
And some disconnection problem.
Visual that, sir?
Yes, sir. Visual.
Yes, sir. Visible, yes.
इसको slide mode में डाल दो. So, as I said that biofertilizer is a very good option, only with the few limitations. You look to this your picture in 1989, it was only 1,000, and 2014-15 that has gone to this year more than 80,000 metric tons up to this year productions of biofertilizer, and presently that is the 2.3 billion US dollar markets, and it is going by 2020 to 25 almost 3.9 billion dollar of the market. Very good efforts has been taken part to this or different type of this or biofertilizer for enhancing to this or nitrogen, then PSP to this or arbusculus, mycorrhizals, and different up to this or it's a endophytes and others. That is the efforts. A very primary work has been taken at our College of Horticulture and Forestry by how these were very visible efforts where to this your biofertilizer has been applied into this your fenugreek crops, methi. So that is even very green lustrous up to this your colors, and other way even you can enhance to this your self life also, and such type of these parameters are very much acceptable into this your vegetable cause. And two very important findings I could collect from to this your one publications from Vice Parmar University, where they have applied to this your Bacillus pumilus part to this cauliflower crops, and to this your biofertilizer part to this your pea crops.
डिस्कनेक्ट हो गए.
Signal lost.
Raghavendra, slide की visible to.
It's visible.
नहीं हो रहा. तो reconnect. Okay.
It's visible now.
Visible. So, our strategy for to this your next generation of to this your biofertilizers, as I said, that is the very unique gift for to this your nature and that mechanism. And recently, some effort has been taken part to this your making to this your biofertilizer rich biofuel. And certainly that will be the answer part to this your our most of this your biofertilizer experiments, where we complain that that biofertilizers gives very good response under the controlled conditions, but such type of this your response we don't get into this your our field conditions because of a number of to this your antagonistic activities, etc. So certainly such type of this your research will solve these your problems. Coming to this your need, right from to this your morning we are listening about to this your Neem. It's a number of potentials and very elaborative lectures after this your in first sessions by Dr. Sarpathy and others, and where they have very nicely highlighted that it is having a very good potentials, and it's this your but and different pesticide composition is having one unique properties that most of these your insect they don't develop to this your resistance. Present the major problems of that most of these your insect species are going to this your develop the resistance towards this your common application of to this your pesticides. Even he was Dr. Satpathy was citing about that the 500 insect has been reported to this year, has developed to this year resistance toward the
Signal is lost again, sir. You are not on the screen, sir. Slides are not visible, sir. There is some issue, I think.
Can present some other connection.
आई थिंक तब तक सर पहले वाले का रिमार्क्स दे सकते
एक साथ ही दे देंगे.
यस सर.
It will better to give at a time.
Yes sir, please.
Good task, no issue. But he can call to Pandit in your chamber, na?
Some connection problem with it.
He can present through your chamber.
हमारा फ्लोर ही अलग अलग है ना सर.
The same floor somebody will be there.
सर का देखो क्या हो गया दिन साहब का. वो शायद कुछ प्रॉब्लम आया उनके कनेक्शन में.
वीडियो चला देते जब तक क्या.
हाँ वीडियो चला दे सर.
वीडियो सेकंड है शेयर साउंड. हाँ ठीक है.
Agriculture is the backbone of the Indian economy. Farmers strengthen the spine of our nation. So long as the farmers are equipped and empowered with the best of resources, prosperity of India will grow in leaps and bounds.
Save water, save energy, save fertilizer. इस मंत्र को लेकर के हमने हमारे कृषि जीवन में आंदोलन खड़ा करना. हमारे देश में किसानों के नाम यूरिया जाता है, लेकिन वो यूरिया 15%, 20%, 25% केमिकल की फैक्ट्रियों में चला जाता है, रॉ मटेरियल के रूप में. यूरिया का 100% नीम कोटिंग करने का काम पूरा कर दिया. और इसके कारण अब ये यूरिया खेती के सिवाय किसी काम नहीं आ सकता. और नीम कोटिंग होने के कारण उसको जो न्यूट्रिशन वैल्यू चाहिए जमीन में 10% कम यूरिया उपयोग करते हुए भी उसको इसका लाभ मिलने वाला है.
Following the directive of our Honorable Prime Minister Shri Narendra Modi in May 2015, the Government of India made it mandatory to neem coat 100% of urea. This became the driving force of a unique initiative. An initiative towards harnessing the benefits of neem, a tree native to the Indian subcontinent and highly regarded for its many uses and benefits, and as an organic fertilizer. Gujarat Narmada Valley Fertilizers and Chemicals Limited, GNFC, implemented this policy mandate in an innovative, integrational and community-led, first-of-its-kind neem project. The neem tree sheds fruits for about 45 days every year. In Gujarat, this is during June and July. On an average, each tree sheds 25 kilos of seeds every season. GNFC successfully developed a process of neem fruit collection with the help of rural grassroots level organizations like fertilizer service providers, self-help groups, water societies, milk societies, and poor farmers. A large number of women involved in the process of neem fruit collection have achieved socio-economic empowerment as their additional incomes have grown substantially. A well-organized system has been established for buying neem fruits from them at a fair price, due to which the prices of the neem seed have risen from 1 to 2 rupees per kg up to 16 rupees per kg, an 8-fold increase. These neem fruits are processed to get neem cake and extract neem oil, which is further used in coating over urea. The seemingly simple process of neem coating over urea has created employment and earning opportunities on such a grand scale. The statistics speak for themselves. Total estimated annual urea consumption in India is about 31 million metric tons. Total neem oil requirement for urea coating is about 26,000 metric tons. And neem seeds required for extraction of this oil are about 400,000 metric tons. Since the initiation of the project in 2015, the neem project has seen great success. A collection of approximately 45,000 metric tons has generated almost 400 million rupees in supplementary income for around 450,000 women. An additional 200,000 rural people have benefited from indirect employment. Just imagine the results when the target of 400,000 metric tons will be achieved across the country. The project has been appreciated in numerous forums and has been honored by reputed award platforms including Porter Prize, awarded by eminent Professor Michael Eugene Porter of Harvard Business School. It also got a special mention by the Prime Minister during an interview to a TV channel in India in September 2016.
गुजरात में जीएनएफसी ने तो नीम कोटिंग करते समय आदिवासियों को नीम के फली इकट्ठी करने के लिए काम में लगाया तो उनको रोजगार मिला और अब वो नीम का तेल भी निकालने लगे और मैंने सुना है शायद 10-12 करोड़ रुपए का मुनाफा उन्होंने नीम के तेल में से भी पाया।
Heartened by Prime Minister's encouragement, GNFC expanded forward and trained women in manufacturing of neem soap, neem hand wash, neem shampoo, neem face wash, neem hair oil, neem mosquito repellent, de-oiled neem cake and neem pesticide. This has further improved the livelihood prospects of a large number of women. Recently, Honorable Prime Minister dedicated various forward integration plants of neem project and also laid the foundation for a big extraction plant of neem oil.
गांव-गांव नीम के पेड़ होते हैं, लेकिन कभी उसका उपयोग नहीं होता। आज जिस दिन गांवों में नीम के पेड़ होते हैं, जैसे आम के पेड़ के नीचे से फल इकट्ठे करने वाले लोग होते हैं, जामुन के पेड़ के नीचे फल इकट्ठे करने वाले लोग होते हैं, आज धीरे धीरे गांवों में महिला इकट्ठी होकर के नीम की फली एकत्र कर रही है और नीम कोटिंग के लिए कारखानों को बेच रही है। एनएफसी द्वारा ही जो नीम की फली खरीदी गई है इन गरीब महिलाओं को 40 करोड़ रुपयों की आय हुई है। 40 करोड़ रुपए। हमारे देश में वर्षों से सामान्य मान्यता रही है कि नीम का साबुन हो, नीम का तेल हो, ये शरीर के लिए अच्छा होता है। अब ये जीएनएफसी का साबुन अच्छों की धुलाई कर देगा। इसके लिए मैं जीएनएफसी को बधाई देता हूं।
A recent large-scale survey by UNDP of the socio-economic impact of the Neem project has shown a substantial increase in the average income of women, reduction in domestic violence, and enhanced role of women in economic decision-making in households. The project has transformed thousands of daily wage-earning women into rural entrepreneurs. The Neem project, an example to be emulated, has become part of case studies for students in Harvard University, University of Chicago, and Indian Institute of Management, Ahmedabad and Bangalore. GNFC has also been actively participating in formulation of policy decisions at the state and the central level to organize the neem industry in a way that makes it more transparent and fair. Ministry of Fertilizer has directed other fertilizer companies to adopt the Neem project model, and Ministry of Agriculture has laid down specifications of neem oil based on GNFC's recommendations. GNFC is currently hand-holding these fertilizer companies. Just imagine the socio-economic impact and enhancement in standard of living in rural India when the initiative is scaled at a national level. Just imagine millions of empowered rural women. Just imagine the unimaginable and see it happen. GNFC Gujarat is spearheading the Neem revolution, and other states are enthusiastically joining the movement. GNFC Neem, सबका नीम।
सबका नीम है।
So thank you for sharing this wonderful video from GNFC. Now I would like to request our Honorable Dean sir to please continue with his leftover presentation.
गौरव का नाम है शर्मा जी। शर्मा जी है वो है। इनके बाप हमारे यहाँ डायरेक्टर रिसर्च नरेंद्र देव।
I may request please mute yourself other than the speaker.
So friends, dear delegates, you got this your opportunity to again to this your have a very good video that developed by to this your GNFC. In fact, today's this your webinar is the very holistic approach to this your cover the different aspects, and I repeatedly thanks to this your GNFC for bringing such a beautiful informative video where with that venture up to this your neem coating area that has given to this your job opportunities, and how to this your once it was considered a waste to how this your from waste to this your wealth that has been converted. So friends, I was discussing about this your organic aspects and how this your neem can be pivotal role. In fact, most of to this your promotion of this your organic farming, we are getting the trouble with this your pesticide problems, and especially for some of these your crops, especially I talk about to this your vegetables, where vegetables being a succulent crops, we get a very much infestation of to this your pest, and on that accounts we spray a lot of to this your pesticides, and in response that comes to this your pesticide residue, and that how this your pesticide is harmful to this your health. You are all know that how these your cancers and others are these your problems are increasing. Morning sessions, I was very much happy to listen our very good speaker, renowned speaker, Dr. Saspati, and very good remark of Dr. E.V. Rai Sahab, and all these your good presentations were made, how this your neem is having to this your different of to this your it's a chemical properties, whether that is the azadirachtin to this or selenin or nimbin to nimidin, that is having one of these your very beneficial role, that they they don't allow to this your pest to become to this your resistant. That is one of to this your major attributes, major quality of to this your neem based pesticides. And certainly there are a number of researchable issues, whether azadi reactant content into this your and some part of to this your country is low or some part of to this your high. With these things that can be addressed by the some some new tools, and especially for our biotechnologists or persons concerned into this or genetic improvement of to this your crops can take care. And one very good aspect he was describing, that it is a very less harmful, rather you can say that is the free from to this your from to this your harm for most of to this your our predators, whether that is the Bavaria bassiana or to this your Caloperla cornea, or number of these your the predators are having a very good role, and their populations must be maintained. And once we go part to this your pesticide applications and very huge way up to this your pesticide applications, that time we don't harm only to this your deaded pest, but we harm to this your our beneficial pest also. So in that context, neem based pesticide is having one added advantage, and that its potential must be to harnessed. And how this your neem is a part to this your soil health property. Very good this your information has been given by neem coated urea, and it's a related concern, that is the less leaching, less this your nitrifications problem, less up to this your and secondly morning sessions our one of these your speaker was describing that once it is a neem coated urea, that avoid to this your nil gas and this your infestations also. So that are some of these your very good informations, and that slide is a very good part of this your how this your neem oil control to this your nitrifications process. I will shortly go that in future today's topic is how to this your nurturing the neem for nature. And as this your neem has got to this your very holistic properties, how it's this your oxygen releasing capacity is much more, that also it's a tangible and intangible benefit all should be taken into this your consideration, how it is giving to this your all it's this your social eco services to this your. And such type of this your problems we are going to face into this your future, as this your temperature are increasing, greenhouse gases are increasing, certainly that will cause to this your our very bad effect onto this your our availability of micronutrients, that will cause to this your less synthesis of iron, zinc and protein content, and our C3 crops will be most worst affected. So on that line, such type of this your organic efforts and such type of this your harnessing the potential of like this your plant, certainly that will be one of the good efforts. Even that's shortly slightly change into this your our food habit, that is also good effort, or that is also a good thing for to this your our environmental point of view, or releasing to this your less footprint. Friends, we have to look to this your all these your aspect in very holistic manner, and not into this your single phase. Multiple efforts, well knitted programs should be that. Then we can address to this your our SDG goal, and our 1, 2, 3, 1 no poverty, 0 hunger, and good health and well-being. And further 13, 14, 15, 13 this your climate actions, and 14 to this your life below water, and 15 on to this your life and land. All these are very much related to this your our agriculture and agricultural related activities. And in that gamut, our such type of effort as this your we have paid a very good attention for to this your enhancing to this your our nitrogen use efficiency through this your coating urea. Further, if you are going to harness the potentials of neem part to this your reducing to this your pesticidal hazards, certainly that will be the good things, and providing to this your such way good job opportunity to these your local peoples, and how to this your it's a one neem tree if planted. And morning sessions our honorable vice chancellor sir was citing that a neem tree almost having to this your 200 years of life. And if 200 years of life, if it calculates its total worth, that will come to this your millions and millions. So we should have such a visionary approach, such a vision to this your nurture such type of these your plants that has got to this your unique to this your capacity. As we are saying that single plant is having to this your pharmacy up to this your Ayurveda to this your Siddha to this your homeopathy to nutrient. One very important slides, only one or two slides I am going to so far to this your new efforts where we should make. We are currently we are emphasizing to this your natural farming and to this your organic farming. Our most of to this your literature what is being generated. That is, as morning this your Dr. R.P. Singh was citing about to this your NEEM's this your literature, that most of this your NEEM research on plant health management is concentrated on to this your pest problems. And once coming to this your fungal diseases problem, very less information has been generated onto this your bacterial and for to this your viruses also. That was a very pertinent comment. That was a very important comment. I was also exploring to this your literature. I got a very limited literature, but in recent 2-3 years, I was getting some of this your literature onto this your reducing to this your virus infestation through this your applications of to this your NEEM based on these your formulations. But certainly that needs the attentions. Much research should be focused on other aspect of also such as bacteria and viruses. Same way for to this your natural farming and organic farming. Here you look that maximum in natural farming, whatever this your literature has been generated, 41% its proportion goes only for to this your soil and nutrient, 23% yield. And greenhouse gas emission and to this your biodiversity loss, very less information. Health related, very less information. So our research should be focused on other aspect also. We should not concentrate only on the one aspect to this your give the this your fertilizer, enhance to this your yield. We have to this your or to this your very holistic approach to this your and especially the different climatic actions must be taken into consideration into this your while finalizing our research priorities. Same way for to this your organic farming, maximum information that is the on yield, gender, etc. Very less information that how this your organic farming is energy efficient, how that is saving to this your water, how that is good for to this your health, and how it is this your maintaining to this your biodiversity. In fact, all these are very major concern for to this your tomorrows. We should have such a to this your visionary approach. Finally, I will say that in last November this year in Glasgow, this year conference, one of this year very reputed persons, John Rockthorne, director of the Potsdam Institute of Climate Change. In the future, there can only be one form of agriculture, and that is the organic. All other practices do undermine any future possibility to do agriculture at all. Very holistic thoughts he has given. And one very Albert Howard, whom we say as a father of organic, he has mentioned health is the birthright of all living things. And certainly this your organic farming is having one of the very good components, very good way up to this your agriculture. And in that total gamut up to this your organic farming, natural farming, we are looking a very good scope up to this your harnessing the potentials of neem tree. Neem tree, if we say it is a panacea part to this your most of the problems, so it will not be incorrect. So with these words, I thank to this your organizer for giving to this your opportunity for on this aspect where we can harness or we can have a very holistic approach to this, your harness the potentials of Neem into this your organic farming, and in present day talk up to this your natural farming also. Thanks a lot.
Thank you very much, sir, for your informative talk. You also highlighted the organic farming and role of neem-coated urea, and also highlighted the content of heavy metals and information organic farming, its concept, components, and how globally the trend for organic farming is increasing, and there is increase in biofertilizer market. So thank you very much, sir. Now I would like to request our chairman and panelist of this session to share and review this session. Dr. R.S. Yadav, sir, please.
Thank you, Dr. Vinodji. First of all, I would like to give my congratulations for excellent presence in this session. Really, the preparation given by both the speakers was very prominent, required to take it further. And I can say that they have justified their lecture. As Dr. Patel Sahab, he has given all the steps required to generate, to produce neem-coated urea. And he has also highlighted the importance of this neem through the video presentation. Really, in our college days, we studied this role of neem-coated urea now, but today we have seen the practical use of this neem. And definitely, if I say by coating the urea by neem oil, that means the effect of urea as far as this greenhouse gases is concerned, it is reduced by 10% as far as contribution from urea is concerned. And he has rightly compared that this neem is a tree of 21st century. To take it further, I can say neem is not only for 21st century, it is a neem forever for the earth. In this, we have discussed on the role of neem seed. What about this neem leaves? What about this bark of the neem? They are also equally important. And the model which they have worked with for the collection of the neem seeds and the socio-economic impact also endorsed by our former Prime Minister, the importance of this seed collection. I think really it's wonderful to see his presentation, but at the same time, I also would like to put a question: is there any demand supply gap as far as neem oil, neem seed is concerned? Is there any demand supply? If demand is supply is short, then what should be roadmap for that? Because I think still crore of trees are there which are giving their seed, but we are not able to collect. There may be the only requirement is fulfilled by from the nearby places. That is the issue. And second, actually he has given the rate of this 8 rupees around per kg. I think that rate is only for collection. What about the trees from where they are getting the seed? They might be getting from individual farm. And what is the cost for that? Whether it's available free or only they are paying the cost towards collecting the seed. Really is wonderful, Dr. Patel Sahab, and the model of collection, I think it should be adopted by others also, those who are working in the neem. And Dr. Pandey Sahab, really, he has justified right from the present day problems where we are and where we want to go. And present times, government also emphasized the role of organic farming, natural farming. And I can say it will succeed only when when farmers have 4 or 5 trees at least in their farm from neem. There are many uses. No need to get oil from. There are many uses which can directly useful as far as promotion of organic farming, natural farming is concerned. And the major problem for adoption of organic farming, natural farming is that how to control the insect pest diseases, and for that Neem is the solution. And instead of seeking neem seed kernels and other things from outside, I think farmers, those who go for this, we should recommend they should plant the trees. But still, I can say every tree, irrespective of any species, they enrich the soil. But how to harvest that enriched soil, that is we have to see. And as far as organic farming is concerned or farming is concerned, crops require nutrients, whatever the source may be organic and organic, that requires nutrients. And can we think of making use of this leaves of the tree, this neem, because we are today discussing neem, and neem responds to 100% pruning. We prune it in at the onset of this winter. We can prune it 100%, and then again leaves comes, and then we can make use of those leaves in compost making. Then what is the effect? What will affect? Can we reduce the load of all these soil-borne microbes, everything? I think very little information available on those aspects. We can think of, if we want to make organic farming a big success, we should think out of box, and then no doubt any tree, including neem, they improve the soil. But every time if you take neem into the agroforestry where intensive culture is there, then you have to think how to plug the seeds and how many number of trees should be there like that, or only few trees may be sufficient on boundary itself. Neem takes a huge shape. Unlike other trees, it's having huge branching when it reaches to its optimum size. So then we have to see for management aspect of the neem tree as far as management of soil health is concerned. And really, I enjoyed both the lectures. And from this onward,
sir, neem is also very much useful part to this earthworms growth. Earlier, we were realizing that earthworm will not be benefited, but neem is also very useful part to this earthworms growth. In vermicompost units and other part to this worm growth, we can very frankly we can utilize to this neems.
Really, sir, it's very useful. And by citing the example, I will conclude my remarks. Because in organic farming, because you have discussed on organic farming, and I worked on organic farming project at Modipuram. There, almost not only in Modipuram, almost in all NR systems, we are supplying nutrients in terms of N equivalent. Then this quantity of organic matter to be applied in a maybe rice paddy system, paddy wheat system, it comes to tons. Then the eradications from where will get it? Really, it is. Then there was a thought at Modipuram. Then we applied biofertilizer by reducing 20% of required FIM, and that worked. That means by supplementing biofertilizer in organic farming, we can reduce the quantity of FIM or any other organic matter by 20%. Likewise, can we have some estimate by having trees of neem on a farm, and then by pruning of 100%, then we have to see the effect of pruning on neem, this yield of seed also, whether it is going to be increased or manage. Can we reduce the quantity required to raise an organic crop or a natural crop by composting of the neem leaves, or maybe other leaves we can add, other organic inputs we can add. So can it be? I think really it's a good, and I think by looking into the present problems and also looking into the emphasis of present government on the promotion of organic farming, nature farming, really this neem will play a good role, and we can adopt model of JNFC presented by Dr. Patel Sir, and the thought-provoking lecture by Dr. A.K. Pandey. By mixing all those things, we can make a roadmap how to move. And I think one more thing which I would like to say here, we do many webinars. I think the conclusion of this webinar should go into the hands which are policy makers. Unless we do that, really is very I think we are not serving our purpose. Anyway, it should go. But at least by getting learning from such type webinars, at least if they are not taken, we should start something new at our own places of work. With this, again, I congratulate both the speakers, eminent speakers, they have justified their talk in this session. And I also thank the organizers, Professor A.K. Pandey, Professor Dobrial, who invited me for this session. Thank you very much.
Thank you, sir.
Thank you, Dr. Yadav sir, for generous and kind remarks.
Thank you, sir.
I would like to clarify 3 points. If only GNFC is doing this by backward integration of seed collection and extracting the oil, if all the industry players will start, there will be a huge potential for employment. Number 1. Number 2, last year, real seed collector has actually got from rupees 8 to 16 rupees per kg. In Gujarat, they are selling on 20 kg basis. So they got 160 rupees per 20 kg to 280, 300 rupees per 20 kg. And number 3, I would like to clarify that GNFC model can be replicated all India wise. And major demand of GNFC, Neem seed is utilized for producing azadirachtin powder, dry powder. And there is a huge export potential. And people are using this azadirachtin powder for supplying other oils as a neem oil also. So there is some nuisance is there, but government is also trying for some good things, but it is very slow process. I would like to clarify. Thanks.
Patel sir, actually, what about the cost of seed from where you are getting? You are giving the price only for collectors.
After purchasing seed at the rupees 8 to 10 rupees at a grassroots level, our cost at the time of crushing will be 18 to 20 rupees. Because moisture loss will be there, weight loss will be there. Storage is very, very costly. Storage is very costly because it requires huge space. Because for gunny jute bag, only 5 to 6 bags stacks can be done. Not like urea bags, 20 bags stacking. And each and every jute bag should be kept open. So some space required also nearby. And 3 resupplying processes are to be done during July to October. Resupplying is to be done and rebagging is required because bags gets damaged and new bags are to be required to be used. So labor, loading, unloading, resupplying, jute bag cost, moisture loss, and 2 types of transportation. One transportation to primary godown and then another transportation to plant area. So it adds rupees 8, around rupees 8 rupees per kg to us. These are the secondary costs, I can say.
It's okay. Suppose a farmer is there having 100 trees of neem. A farmer is having 100 trees of neem. That farmer wants to sell the seed of neem. So you are going to give 8 rupees.
Yeah, we are giving 8 rupees. Actually, in Gujarat, market has become competitive and players from the other states are also coming to purchase seed from Gujarat also. They are purchasing seed from MP and Maharashtra also. And the market is self-driven now. Market is already self-driven now. No support is required also. Sometimes we are also facing difficulty to purchase the seed at the right price. Many times market is get inflated also.
That means 8 rupees includes both the cost of labor plus the cost of the seed which owned by somebody.
Pardon?
That means 8 rupees includes both the cost of collection of the labor plus to be paid to the farmer.
Yes, collectors are collecting from the open field. They are not getting from the farmer or any field. Actually, small farmers and landless laborers are collecting this seed, particularly women. And many places, adolescents, girls, and children are also collecting it during their free time. They are doing like playing activity.
Then farmers are allowing to collect freely.
Yes. Why not? Why not? Because big farmers are not interested in collecting this seed. They feel that this is small activity. And generally, seed are collected from government space, gochar land, roadside area, and other things. Thank you.
So thank you very much, sir. I thank you, the chairman, Dr. R.S. Yadav, sir, for viewing this session. And I'm thankful to the panelists and eminent speakers for this session with such an informative discussion on NEEM and its potential for soil health management. With the permission of the chair, I want to end up this technical session and ask my colleague, Dr. Pawan Kumar, to please be started with the next technical session of this national webinar. Thank you. Thank you very much, sir.
Thank you.
Okay, thank you. Dr. Pawan.
Hello.
Yes, start please.
Okay, sir. So, good afternoon to all of you. Myself, Dr. Pawan Kumar. Welcome to chairman, panelists, speakers, reporters under the theme genetic resources and the improvement of the NEEM. So now I am going to introduce to our chairman, Dr. S. Biswas sir. Dr. Biswas has over the 49 years of the experience in the research, education and the training activities in the forestry and the allied subject. Presently, Dr. Biswas is professor and head in Department of Forestry, Dolphin, postgraduate, Institute of Biomedicals and the Natural Sciences, Dehradun. Formerly, he headed the Forest Botany Department of World Famous Research Institute, Dehradun. Dr. Biswas imparted training, education, extensions, and awareness in the various forestry and allied programs. He has over the 150 publications to his credit. He has been visited professors in the different forestry and educational institutes, presently working on the germplasm characterizations of rare threatened biodiversity and the FGR of the distinguished landscape for the conservation and the management. I welcome you, sir. The second one is I welcome to our panelists, Dr. Ashok Kumar and the Dr. KP Mahapatra. Dr. Ashok Kumar, scientist, G, Division of Genetics and the Tree Improvement, Forest Research Institute, Dehradun. Dr. Ashok Kumar is a scientist at the Forest Research Institute, Dehradun, and working on the tree improvement and the biotechnology for the last 25 years. His contribution in the field of the tree improvement and the biotechnology have been well recognized world over. He is the only Indian so far to receive the prestigious Dr. Y.S. Rao Forestry Research Award in 2003 of the Asia Pacific Association of the Forestry Research Institute. He has also been awarded his Brandeis Prize twice of his scientific achievement and the publication. He has very recently released the 6th variety of neem with characteristics of the high oils and the aza contents and ability of flower and the fruit from 3rd year onward. He has also released on clone of Dalbergia siso for the northern India. The clone has been in great demand by the growers owing to higher productivity and the disease resilience to the dieback of the session. His 10 released variety of the Milia dubia are in great demand of the commercial cultivation with the average productivity of the 34 million per year, 34 million per hectare per year. Now I welcome to the Dr. KP Mahapatra sir. Dr. KP Mahapatra, principal scientist, has obtained his master and the PhD degree from the Dr. Y.S. Parmar University of the Horticulture and the Forestry, Solan, Himachal Pradesh, in the tree improvement and the genetic resources. He was introduced to the Indian Council of Agriculture Research through the ARS and started his career in the Indian Institute of the Soil and the Water Conservation, Dehradun, in the Division of the Plant Science. He has more than 20 years of experience in the research, teaching and the extension, guided 5 MSc and the 1 PhD student and published more than 60 research papers in the national and the international journals. 5 books and the many scientific other documents. Presently, he is serving the National Bureau of Plant Genetic Resources, New India, as principal scientist in the division of germ plots evaluation. Now I invite to the Dr. Swati Segde, reporter of this event, to introduce the speaker of Dr. S. Vanilla and the Dr. K. Rajarajan. Ma'am, please.
Thank you, Dr. Pawan. Very good evening to all delegates and dear colleagues. I am Dr. Swati Shedge, working as an assistant professor in the Department of Forest Biology and Tree Improvement, College of Horticulture and Forestry, Rani Lakshmibai Central Agriculture University, Jhansi. On this first day of national webinar on Neem and under the theme Genetic Resources and Improvement of Neem, our first speaker is Dr. S. Vanilla. It gives me immense pleasure to introduce Dr. S. Vanilla. Madam S. Vanilla currently working as assistant professor at the Department of Forest Biology and Tree Improvement, Forest College and Research Institute, Tamil Nadu Agriculture University, Mettupalayam. She has been working there for last 10 years. Under her research profile, she is working on tree breeding and improvement of industrial wood species, improvement of pulp and paper wood species, then the improvement of natural dye yielding species, and also working on the improvement of neem. She has written about 5 books, published 15 books chapter, and more than 30 research papers in the sphere of forestry in the peer-reviewed national and international journals. Important books on her name are Objective Forestry for All Competitive Examination and Subjective Guide for IFS Aspirant. I welcome Madam Vanilla to share her knowledge and experience on genetic resources and improvement on neem. Ma'am, you're mute. Unmute yourself. You're not audible.
Unmute, Dr. Vanilla,
yes,
unmute.
Yes,
Okay.
yes, sir, yes, sir. Yes, sir. So, good evening, one and all. Today, my seminar topic is genetic improvement of neem for oil quality and alkalis. So the first introduction of this species is the neem is one of the most suitable and valuable tree species found in India. So among the 202 families, the Meliaceae family is the most economically important species because almost this species is its economically utility for all parts of the species. So it's native to Indian subcontinent. So more than 25 million trees growing all over the India. So among the states, so Uttar Pradesh is the most predominant distribution of neem trees. So that is 55.7%. The next one is Tamil Nadu, that is 17.8%, and the Karnataka, so that is 5.5%. So the seed production is 4.42 lakh tonnes, and the neem oil also 0.88 lakh tonnes. The neem cake is 3.53 lakh tonnes. So that is 10 to 12 years old neem trees, they produce yearly 30 to 60 kg of fruits per year. So this neem trees are otherwise called as arista. That is, arista means that is a liver of sickness. Okay. So the next one is this species is mostly used for biopesticide. So the next is a major distribution of neem tree in India. So this it's a major distribution is Uttar Pradesh, the predominantly distributed, that is more than 7 lakh tree species, and the next is Tamil Nadu, that is 2 lakhs 54,000 species. So what are the major drawback of this, otherwise character of this species is it's a high flower drops. So that is 2% of the flower is converted to fruit. So another one is a pollination. The pollination also amphophily. The amphophily means the pollination by both wind and insect. And another important problem is incompatibility of flowers. The next one is the fruit set was low in natural pollinations. So because the flower is converted to fruit is less than 2% of the flowers. So the coming to tree improvement programs, because this species is mainly used for, one is azadirachtin content, another one is oil content. So based on that objectives, we frame for 300 species are screened. So 300 species are identified. Among the 300 species, only we identify 34 plus trees. So based on the morphometric traits, for example, height, girth at breast height, clear bole height, seed yield, oil percent, azadirachtin content. So based on that morphometric traits, we screened from 300 species to 34 plus trees by using subjective grading method. So these are the plus trees identified the two states. One is a Karnataka and another one is a Tamil Nadu. Karnataka like Bangalore, Chitradurga, Chikbellapur, Mysore and Samrajnagar. And Tamil Nadu, it's a Coimbatore. So the totally 34 plus trees are identified based on the oil and azadirachtin content. So the next one is the fruit variations. So the fruit variations are present in the 34 plus trees. So highly the fruit length, fruit birth and 100 fruit weight and fruit to seed ratios. This was highly observed from one plus trees to another. It's a highly variable. So for examples, Karnataka, that is Chikbellapur, it's a highly, that is a recorded fruit seed ratio, it's highly recordable, that is a 8.58%. The next one is Tamil Nadu, Mettupalayam, 07 is high observed in 8.46% of fruit to seed ratio. So the next one is the seed variation. The seed variation of trees, the seed variation of plus trees. So it's a significant variations are present in that seed morphometric traits. So for example, seed length, seed birth, 100 seed weight, kernel weight, seed to kernel ratios. So that is because of that, because the oil content and azadirachtin content only, it's very, very important. Based on that purpose, we observed seed characters and fruit characters. After that, that is a seed biochemical characters. So based on that 34, among the 34 plus trees, we observed high aza species, high aza plus trees observed in, that is a 6 plus trees. For example, Karnataka, that is a CHB, that is a Chikbellapur 03, that is we observed 0.972% of azadirachtin content. The next one is Samrajnagar, that is 0.970. The next one is a Mettupalayam MTP 11. This plus trees was observed in 0.964% of the azadirachtin content. The fourth one is a 28, that is 0.936%. The last two is a Bangalore, that is was observed in 0.943 and 0.936% of azadirachtin content. So based on that aza content, so we selected 300 trees. Based on the aza content, we only screened and we established the seedlings in our nursery. That is a high aza planting. So this is an overview of our nursery. So the next one is a seed germination. So based on that, we selected the plus trees. We conducted the nursery evaluation and field evaluations. Based on that, the nursery evaluation studies, the high germination percent was observed in Chikbellapur 03 and Tamil Nadu Mettupalayam 10 and Mettupalayam 28. So these 3 plus trees was observed high seed germination percentage. So the next one is a nursery evaluations. That is a 180 days we conducted the nursery evaluation trial. So we observed that is the shoot length, fruit length, collar diameter, fruit shoot ratio and total biomass, and that is a volume index, vigor index. So based on that observations, we observed TN-MTP 08 and TN-MTP 21 and Karnataka CHB 02 observed higher performance compared to other plus trees. So this is a progeny evaluation of selected genotypes. So we adopted the design is RBD, the replication is 3, the total number of progeny is 34, the spacing we adopted in 4 into 4, the number of progenies per replication is 9. So this is our overview of field, that is a progeny evaluation trial. This progeny evaluation trial conducted at Forest College and Research Institute, Mettupalayam. So based on the progeny evaluations, so we identified the superior 5 plus trees, that is a that is a Chandradurga, that is 14, and Metupalayam 8, and Metupalayam 14, Metupalayam 21, and 34. These, based on the progeny evaluation, we observed maximum, that is a height, basal diameter, number of branches, and volumes. So the next one is the application of plant growth hormones. Because this species is major drawback is only the 2% of the flower is converted to fruit set. So we improve that 1% increase in the fruit yield also. So that's why we sprayed some, otherwise we treated some plant growth hormones in flowers. So in this view, we framed 11 treatments like naphthalene acetic acid 100 ppm and 200 ppm, and ethylene 500 to 500 ppm and 1000 ppm, mafecude chloride 500 ppm to 500 ppm and 1000 ppm, chloromepcute chloride 500 ppm, 1000 ppm, and tricotinol 500 ppm to 500 ppm and 1000 ppm. And another last one is it's a control. So this application was conducted before that before the flowering time. So otherwise primordial time. So that's why we conducted this experiment is last year February. That is more we sprayed the plant growth hormone is, that is at the time is it's a morning 6 to 9 a.m. So after that spraying that plant growth hormones, we completely wetted with flowering hormones and frequent irrigations. Because of that, that is they dried frequently. That's why we sprayed irrigations. So the statistical design is RBD, that is a randomized block design. The total number of treatment is 11. The replications 3. The trees per replication is 3. So the next one is based on the application of plant growth hormones, how the flower traits was improved or increased the flower average flower per tree or number of inflorescence per twinkle. So based on the 11 treatments, we observed tricotinol 1000 ppm, observed a maximum number of the flowers per tree. And the next best treatment is ethylene 1000 ppm. So this also observed high average flower per tree and average fruit to seed average flower to fruit set ratios. The next one is the application of plant growth hormones on fruit and seeds treatments. So among the 11 treatments, the tricotinol at 1000 ppm, so the 100 fruit weight is highly observed in the tricotinol 1000 ppm, and the fruit length also observed in mepicute chloride at 500 ppm. The fruit width observed in ethylene at 1000 ppm, and the 100 seed weight of the neem tree is observed in tricotinol 1000 ppm, and the seed length and seed width also observed with the tricotinol at 1000 ppm. The next one is the application of plant growth hormones and seed yield. So this application is so after that the flower is started and they converted to seed. So how much of the flowers is converted to fruit? So based on that, that is a fruit yield per tree is 7.96 kg. So it's a control only 2.46. The tricotinol 1000 ppm, it's increased the yield of 12.33 kg per tree. The seed yields only 3.76%. So the control only, that is 6.066 only the seed yield per tree. So the next is what is the phenological behavior of treated neem trees? So already the treated plant growth hormone is earlierly they induce the flowers, and also earlierly the end of the flower setting is earlier. So that is a tricotinol treatment is at 500 ppm and 1000 ppm also. The flower initiation also it started earlier, And the end of the time, the end of the fruiting also it's earlier. That is so it's a take 108 days. So that is the control. They start the flowering is May to September, up to September. But the trichotinol, the treatment, the started the flowering is March 22. That is the end of the March. And normally the flowering started May last. So with this treatment, the flower initiation also start earlier and the ending also it's earlier. It's take only 108 days. So the next one is supplemental pollinations. So this species, that is a self-incompatibility, is a major problem of this species. So we conducted, that is a fruit, that is seed, flower to fruit set ratio also it's less than 2%. So within this, so the fertilization, how to improve means that is a pollination method. So the different pollination methods was introduced. One is a natural pollination, closed pollination, and supplemented pollination. Before this method, first we collected the pollens from the seed, from the trees. So the collection of twins before the anthesis period. After that, that is a detachment of anthers. After the detachment of the anthers, so we kept in this that anther is one night in dark place. After that, the pollen was removed by brush. So how much the pollens are present in that one flower? So that the instrument is hemocytometer. So based on that hemocytometers, one single flower they found in 11 anthers. So the 11 anthers they found in that 3,744 pollens are present in that one single flower. So the next one is that is a supplemented pollinations. So already I told that is a closed pollination and hand pollinations. Open pollination, and another one is a supplemented pollination. So based on that supplemented pollinations, we fixed for 5 treatments. That is 0.1% of the pollen, and we be using some of the chemicals like boric acid and 0.1% agar, and water and sucrose and basal sucrose solutions. So these are the solutions. These are the chemicals. It's inducing the pollen tube. So based on the treatment, among the supplemented pollination, that is the chemicals and closed natural pollinations. So the fruit setting is, that is a fruiting of the flower to the flower is converted to fruit is, it's nearly 21. So the next one is, that is a fertilization percentage also, it's increased 8.86%. So that is 2.5 gram of pollen per liter of water. This treatment is increased that fertilization percentage. This is overview of supplemental pollinations. So first one is the collected the anthers from neem trees, and the next one is the spraying of chemicals. So the next one is the supplemented pollination of neem fruit. How to variation from one treatment to another. So fruit length also it's highly observed in 0.1% of the pollen and boric acid 100 ppm, and the fruit width also observed in, that is a 2 treatments, that is a 0. boric acid and 1 liter of water from, that is a 2.5 grams of pollen. And the third one is that is a 100 fruit weight, that fruit weight is observed in 0.1% of the pollen and boric acid 100 ppm. So the next one is supplemented pollination on neem seed. So how to vary the neem seed traits? So based on the treatments, so the seed length also it's increased. So for example, that is a closed pollinations was observed in 10.33 millimeter, and the natural pollinations was observed in 13 millimeter. But the treated flowers, the seed length was observed in 15.33 meter, and the seed width also 9.24 millimeter. The next one is a 100 seed weight was observed in natural pollination, and the kernel weight also 10.70% was observed in 10% of sucrose plus 0.2% of the boric acid. So the next one is the supplemented pollination on oil and azadirachtin content. So this was highly observed in, that is a natural pollination, and also that is a 0.2 m basal sucrose solution. So that is this treatment is compared to high oil and azadirachtin content, but so only that so that is a 0.1% of the pollen plus 2 grams of pollen is highly recorded that fertilization of seeds. So the next the experiment is the pruning techniques for increasing the seed productions. So major problem of this, it's how to improve that soil and seed quality, seed productions. So we conducted that 3 pruning techniques. That is we framed four treatments. That is a 15% of the pruning and 25% of the pruning, and the third one is a 45% of the pruning. Among the 4 treatments, the 25% of the pruning is recorded high flowering morphology and fruit and seed traits and traits. So that is a flower traits. That is the 20% of the pruning is recorded number of inflorescence per twig, average inflorescence length, the average number of flowers inflorescence, that is the average number of fruits developed. That is a fruiting. So these that is a 25% of the pruning techniques is highly recorded to increasing the flower, increasing the inflorescence rate and flower fruit ratios. So this is also same. So based on that, our 300 selected the species, only 34 progenies is high aza content. Earlier, only the 0.6% is a aza content was reported in literatures, but we assembled more than 0.8 to 1.12% azadirachtin content plus trees. So based on that azadirachtin content, we identified superior high aza with oil content like the TN-MTP 72, the TN-MTP 54, and KA-CHB 03, and MTP 28 and CTP 17. So these are the species high azadirachtin content. And already I told that is a floral induction, that is a trichotinol. It's increasing the flower induction to earlier, and also the flower to fruit conversions also, that is a fruit set ratio is increased. So the trichotinol is the best treatment for flower induction, and also the 25% of the pruning intensity also increasing the seed yield. So we identified the sum of the plus trees. So with the Coromandel International Private Limited, we collaborated, we signed that MOU. So based on that MOU, we that is a Neem contract farming we framed. So based on that requirements, the Forest College and Research Institute, they produce the quality planting stocks, and also they give the technical guidance to farmers and also industries. And the industries give the assured market price to the farmers. For example, that is a minimum support price based on the prevailing market price, that is a past 5 years, or that is support price. Whichever is higher, they give the minimum support price. So way to forward, based on that our research, futurely we developed one variety. So that is with high aza and oil content, and also optimizing the harvesting index for maximum aza extraction. And the mechanization is a very big problem for because the tree is a very big trees. So the harvesting of fruit also it's a very big problem, because that is after ripening of the fruit, the aza content is low compared to that is a green content, that is a matured fruit, only the high aza content. So the harvesting of fruit, seed collection and extraction and post harvest, that is these all are it's in mechanized, will improve the techniques. And also the canopy management studies, that is a minimum, that is a 25% of the pruning intensity. It's also increasing the seed production and oil content, high density also. So thank you, sir.
Thank you. Dr. Pawan.
Thank you, ma'am. Thank you for sharing your research experience on neem improvement by using different techniques like cluster selection, progeny evaluation, and application of plant growth hormones with the spraying pollination method and different pruning methods or pruning techniques. Thank you so much, ma'am. Next, I would like to invite our next speaker, Dr. K. Rajarajan. Presently, Dr. K. Rajarajan sir is working as a senior scientist in the genetics and plant breeding at the Central Agroforestry Research Institute, Jhansi. He got his doctorate degree from Tamil Nadu Agriculture University, Tamil Nadu in genetics and plant breeding. His research interest or specialization is especially in molecular biology and biotechnology in agriculture. He received several awards in various organizations.
Please, other mute yourself. Please mute yourself.
He is a member of several professional societies and in various research journals. Dr. Rajarajan sir published more than 20 research papers in national and international reputed research journals, and also written various technical bulletin, book chapters, proceedings paper, and popular articles. He also organized numerous seminars and trainings. He has more than 9 years experience in the field of genetics and plant breeding. Currently, he is handling 7 externally funded research project at CAFD, Jhansi, and working on the neem germplasm evaluation for higher oil production by using molecular marker technology and tree genomics. Thank you, sir. So I will request Dr. Rajarajan sir to address participant on tree improvement aspect of neem. Thank you.
Thank you, madam. Respected dignitaries and viewers, good afternoon to all. I'm going to present on neem genetic resources and its genetic improvement. Yeah. So, neem, as we know that neem is one of the most important tree, which is since from way back. It is associated with human life in the form of socially and culturally as well. And coming to the introduction, I don't want to go much on introduction since from morning we are a lot of things we are hearing about the neem. So I'm straight away entering in the topic that Neem Azadirachta indica is an attractive evergreen tree that grow up to 20 meters tall. And basically this is a diploid species having a chromosome constitution that 2n is equal to 28. And neem native to Burma and Sivalik, Deccan and other parts of southern India are to be its primary native. And apart from that, this species also invasive to various countries like Australia, Philippines and Saudi Arabia as well. And basically 2 types of neems are there. One is Azadirachta indica, that is called Indian neem. Another one Azadirachta indica variety, Hermensis, that's Thai neem. And coming to the significance of neem, as we know that the major demand of neem is for seed oil because it contains several bioactive compounds which is responsible for antimicrobial activities. Neem products are being used to treat malaria, fever, pain, and also it is used as contraceptives. Azadirachtin, as we know that among the various bioactive compounds ranging from nimbin, salanin, azadirachtin is a major compound, a highly oxidative limonite, predominantly present in the neem seed kernels, which are responsible for diverse biological activities. These activities are including insect repellent, antifidant, larvicidal, and also growth inhibiting properties against wide range of pests. And so that it has been well recognized as environment friendly and biodegradable pesticide. This is why the world has been attracted towards the neem as of now. And coming to the origin and distribution, neem have been originated in Assam and Myanmar, where it is common, and throughout the central dry zone and Sivalik hills. Here only the neem primarily originated, and then later in 19th century, neem was introduced by an Indian immigrant to the Caribbean island and South America and parts of South Pacific region. And then the cultivation of neem spread to Africa in 1920s when it was introduced to Ghana, Nigeria, and Sudan. And now currently the species is well established in more than 30 countries in the world. And coming to the India, the neem distribution ranges from Uttar Pradesh, Bihar, West Bengal, Orissa, Delhi, Maharashtra towards south. And of course, in India, neem is largely distributed in Uttar Pradesh and also some parts of southern India, particularly Tamil Nadu and Karnataka. Coming to the neem genetic resources, as we know that the genetic resources are the valuable part of tree breeding, particularly these genetic resources holds many valuable genes, and these genes can be utilized in the future when time to time depending upon our needs. So coming to the neem genetic resources, this is the exploration of germplasms, or the genetic resources are the major and very fundamental baseline activity in any tree breeding activity. And so the explorations, as far as concerned in neem, it can be done in two ways, that plus tree selection and provenances collection. In that way, it can be done. Once the superior tree for a trait of interest has been identified, the tree breeder usually will select the particular tree which are phenotypically superior for the traits of interest, and the seeds can be collected from the single tree and can be used as subsequent breeding programs. And coming to the provenances collection, here generally the genetic gain will be more in the plus trees, because we know that the trait of interest, according to our goal, is it's perfectly captured in that particular plus tree. And coming to the provenances collection, this is provenances collection also have very strong differentiation for adaptive traits particularly. And here the locality has been represented by 10 to 20 traits, and these trees, as the seeds will be collected and composited and represent as the particular locality and called as provenances collection. And these are the major parts of exploration. And coming to the next part is conservation. This is again a most important part in neem genetic tree resource maintenance. The conservation of neem germplasms can be done in two ways. That one is it can be done in the primary distribution, where it has originated in the form of gene sanctuary or biosphere reserve. And second, it can be maintained in the form of field gene bank. Also, the seeds can be stored in cryogenic condition for long-term storage. So in that way, it can be neem genetic resource can be conserved. And further, once the conservation process is successfully completed, then we can utilize those conserved germplasms for various tree improvement programs depending upon our needs. And as far as concerned in neem, the seed productivity or the kernel productivity are the key areas to be improved. So also, besides quality characteristics, also very important as far as concerned in neem, particularly high oil quality, high bioactive compounds like azadirachtin. And third, as of now, we are due to climate change, adaptation to the wide range of climates also, we have to be focused very well. And this can be achieved through germplasms which are conserved in the form of field gene bank or the gene sanctuaries. And these existing genetic variabilities could be used to produce new gene combinations, and that will further add up to the genetic diversity of the conservation pool, and that can be again utilized. And all these Neem genetic resources activities can be, we can think it in that way, that kind of pre-breeding activities for successful tree improvement. And coming to the Neem genetic resources at Kafri, Jhansi, we can say that the country's largest Neem reservoir is maintained by Central Agroforestry Research Institute. And initially, we had 270 accessions collected from various states, from south to north, particularly from Andhra Pradesh to Rajasthan. And these explorations were made during 1993 to 2006, where various trips has been made towards exploration. And then as of now, we have 170 accessions at our field gene bank. Among that, 139 accessions were received IC number from National Bureau of Plant Genetic Resources of New Delhi. And further, the remaining germ blossoms also we are in the process of registration and further getting IC numbers as well. So this is about the germ blossom status of Kafri, Jhansi. And coming to the genetic variation of Neem, as we know that Neem is highly variable species within its native and introduced ranges. And Neem is basically a cross-pollinated species. Then it is in the native and the introduced ranges, generally the variation will be more, and the species shows variation across also different agroclimatic zones in the seedling as well as for the mature or adult tree growth for various growth characteristics. And particularly if you see that there is a relationship that the growth of Neem and the production of azadirachtin. Say for example, for the lower rainfall areas showing slower growth but higher concentration of azadirachtin, vice versa. And if you see that the growth, particularly the adult tree growth characters like tree height, collar diameter, TBH, clear bold length, and canopy diameter also are highly variable characters. And in the Neem, in the case of Neem. Coming to the fruit and seed characteristics, the fruit and seed characteristics like fruit weight, under fruit weight, fruit length, width, diameter, yield, and azadirachtin and oil content are highly variable both within and between provenances of Neem. And if you see that the seed shape also ranging from oval, long, ellipsoid, and if you see the 100 seed weight, it ranges from somewhere 11 to 16, and then 27 to 33, likewise 17 to 22. Like in seed color also, you can see the variability that white, gray, brown, like. So if you see in this table, so many quantitative characters under the range, and so this very clearly indicates that the variation for fruit and seed characteristics are a wide scope in Neem to select. And so this is about the variation on Neem, about for the growth characteristics as well as for fruit and seed characteristics. And of course, the focused objectives for Neem genetic improvement would be assessment of genetic diversity and variation of Neem. And of course, assessment of genetic diversity is a very fundamental step in any tree breeding activity that determines the success of the tree breeding. So in that way, this is one of the major objectives. And of course, conservation, genetic resource conservation is again an important objective. Then genetic improvement and production of improved planting material. Once the successful tree or the provenances has been identified, it has to be mass multiplied in order to have maximum growth, so maximum yield and productivity. Coming to the fourth one, if you see characterization and identification of high seed oil germ plasm or varieties has to be identified and released, so that this will enhance the production and productivity of Neem. And then finally, the bioactive compound like azadirachtin. The variation for azadirachtin also equally important, that because Neem is having diverse utilization. So in that way, this has to be focused well. And these are the possible genetic improvement methods in Neem. As we know that the methods, we can say that conventional tree breeding techniques and modern techniques. We see that conventional tree breeding techniques, it ranges from plus tree selection, progeny testing, provenance test, vegetative propagation, establishment of seed orchard, then clonal propagation, or the conventional tree breeding techniques for Neem improvement. Of course, then modern techniques, if you say that in vitro propagation or tissue culture techniques, then marker-assisted tree breeding, and very recently the omics tools, particularly the transcriptomes, which focus on oil biosynthesis pathway and azadirachtin biosynthesis pathway, which gives an immense scope on working on to enhance understand the system biology and to develop appropriate tool to identify and hasten the breeding activities particularly. And if you see that Neem long-term breeding strategies would be like this. First, we have to introduction of provenances. We have to introduce the provenances. Then followed by we have to establish the seed production areas. Then we have to make genetic selection and testing. We have to make followed by the selection of plus trees based on the provenances should be identified. Then the plus tree progeny should be evaluated for the performance of plus trees, and then based on this we have to establish the seed orchard for long-term genetic gains for our trait of interest. Then from this we can straight away go for we can go for clonal strategies. There we can develop clones, for superior clones to multiply rapid multiplication to maintain the genetic purity as well. Then finally, the conservation of genetic diversity. This can be done in both a long-term and short-term breeding strategies by population improvement technology particularly. And this population improvement technique will give more genetic gain and wider genetic base in the population in course of time. And that will be utilized for the future breeding activities depending upon our need and trait of interest. I would like to have some small research highlights which was happened in at the international and national level. And of course, in before 90s and 1994, there was no systematic Neem genetic improvement activity in the world. In 1994, the International Neem Network was established with the objective to study multiple sites genetic variation and particularly adaptation and of growth of provenances, including the provenances trials. And then the network collaborators decide to undertake activities in relation to provenance exploration, seed collection, exchange for establishment of internationally coordinated trials. And then they also decided to undertake research in particularly seed physiology and technology, genetic diversity and reproductive biology, because this area was so lag behind at the time. And it is they identified that this area should be much focused and the attention must be given on that area to further Neem genetic improvement. And at the time, the countries collaborated in INNA and the corresponding institutes were shown in this list. As far as concerned in India, ICFRE was the focal point institute at the time in India. And coming to the various provenances trial and that designs made in different countries and number of trials established in the table you can see. And coming to the India, the highlights of Neem improvement, you see that of course FCRI Metropolitan has the very oldest, one of the oldest provenance trial established at 1991, and the focal area for the selection criteria was seed and high azadirachtin and oil content. And then followed by 1992, A4A established a large provenances trial of Neem containing 40 provenances from 10 Indian states representing different agroclimatic zones. Then that AFRA in 1998, the best performing provenances were identified at Rajkot, Gandhinagar, Amaravati, with increase in the height of 5.80, 5.4, 5.35 meter respectively. Also, ICR, Kasri have a collaborative project with the AFRA funded by NOVAD project, and they have also evaluated the various genetic resources, and they identified some of the provenances for the target traits. And coming to that provenances and progeny trial at Kafri, if you see that we are working since 1994 so to till date that on Neem. And during that, we, as I earlier said, that we have made a lot of exploration trips, and we have conducted various provenances, progeny trials, and from time to time, and also we have established a seed orchard for these important key traits as well. And we have in that seed orchard has been established with 6 outperformed progenies for various key traits, particularly high oil content, and because our objective was even clean pole also. So these have been included, and we have seed orchard. And coming to the modern tree breeding, Neem genetic improvement, we can say that with the biotechnological intervention, the modern tree improvement activity was set out. In that, we know that the conventional breeding programs have its own difficulty, that it's due to the prevalent heterozygosity, perennial nature of tree. There is a major constraint in conventional tree breeding, and to overcome that, biotechnological interventions are much more helpful to efficient Neem genetic improvement. In this regard, of course, plant tissue culture is an alternate a technique for quick and mass multiplication of plus Neem trees. Various explants ranging from nodes, leaves, zygotic embryos, endosperm, nucleus, cotyledon, protoplast, and then anther ovary culture have been used in Neem so far for optimizing and developing various micropropagation techniques in order to have a mass multiplication of superior trees identified for various key traits. Also, the in vitro raised cultures from many of the above stated experiments have tested positive for presence of important metabolites like azadirachtin, not only to produce mass multiplication, also to produce azadirachtin also can be, other than azadirachtin, a kind of secondary metabolites like other bioactive compounds also can be produced from these tissue culture. So they're having a great breakthrough in Neem genetic improvement. And these are some of the micropropagation techniques in Neem genetic improvement they studied. Of course, marker-assisted rebreeding. This is, I can say, this is one of the most important breeding tool in Neem genetic improvement, I can say, because the genetic diversity of Neem tree species in any region of the world may contribute or creation of effective strategy for the preservation and future use. So, due to lot of limitations in the conventional breeding techniques, particularly the environmental influences will play a greater role in maintaining or conserving the germplasm, or for using any trait improvement. That could be the major limitation, and that can be overcome by this marker-assisted tree breeding. And also because since markers are used to detect the genetic variation of the genotype at DNA level, so the level of precision will be more. So the accurate trait transfer or trait improvement can be done with the help of markers, and particularly the marker technology allows the uncovering extent of genetic variation that cannot be done by the conventional breeding, by covering in the whole coverage of the genome. And also, if you see in the marker itself, a combination of diverse marker types, still it hastens the breeding activity, for recommended to provide an accurate assessment of extent the intra and the interpopulation genetic diversity, to understand population structure, the variation between population and among population and within population can also be done more precisely by this marker-assisted tree breeding. And the advantages of marker-assisted tree breeding over the conventional breeding, as we know that it is a very time-saving process, because at any developmental stages of the tree or plant can be, a neem tree can be taken into consideration to make our trait of interest can be studied with the help of marker. And stability and reliability, as I told earlier, and it have a higher stability and reliability than the conventional breeding because lack of environmental influences is there. And coming to the biosafety, particularly the disease resistance breeding, if you say that, mainly we will see that presence and absence. And in that way, if the conventional breeding, we have to inoculate it and we have to see the response. So in that way, it doesn't require those things. Only the pre-available information on that way, whether the particular fragment is present or not. In that way, it can be studied very easily. Of course, it is having high efficiency than the conventional breeding. And of course, as far as concerned in Neem, many traits are quantitative in nature and more complex in nature. And locating these QTLs, these QTLs are very difficult in conventional breeding due to the environmental factors. But however, the QTL will the marker could have tightly linked with the particular QTL can be treated as single Mendelian factor, and that way it can be very accurately and precisely locate that particular QTL, and that QTL can be successfully integrated in the target species or elite line with our trait of interest. Marker-assisted pyramiding, gene pyramiding in Neem, also it's highly helpful, particularly this resistance breeding in Neem, particularly disease resistance or insect resistance, pest resistance, or many genes, not a single gene, many genes will control this resistance. In that way, these QTLs can be effectively tagged and can be introgressed in the target genome to give multiple stress resistance in Neem. So that this way the markers are highly useful than the conventional breeding. These are some of the marker studies and early used in Neem, and many more studies are there with RAPD and AFLP, and some studies also with these ISSR markers they used. And of course, this is one of the important tool, genomic tool. That omics tool is an important next generation tool to Neem genetic improvement it is using, because we know that Azadirachta indica, the draft genome was published in 2012. The estimated genome size was 364 MB. And then Neem in Mediaceae, this is the first tree to be sequenced, and as a whole, 38 plant species was fully sequenced, and these were further updated the genome assembly in 2016, and then in very recently the hybrid assembly was carried out. Here the combining of both Illumina as well as Roach 454 reads, and then they used a combination of short reads and long reads to have more accurately, more precisely they sequenced this Neem. And then apart from that, organelle genome also sequenced here, like chloroplast and mitochondria genome. Of course, these omics tools, that particularly organelle genome sequencing, will help us to help in evolution of Neem species, and where the evolution, the co-species, in that way it highly helpful in Neem phylogenetic studies, and that will highly helpful in to understand about more and more about Neem system biology. Apart from that, the most important thing is in transcriptome is another way that another tool that particularly the terminoid biosynthesis genes and signal transduction with respect to the bioactive compounds, particularly azadirachtin and other bioactive compounds like nimbin and solanin, and many unigenes have been identified, and collectively these all together will help us to locate and to understand, to have an improved understanding about the system biology and genes and gene pathways involved in this triterpenoid biosynthesis. And that way it can be, and these genes can be sequenced more and more, and we can have markers which is tightly linked with these genes in order to have better evaluation for these bioactive compounds. And of course, Capri is working since from 90s almost in Neem, and I would like to have some of the research highlights at Kafri. This already I narrated that we have 170 accessions, and we have established various provenances and progeny trials and seed orchards. We have optimized vegetative propagation techniques in neem for especially stem cuttings, air layering as well. And also these findings we have successfully documented in the form of neem catalog, neem germ blossom catalog, where you can find all the information about the tree improvement activities which was carried out at Kafri. And this is our field gene bank at Kafri. You can see, and of course, currently we are running a project on neem genetic improvement, molecular, with the help of molecular markers, and these markers are developed from Neem transcriptome. We mined Neem transcriptome database, and we have identified some of the markers, particularly like 20 markers. We identified SSR markers. These are Neem transcriptome based SSR markers. With this help, we are evaluating these 170 gem blossom. Apart from that, the morphological traits also we are considering for evaluation of these Neem gem blossom. This project was funded by National Rainfed Area Authority, New Delhi. And this is the variation you can see for in our study. We have come across a lot of genetic variation for almost all the quantitative traits. It indicates that immense scope for selection of elite lines for our future neem genetic improvement. And you see that marker N010 profile for neem, then marker N001 profile. Of course, in the variation at DNA level also, we have high polymorphism among the germ blossom, whatever we are having at our center. And this has wide scope for selection, and it can be that marker data can be well correlated with the morphological, and then we can more and more precisely we can identify the high yielding germ blossoms for future trait improvement. And apart from that, we have incorporated a new technique called radio frequency identification tagging, that is RFID technology for monitoring, real-time monitoring of neem germ blossom. And here the RFID tags has been linked with the unique accession number of the germ blossom, if you see here. And this way, the data will be updated, the growth data or plant health data, germ blossom data updated in the SAMS portal from time to time. And that way, this will go and saved in the cloud server. So in that way, we are monitoring the neem germ blossom, real-time monitoring also we are doing currently. And some of the achievements I wanted to share that Kafri has identified some of the prominent germ blossom for high seed kernel and oil yield. You see that for seed oil, this VKF 99, 20, 122, 94, 115, 121 was identified as prominent seed yield germ blossoms. Likewise for seed oil content and azadirachtin also we have identified. Apart from that, very recently FRI also has released 6 varieties on neem for high seed oil in collaboration with IFCO, and FCRA also have some of the plus trees from Tamil Nadu and Karnataka as well for these economic traits. And coming to the future strategies, I would like to say that, of course, we have to focus that multiple stems. We have to production of multiple stems that needs to be focused, and that will give more scope for to improve the biomass as well as fruit. Both the things it will be useful. So this should be the selection criteria we have to keep. And for early establishment of plantation, especially in dry areas where the water scarcity or more problem, there we have to focus on the survival rate and fast growth may be the better choice for the climate resilient neem production can be made through like way. And then for long-term utilization as timber provenances that have been straight stem and more promising in intraspecies hybrids could be among the selection criteria. Like for agroforestry perspective, we have to focus that deep-rooted habits and then resistant to high wind run, then the narrow canopy. Also, these are the focused areas needs to be targeted. Of course, for increasing yield, net photosynthesis is an important physiological trait, needs to be focused, and leaf and stomatal traits, particularly stomatal density, are highly correlated with neem yield. So focusing of these traits will indirectly improve the neem yield. And apart from that, the use of new tools such as genomic tools are very resources are imperative tool for trait improvement program. Of course, it has to be focused well, and the tree genomics has the near future for understanding the more and more improved understanding on system biology and developing policy actions to conserve nature and managed tree genetic resources. In that way, tree genomics must be focused, and of course, genotyping through high throughput technologies like GWAS and genotype by sequencing, GBS technology and SNP must be incorporated in this study. And of course, no doubt, these tools will definitely hasten neem genetic improvement programs. So with this, I conclude myself. Thank you.
Thank you, sir, for your informative lecture on the neem genetic resources, its significance, assemblage and clonance and progeny trials at Kafri, long-term breeding strategy for neem, and you have shared about national and international network on the genetic studies of neem and application of biotechnology and tissue culture in improvement. Thank you so much for your such a nice presentation. With this, I would like to request our respected chairman and panelists to give their remark. First, Dr. S. Biswas, sir.
Yeah.
Dr. S. Biswas, sir.
Are you able to hear me?
Yes, sir.
Yes, sir. Yes, sir.
Okay. So, first of all, I would like to acknowledge the authorities of this university, Professor Arvind Kumar, Vice Chancellor, then Professor Dr. A.K. Pandey, Dean, College of Horticulture and Forestry, then Professor Manmohan Dobriyal, HOD, Silviculture and Agroforestry, co-organizing secretaries, Dr. Ram Prakash Yadav, Dr. Gaurav Sharma, and panelists whom I can't see, Dr. Ashok Kumar and Dr. K.P. Mahapatra. They will be giving their comments in the chat, I believe. Now, before I come to the points that I feel to take forward, which has already, this national seminar, I believe, has been already taken to the international stage. So, so much of the thing, I think it should have been named as the international seminar and all. It is super, wonderful. I enjoyed it. I remember that was the year 1995 when our late Director General, emphasized on neem. And he asked the scientists, I was one of them, to make the monograph on neem. So I was going through several works and all, and there I found in one that, astonished me. That was in Khadi Gramodyog, Maharashtra, Pune based. They had developed from the oil, it was tried in even for aeroplane fuel. So that was most astonishing this. And I'll have some comments first, general comments, and then I will come to the two presenters. First thing which strikes me immediately, and it was addressed. In fact, I was in 1.5 of the sessions, including the previous sessions. It was very enjoyable, but I was busy with the International Urban Forestry, seminar organized from Hong Kong. So I couldn't get the time. In fact, after this also, I may have to join them. Anyway, so what I find, climate change is one, which other experts also, or dignitaries also talked. So, there's a changing locality. Locality factors are changing. And even there are, change in the microhabitat of some types of forest. I found in the list where I found, I tried to find if I could see Uttarakhand, about the neem trees in Uttarakhand. But there are. And, as you enter the, Uttarakhand, where, Shivaliks, via Saharanpur, in the forest, they are started coming up towards the north. And there are good regeneration, but it needs assisted regeneration. Then this was the range extension. And now about the phenological period, which a man mentioned, I know there are some population in Murshidabad in West Bengal where the neem flowers almost throughout the year. So that also becomes one of the most important features for studying out how we can have this. And then about the economic aspects, I was very curiously listening about the honey. And I must tell you, I'm now here in Bengaluru. I couldn't go back to my place in Dehradun because of the COVID third wave. And I do remember a year ago, I had gone in one seminar where the honey is specifically made out of the neem nectar that was naturally collected, and it was being sold by some SHG, and it was very fascinating. Although bees are there, I find the bees are being mentioned. And now when we do talk about its pollination biology, anemophily comes, but at the same time, entomophily is also there. Insects also. Honeybees are, extracting the nectar and providing that. And in fact, that honey costs much more than the honey, that is collected from the other plants like Terminalia, Balearica, or Chabula, because of it has got the more of medicinal property. So this becomes one of the important features that I do find. Yet another one, I was reading only this morning where I found that those 100 smart cities which have been mentioned, Neem must have the place. I sent to Professor Manmohan Dobriyal one, YouTube channel that I made. So between Bangalore to Mysuru, there is about 3 kilometer long stretch of separator made by introducing the neem. And interesting thing, all the neem trees, if you can see, I would like to share with delegates, all are of the same size. And it makes a perfect separator. And this becomes one of the most important from that point of view. And of course, in South India, we do have growing it naturally. About 800,000 from Karnataka have been mentioned. And the Karnataka, what I believe that, in one presentation it was made, that when there is a, high rainfall area, the growth is more, but the yield is less, but vice versa. Then when it comes to, although there is a next session on agroforestry, but I feel this can find a place in home state. That's very much important. And the film that I saw is the most enlightening. And then what I feel, we can have a kind of, species selection for the agroforestry model. For example, what I find, tamarind, neem, and Centralum album are growing naturally in this part of the country, in Karnataka. And another feature that we do find, when we go for the cross breeding and all, why not to go with intrageneric cross breeding? For example, in media or Milladubia or any other species which has got the similar property in its chemical constitution. And when I come to our presenters, yet another very interesting thing, what is most important, I feel Department of Forestry of Rani Lakshmi Bai University and with others should go for as how we can go for the patenting. Although we know there were 14 patents already made on the neem, and now it is said that 54 are more, and there must have been done. That's what I learned once. And while US has got 54, although we have one like that of in case of turmeric, but we do require to go with more, because what I found even the last presentation where it was mentioned about the RFID technology, gene typing, Omicron tools and all, they all reach the process towards the patenting of the process. Now, it was also mentioned here that it has got the antiviral property. What I found, it has been tried against even the COVID-19 and extract from the bark. And that was done Indian Institute of Science, Education and Research, Kolkata. And they have tried this, and it is being found doing good. Now, I come to the most enlightening, presentation by Dr. Vanilla. It was wonderful, mentioning about the percentage of the individual population in different states of like from, Tamil Nadu, Uttar Pradesh, and then in Karnataka. But Uttarakhand is a gap which is fighting. Dr. Manmohan comes from Uttarakhand. He should take care of that, and we will go for the collaboration also in that aspect for the thorough, what I will call it, ground truthing about it. And then the pollination biology, about the incompatibility. So now when we do talk about, you mentioned in situ, ex situ. So in situ, which your presentation, in fact, enlightened me why we cannot go for developing a kind of the corridor where the pollinators can reach from one place to another place. So this could also enhance the population of bees, and also this can also connect to the international network on pollinators. And South India, or you can say south of Uttar Pradesh, all those areas are very much important from that point of view. So I find this is the point which which can be done from cluster to corridors. And of course, the kinetin concentration, which were mentioned and resulting into the formation of callus, this is also, I find in our presentation very nice. And besides, she has given a very viable suggestion. That was the canopy management. I don't think we have got, in case of the management rule of the canopy, where we could find this neem. If you happen to go to Banaras Hindu University, there is a line of neem. You go to Parliament, not Patiala House, towards Patiala House in Delhi. There also a line of that was all done by our honorable President of India, Sarvapalli Radhakrishnan, long back. And now you just see, because of the environmental regime, they have got the canopy which is naturally controlled. Even the vehicular traffic, they are coming. So what is most important? What I feel like, I'm in contact with the Christ University here. So we are going to work how if we introduce this neem tree avenue, how much of the exhaust from the vehicular traffic and all could matter on this. So this is about it. Now I come to yet another presentation by Dr. Raja Rajan. It was wonderful. I enjoyed it very much. And And I must tell you, I learned a lot that I would percolate to my faculty and the students of forestry in Dehradun. What do you want? Genetic variability, provenances, gene combination, and then cryogenic storage. And here I would like that we should also think of how to introduce, be it a road from Meclopodyam to Nilgiri Hills or towards Palni Hills. Why not to think of uncovering other green spaces? If we see the parks and all, and many places, the percentage of neem is very less. Maybe somewhere it's only 20%. If they have survived by their own. But all the plantations and all those are there. But in those parks and the green spaces, we don't find. And then important point that is why not to have the glow as the medicinal plant along with the tree. And of course, he talked about the climate change, and I understand that the range extension now will matter. And Kafri has got about 300 accessions. What do we require? Can we have the accession-wise introduction in field in different places, be it North India or South India and all? If it is done, well and good. And they can be put into the clusters and then as individual to see the impact of the changing climate. And then, of course, omics tools, transcription, that was wonderful. And I loved it. Biotech marker-assisted tree breeding, molecular markers, and RFID technology. And that is very much enlightening. I found both the presentation very much your learning experience. Thank you very much.
Thank you, sir. Thank you. Dr. Pawan. So, thank you, sir. I thank both the panelists and Vishwas sir for his chairman remarks and sparing time, his valuable time for this. I also thank both the speakers, Dr. Rajarajan and Dr. Vanilla, for giving a wonderful presentation on the tree improvement aspect of the neem. So thank you, sir. And we proceed for the next session. Thank you. Dr. Rakesh.
Okay, thank you, sir. Good afternoon and very good afternoon to one and all. And I am Dr. Rakesh Kumar, Assistant Professor, Department of Environment Science, RLBCAU Jhansi. And I am thankful to our respective Dean, College of Horticulture and Forestry, Dr. A.K. Pandey sir, and Organizing Secretary, Dr. M.J. Dobriyal sir, for giving me an opportunity to introduce the eminent chairperson and panelists of this session. I feel honored to have this opportunity, sir. And the chairman of this session is Dr. Anthony Joseph Raj, Dean College of Forestry, Sam Higginbottom, University of Agriculture, Technology and Science, Prayagraj, Uttar Pradesh. Dr. Joseph Raj is an eminent personality in the field of forestry. He has covered 90 publications in his credit, including 63 research papers, 2 textbooks, 19 book chapters, and 6 instruction and training materials. He has also implemented 8 research projects funded by external funded agency. And he has also awarded by various organizations for his excellent work in his field. And he has received many awards and distinctions such as Eminent Scientist Award, Young Scientist Award, Best Paper Presentation Award, Award for Outstanding Contribution to Climate Science and Environmental Sustainability. And he has visited Germany, New Zealand, Malaysia, Iran, and Ethiopia for training participation in international congress and presenting research paper and job. And thank you, sir, for your gracious presence And the panelists of this session are Dr. Arvind Bijalwan, Director Academic, Veer Chandrasekhar Singh, Garhwali, Uttarakhand University of Horticulture and Forestry, Rani Chori, and Dr. Salil K. Tiwari, Professor and Head, Department of Genetic and Plant Breeding, and from Govind Ballabh Pant, University of Agriculture and Technology, Pantnagar. And Dr. Arvind Bijalwan has 19 years of teaching, research and extension and consultancy experience in the forest management, agroforestry, silviculture, remote sensing and GIS and nature resource management. He has handled half a dozen of research and extension projects. He is live member of 6 scientific and professional society and also an annual and biannual member of 9 professional society. He has delivered 40 radio talks and various TV programs also. He has guided 3 doctoral and 6 MPhil and master level student and has written 3 books. And another panelist of our session is Dr. Salil K. Tiwari sir, and he has published many research papers, articles and book chapters in national and international journal. He has a rich experience in the field of teaching, research and extension. He has worked in various fields in forestry, and his key area of research are tree genetics, agroforestry, climate change, carbon sequestration and bamboo cover. Dr. Tiwari sir has been awarded Brick Fellowship holder, Harbhajan Singh Memorial Award 2003, Dr. KG Tejwani Award 2016, Faculty Excellent Award, Agriculture College in 2015, and member and board of management in FRI and IGKV, and he is also vice president of ISAF. And he also visit many foreign countries such as USA, Canada, Maldives, Nepal for academic purpose. So I welcome both of you, sir, to this webinar, and the topic is silviculture and agroforestry of Neem. So now I request my colleague, Dr. Pawat Tiwari, to invite and introduce our speaker.
Thank you, Dr. Rakesh. First of all, very good evening to respected chairman, sir, respected panelists, respected speakers, our respected Dean, Dr. A.K. Pandey, sir, head of the department, Dr. Dobriyal, sir, Dr. Ram Prakash, sir, my dear friends, and all the participants. First of all, I would like to welcome our speaker, today's speaker, Dr. U.K. Tomar, sir, in this webinar, sir. Sir has retired his service as a scientist F from Arid Forest Research Institute, Jodhpur. Dr. Tomar, born on 10th of June 1960, and is basically from Noida, Uttar Pradesh. Sir has completed its PhD from University of Delhi in the year of 1989 with the specialization of botany and the plant tissue culture. And sir has completed 2 certificate course from Institute of Forest Genetics, Grasendorf, Germany, and University of Melbourne, Australia, in the discipline of forest genetics and genetic transformation and DNA markers. Sir has been honored several awards and medals. Some of our award best-of medals by Parker Intel College, Moradabad, and prestigious Brandeis Prize for the year of 2019, and many more. Sir has visited several countries for research and administration purposes. Sir has started his career as assistant professor in botany department, Hindu College, Delhi University, and sir has guided several PG and PhD students under different courses organized by Arid Forest Research Institute, Forest Research Institute, Dehradun. Sir has handled several other assignments, administrative experience like course director, like research coordinator and advisor, like head genetics and tissue improvement division, and many more administrative assignments. Sir has handled several research projects basically from related to the forest trees. Sir has published more than 50 research articles in various reputed national and international journals. Sir has published several books many chapters and several proceeding popular articles and many more publication things. And sir has guided 9 PhD students for research purposes. Sir has vast experience on Neem, and we are quite lucky because we are going to hear sir thought about the Neem. So without wasting any time, I have directly invited Dr. UK Tomar sir to present his presentation on today's topic. Please, sir.
Hello, I'm audible?
Yes, sir.
Okay. First of all, I would like to thank Dr. A.K. Pandey, Dean, RLBCAU, CAU University, and Dr. Manmohan Dobrial, Organizing Secretary of this conference, and the respected chair and panelists. I will start my talk on Neem. I'm retired just 1 year before in 2020 from Merit Forest Institute. And here, my presentation is visible.
Yes, sir. It's not in slide board, but it's visible.
Now it is in slide board. Okay. When I joined this Arid Forest Institute, Jodhpur, Rajasthan, in 1992, the Neem work was started by this institute. And they have established a germplasm, provenance trial from all over the country. And that time, most of our institute people were working on Neem. So I joined this. I was basically a biotechnologist, did PhD in tissue culture. So I did some work on clonal propagation also. Later on shifted to the their germplasm evaluation using DNA marker techniques and all tree breeding programs. So before going into the work which I have done in last 2 decades, I will tell you the background. Indian Council and Forestry Research and Education, they have they have started the work on Neem, and actually the AFRI was made as a nodal agency for other ICFR institutes on Neem. The first international consultation meeting on tree improvement was held in Bangkok in 1993, and subsequently second was in AFRI, Jodhpur. And that time they have established the International Neem Network. Almost 23 countries have joined this International Neem Network. And the International Neem Trial was established at 3 locations in India. One is Jodhpur, Jabalpur, and Coimbatore. 3 different institutes of ICFR. And in 2001, these international Neem network, these all trials were evaluated, and this meeting was held in March 2001. This report is available on FAO website. The link is, you can see at the bottom. And I will not go into the detail of this report. Now, I will tell you about the national, first start with the national provenance trial, which was established. There were 40 different provenances. Seeds were collected and established this trial. Then this trial was first evaluated for overall assessing the variability by using DNA marker in collaboration with Tata Energy Research Institute, New Delhi. And we found the technique we have used AFLP, and we found these 7 primers combinations give very high polymorphism, and you can say that this Neem shows very high genetic variation. And when use component principal component analysis 2 times, we found that the Indian Neem and the other Thailand Neem, they are far apart from each other. And in fact, later on, this Thailand Neem was given the separate species, Azadirachta siamensis. Then subsequently, we got few more projects. One project was funded by Novo, and in this project, almost 10 different universities were involved. This was for 3 years. And under this project, AFRI was given the area of Gujarat, and whole country was divided into different zones, and the different area was given to different institute or universities for this project work. Subsequently, other projects was also supported by some was ICFRE and IFCO. Then recently, a big project I was prepared as a national project coordinator before my retirement. This was again with the different institutes of ICFRE. This is running right now. Now I will summarize the report progress of these projects stepwise. First, this Novo board project, where we have collected the germ plasm from all over India, and these were the 8 institutes involved in this project: NBRI Lucknow, FRI Dehradun, Kazri Jodhpur, TERI Delhi, NBPGR New Delhi, HF Haryana, this is in Hisar, Neem Foundation, IIT Delhi, Novo, and AFRI Jodhpur. Under this project, different samples were collected from all over the country, from different states. And these seed samples were evaluated for agent oil contents. These are the showing the number of samples collected in different states. Now, when we studied the variation to 16,000 ppm, and average variation of the country was 3,043 ppm, and out of these 50,000 samples, 27 samples were recorded which were having more than 10,000 ppm azadirachtin content. So this shows that there is a tremendous scope of selection, particularly for azectin content for this tree in India. If we group these samples into the state-wise, we found that maximum azectin were from Andhra Pradesh, Karnataka, and Tamil Nadu, which were above average, and rest of the places they fall below the average. And if we group them, that Tamil Nadu, Karnataka, and they group together, and rest of the states falls in separate group. Then if we group them on the basis of geographical features, we find Deccan Plateau has the highest azectin, followed by Central Highland, and minimum was in the desert area. And if we group them into the climatic conditions in four semi-hot, arid, mild winter, hot, subhumid, hot, humid, hot, semi-subarid conditions, then we find that hot, semi-arid, and mild winter is the ideal situation for high azectin content. And these differences were highly significant. Now, we have studied the samples collected by AFRI separately. And these samples were collected from Gujarat, and Gujarat is having 6 agroclimatic zones, but Neem is present mainly in 4 agroclimatic zones: agroclimatic zone 2, 4, 5A, and B. Seed samples were collected continuously in 3 years, 2000, 2001, and 2002. And we found that initially the average azectin content increased every year. And similarly, we also observed the rainfall also increased every year. So last 2 years, there was a direct correlation. Previous year's rainfall has influenced the azectin content. Then the maximum azectin was observed from Gujarat from AER 5B region. Another finding of from these research was that there was no significant difference because of girth classes. When we group these data into different girth classes and analyze for effect of girth class on azectin content, and we found that there was no significant difference of influence of age on the azectin production. Then we also studied the oil content and different oil components: palmitic acid, stearic, oleic acid, linoleic acid. Then girth class has no effect neither on oil, aja, azectin is having correlation with the positive correlation with oil, and similarly palmic acid having correlation with negative correlation with the aja, and whereas stearic acid positive correlation, and other two, oleic acid having no correlation with azectin. Another factor apart from the girth class and rainfall, the temperature. We also studied the effect of temperature on aja concentration. We found that the azectin was higher when the temperature of fruit ripening period remains between 28 to 34 degrees centigrade. That is the best for producing higher agility in the seed fruits. Then we have established a progeny trial of 17 trees having more than 5,000 ppm agility and 40% oil. This is established in Jaipur, Govindpura, with the with the help of forest department. And from this progeny trials, we found that progeny of CPT number 7 was the best for most of the phenological and survival high area in Jaipur. So there was, we found that there was a casualty of frost and some this
some problem, I think.
Yes.
राकेश फोन है, फोन जरा करो है।
जी सर, जी सर, जी सर।
Why don't we move to the next presenter? Professor
Rakesh.
Dr. Manmohan.
Yes, yes. Dr. Rakesh. Dr. Rakesh.
Yes, sir. Yes, sir.
We can start with other. Meanwhile, later on, you can join.
Okay, sir. Thank you, sir. Now I request my colleague, Dr. Pankaj Lavania, sir, to introduce our next speaker.
Thank you, Rakesh, sir. I welcome Chairman, sir, panelists, speakers, and I am also thankful Professor A.K. Pandey, sir, Professor M.J. Dobriyal, sir, and Dr. Ram Prakash, sir, for providing this opportunity. I welcome and introduce Dr. Sasi Bhushan Saran Pandey, and he is now presently associate professor and head silviculture and agroforestry department, College of Forestry, College of Horticulture and Forestry, and he is a committed forest educationist in Rajasthan, and he has started careers as a graduate student of BSc Forestry at Adnagar in the year of 1997, and postgraduate from the Forest Research Institute, Dehradun, in the year of 1999, and he has qualified ASRB net in the forestry subject in the 3 times, and he has also awarded with the gold medalist for the PhD forestry by NAU Navsari in the year of 2018, and he has served as a pioneer regular faculty as an assistant professor in the Doon College of Agriculture and Technology, and he has also as an assistant professor in a Dolphin Institute of Biomedical and Natural Sciences. Sir has published more than 20 research papers and 17 practical manuals and 15 articles, and after that conduct more than 6 trainings programs, and sir has delivered so many lectures on agriculture, on silviculture, nursery and agroforestry time to time, and sir has got best teachers award 2017 in the field of agroforestry, and Banvardak Purushkar he has got from the Rajasthan government. So, sir, I welcome to you. Please deliver your presentations.
Thank you. Good evening to all of you. I would like to thank Rani Lakshmibai Central Agriculture University authorities, Honorable Vice Chancellor Sir, and Dean Dr. Pandey Sir, Organizing Secretary Dr. Manmohan Sir, and my sessions colleague Dr. Tomar, and our Chairman Dr. Joseph Sir, and panelist Dr. Arvind Bijilwan and Dr. Salil Tiwari Sir. Good evening to all of you. I will be presenting a very simple presentation, as topic was silviculture and agroforestry of Neem. So the topic is silviculture of agroforestry and agroforestry of Neem. Neem is actually slide share करें पहले. I am just sliding share from the mobile because office hours has been. Yes, sir. Is it clear?
Yes,
Slides here? Yes, Dr. Manmohan.
Yes, sir.
Okay.
Visible.
Yes, yes. So this is my lecture. Neem, as the name is Azadirachta indica, from basic point of view, it belongs to family Meliaceae and is nearly evergreen tree, and the species is generally found on ravine tracts, boundary plantations in the state of Gujarat, Rajasthan, Punjab, Haryana, Uttar Pradesh, of course in Uttarakhand, in the areas where the frost is not found, and in Tamil Nadu, Odisha, Bihar, and generally having a growth of approximately 30-meter height. And it is called a gift of God, as it has had many medicinal values. So from the silviculture point of view, if we go through the neem plantations or neem largely grow along the avenues, village homesteads, and farmlands. Neem plantations are not organized in the country as per the medicinal and value of the species. Presently, the trials are being conducted by so many agencies, and Neem Foundation has also been formed. And we are also viewing in the webinar that since last 20-30 years, the efforts are very good for this species. So this is approximately evergreen species which spreads its branches, rounded crown, up to 10 meter in diameter. Short, usually straight trunk, has a moderate thick, strong bark, and these are the general things about the tree. Having good foliage, the fruit is ellipsoid, smooth, and 1 to 2 centimeter long. Yes, that is yellow when matured and comprises a sweet pulp enclosing a seed. Seed is composed of a shell and a kernel, and kernel is also having oil content, and the seed bears, tree bears start fruiting from 3rd year onwards, depends on the locality. And at the age of 10 years, it is very good prolific seeder, and up to 20 years of age or 25 years of age, it can produce 50 kg of seeds. Goget and Gurjar found flowering and fruiting in a 50-year-old plantation of neem in Maharashtra to be highly variable. Out of 331 trees under study, total 291 flowered in single year and fruited in that season, and the fruit production varied from 7.5 to 17.3 kg on dry weight basis per tree. So we have already seen so many things about the tree improvement and reproductive biology, so it is not necessary. But the main important thing is that the flowering time is very crucial, as we are having very less time for the collection of seed in northern India, and in southern India it is somewhat later. The distribution as per silvicultural characteristics, we have to know the distribution of the species, soil types, and silvicultural characters, all these things. So neem is originated in Assam and Myanmar, where it is common throughout the central dry zone and the Shivalik hills. However, the exact origin is uncertain, and some authors suggest it is native to the dry forest of South and Southeast Asia, including Pakistan, Sri Lanka, Thailand, Malaysia, and Indonesia. In the 19th century, the neem was introduced by Indian immigrants to the Caribbean region, to different countries in Africa also. The next is habitat. The neem grows on all types of soils, including clay, saline, alkali soils, but it does very well on black cotton soils. It can tolerate dry, stony, shallow, with a waterless subsoil, or places where there is a hard calcareous or clay pan near the surface. The species is also thought to be an efficient water user in comparison to some other tree species, and requires very little amount of water, can tolerate drought also. that the Neem does not tolerate waterlogging. Perhaps if there is continuous waterlogging for a duration of 10 or more days, then the older trees are also affected badly. It is fire resistant and is a unique property of calcium mining, which neutralizes the acidic soils. So in these types of soils, it can be grown very well. The species is a highly light demander, and when juvenile, Neem will push up vigorously throughout the scrubby vegetation. It is hardy but frost tender and does not withstand excessive cold, especially in the seedling and sapling stage. That's why the Neem is almost absent in the areas of Uttarakhand or hilly regions where the frost occurrences are high. The copicing, it copices well as pollards well, produces root suckers also. The next is propagation. The Neem is normally propagated by natural means, that is by seeds, but we have seen so many presentations in the previous sessions that tissue culture is also being promoted nowadays. These are the silvicultural practices while we want to see when the Neem is being propagated sexually by seeds only. So we have to arrange beds, either mother beds or polybags, and we can keep seeds, dibbling of seeds, 2 to 3 seeds in a polybag and irrigate immediately. Then the fourth one is the picture of germination of the seedlings. Next is the interest in Neem as a source of botanical pesticide has prompted discussion on the need for plantation of the species. There are thought to be more than 17 million Neem trees in India, which occur along roadside in the field boundaries. So these are the data for as per silviculture point of view, in which countries the Neem is found. In our country, Kajri, Afri, of course, Rani Lakshmi, Central Agriculture University, and Neem Foundation, and different state agricultural universities where the Neem is found are performing very well for the silvicultural operations and regeneration trials for the nursery, different medias, in which media which is better for the growth of seedlings. So we can see these are the seedlings, very healthy seedlings of a trial done by my supervision for a PG student. Now in Jodhpur, the Afri Arid Forest Institute have been researching different methods of water harvesting and moisture conservation on the growth, biomass accumulation and nutrient uptake by Neem. The regent furrow method was found very good for the water harvesting. 58, 73, 58%, 73, 11% improvement in the height, collar circumference, and crown diameter, and different bioaccumulations increased by 3.8%. So these are the data giving at different, what is the spacing kept, 4 by 4 meter spacing adopted somewhere, and Hegel 93 recommended 6 by 6 meter spacing and 8 by 8 meter spacing. But as far as spacing is concerned, in agroforestry, if these are the view of roadside plantations of a sound. Hello. Sound here. Hello.
Your sound is coming.
Second sound, another sound is coming.
Dr. Vinod. Dr. Vinod, mute yourself.
हाँ म्यूट हो जाए ना, कुछ डिस्टरबेंस लग रहा है.
Others, please mute yourself. Dr. Vinod. चलिए सर.
Yes, this is a very good view of roadside plantations of a moderate sized neem trees, and these are the flowering neem trees, and these are the fruits of the neems, looking like very fresh and like jamun. So as per silvicultural operations, lopping is rarely required. Lopping for fodder is required, but not for growth and other operations. So trees are also lopped for fodder purpose and for the purpose of medicinal use of leaves. The growth and productivity of neem could also be increased through the application of biofertilizers, azospirillin, phosphobacteria, VAM, etc. The neem seedlings grow very good in combination of these things. So if we use wasteland or some marginal lands, then we use biofertilizers with the seedling material, that is mixture in the neem seedlings, then the growth is found very well. These are the close pictures of 3 to 4 months seedlings root growth, and another picture is of seedling with the root growth. Hello. Yes, now this is the part of agroforestry. As we know that agroforestry denotes a sustainable land and crop management system that strives to increase yields on a continuing basis by combining the production of woody forestry crops, including fruit crops, tree crops, with arable and field crops and herbivore animals, simultaneously or sequentially on the same unit of land, and applying management practices that are compatible with the cultural practices of the local population, given by ICRAF, ICRAF 1982, very well known definition. But the productivity in agroforestry system is higher as compared to sole cropping system, because higher yield of crop has been observed in forest influenced soil than in ordinary soil in so many crops, not in all crops. If the crop is highly light demander, that is we can go for some pulses or wheat like this, then the productivity is decreased due to shade of the trees. But in so many crops, the combined productivity is found higher. These are the glimpses of the neem seedlings grown in green shade net. Now the topic is agroforestry. Now here it is the international combination of agriculture and working trees to create sustainable farming systems. Nowadays we can have different practices of the agroforestry like windbreaks, silvopastoral, forest farmings, and alley cropping. So these are some data given by Jhansi et al. Earlier, Jhansi sir used to be posted in Central Agroforestry Research Institute, Jhansi. That time the paper was published. A total 25.32 million hectare, 8.2% of the total geographical area of the country is under agroforestry presently, and the wasteland and marginal lands are considered for promotion of agroforestry models, then we can go up to 53.32 million hectare, that is 17.57% of the total geographical area could be potential under agroforestry in the next 20 years. Average sequestration potential in agroforestry in India is approximately 25 ton carbon per hectare. So by agroforestry, this is very simple thing. We want produces or sellable products. That is, neem is a tree which is shown in this picture. You can replace the leaves, and the leaves are like these rupees. In case of neem, provide value added opportunities. So many products are obtained from this particular tree. neem, and diversify risk, that is in the era of climate change. If the crop gets failure, then you can have the products or income from the tree crops. Increases property value. The land remains very fertile and rich in carbon, organic matter, and increases crop yields and livestock production, because livestock can be managed only by having different kinds of trees, shrubs, and fodder grasses grown on the farmer's field. Then it is very easy to have livestock. May be eligible for the cost share and land rental payments. So agroforestry is very important for these things. This is a very good illustration, the effect of agroforestry, how we can understand the negative or positive effects of the agroforestry. It has been shown as A, B, C, D. A is shading, B is root competition for the water and nutrient, C is litter fall and pruning, and D is deep rooted trees. The main important thing in this picture is the negative may be shading or root competition at the earlier stage, but the main important thing is D, that is deep rooted trees play a role as a safety net for leached nutrients in the deeper layer or as nutrient pump for the fertile soil. This is very important. No instrument has not been designed till date that the depth which a tree root attains in the soil can take back the nutrients from that particular depth in a very shorter period without having any financial liability. So these are different, again, different pictorial illustration of agroforestry for the improvement of soil fertility, that is litter decomposition and addition of organic matter and nutrients. Even the root systems release nutrients and improve the soil structure when they decompose. Some trees capture nutrients lying deep in the soil, too deep for the crops to reach, bring them to the surface and later return them to the soil as litter, which the crop utilizes when it decomposes. If the leaves and branches are left on the ground to decompose and their nutrients are lost, the tree will have to be nourished with the equivalent nutrients added as fertilizer or organic manure. So now there are some examples, or we can say conditions, how the neem can be understood in agroforestry. The neem has considerable potential within agroforestry systems as recognized by Tilander 1996. However, the species is thought to be underutilized in many areas. The good copicing ability of the tree as well as low mortality following copicing make neem favorable for the use of in agroforestry systems. Chaturvedi 93 and Gill and Roy says that an excellent account of agroforestry potential of neem, which include its use in intercropping and silvipastoral systems. Some authors also recommended in their trials that leaf mulches and compost of neem leaves have been resulted in some crops very well. That is, sorghum is one of that. And in Lucina, in comparison to Lucina, Lygia, and Acacia, the leaf mulch or compost made by neem leaves is better. The use of mulches to conserve soil moisture and increase nutrient status of the soil. In this thing, the neem mulches found better. Institute of Forest Genetics and Tree Breeding, Coimbatore, concluded that neem could be economically cultivated as an agroforestry species. Neem was compared to Albizia, Glycida, and Eucalyptus. 3-month-old seedlings of all species were planted at a spacing of 5 meters within rows and 12 meters between the rows to give an average of 160 trees per hectare. Agricultural crops such as sorghum, black gram, groundnut were grown in the field as per traditional practices. So trials under agroforestry with the different crops is going on for different tree species. In case of neem, only few leguminous and some fodder species have resulted so far. Good. Next is Vivekanandan described a successful agroforestry system from a village in southern Tamil Nadu, whereby crops such as sorghum, cotton, millet, black gram, green gram, coriander, and sunflower are sown between the neem trees that feature prominently in all the farmers' field. Trees are randomly sown or naturally regenerate. Hence, spacing is irregular. Farmers generally aim for about 86 trees per hectare. Here, the emphasis was that if the spacing between neem trees is reduced, then it affects the crop yield. So 86 tree means the trees are having very good distance, that is 12 to 15 meters. Then it is not affecting the agricultural crop production. Neem is commonly growing on the bunds of the agricultural fields traditionally, thus considered to be one of the important multifunctional agroforestry tree species. The basic agroforestry is a land use system that integrates trees, crops, and animals in an approach that is scientifically sound, ecologically desirable, practically feasible, and socially suitable to the farmers. The tree component is preferred in agroforestry system according to the local requirement with the multiple products and easily marketable with good economic value. Adoption of agroforestry on all kinds of wastelands in the country and incorporation of multi-purpose trees is already in practice in all cropping farming systems. It is an ideal option to reclaim and to enhance the productivity of wasteland by planting the multi-purpose trees like neem, which re-establishes the soil fertility and richness. Because neem tree is also very fire resistant, and in very low fertile soil, it can be established easily, and also increases the production of other herbs and shrubs. The good coppicing ability of neem as well as low mortality makes this tree favorable for use in agroforestry systems. Neem is best ideotype tree species for agroforestry system as it possesses both multi-purpose and multifunctional characteristics. It maintains the temperature, humidity of soil and works for soil moisture. Now, another type of system is silvipastoral system in agroforestry. The neem can have beneficial effect on the soil, nutrient, moisture. All the status of the soil is improved and provide shade, fodder in the dry season. Neem generally found in very hot localities and drier localities. Also, animal medicines. Gill and Roy reported that neem, along with other tree species, improved the fodder productivity of semi-arid silvipastoral system in India by up to 8.5%, 8.5 tons per hectare. Hirsch et al. Also studied silvipastoral systems in arid regions. They reported that forage production can be increased 3.6 tons per hectare from only 0.5 tons per hectare by growing suitable grasses, legumes, and trees, including neem. These are some glimpses of different systems. And on degraded lands, neems also perform very well. Chaturvedi gives an extensive overview of the use of neem in plantations and afforestation in India, the ability of the tree to tolerate drought and saline conditions, and likely the fact that the leaves may be used as fodder from 5 years onwards. These are the simple things which we can understand in case of neem. What are the advantages of neem in agroforestry and disadvantages of neem in agroforestry? That is advantages. First, we see the advantages that coppices well with low mortality, provides a variety of products including mulch, fodder, shade, medicine, pesticide, fertilizer, firewood, and timber. It is used for mulching, fodder, timber, and firewood, suitable for the afforestation of degraded land, and provides a source of income generation by selling different parts of the tree and value addition. Negative things are disadvantages may become invasive on fellow agricultural lands. In some areas, it is in productive agricultural fields, the farmers don't want to grow neems as the crown expansion is very high and it covers very high amount of soil under shade. So in highly productive soils, they don't want to grow neems in, especially I found in northern areas. It may reduce the yield of companion crops through the increased competition for light and water. Sometimes competition for light and water is greater in arid areas. Competition for light and water is greater if the neem is densely planted. So we should plant at a very far spacing. Time and labor requirement for tree and crop management is increased in case of agroforestry management. These are the products obtained from the neem, that is food. In food, fodder, fuel, timber, gum or resin, every type or every kind of product is obtained from the neem. These are some pictures. Very good multinational companies are, I think all the Ayurvedic preparations are having neem based products. All the homeopathics and all these companies are using neem in a very high quantity. This is the tannin and diester lipids, poisons, all these things are obtained and made by the neem, by either by leaf or by seed or by kernel or by bark or by different parts. So this is the medicinal use of neem, how the leaves are used in which disease, in leprosy, eye problems and skin ulcers, all these things. Bark is used for different diseases. Flowers are used for different diseases. Yeah, these are directly used by Ayurvedic or raw preparation, and by processing we can have different medicinal tablets, syrups, all these things. Seed pulp is for leprosy and intestinal worms. Oil is also very important, and that is also for used for insecticides, formulations for the agricultural crops also. And neem cake is also very beneficial. All these products are highly useful in the present day need. These are some examples of a finding by Gujarat, Sardar Prashinagar district. The soil fertility status after harvest of different field crops with and without association of neem. The organic matter content was found higher in neem with clisterium and in green gram in comparison to their sole cropping. As well as nitrogen content was also found higher. Nitrogen, phosphorus and potassium, all these things have been depicted in this table. The next is the per hectare average yield of neem in the study area. A study has been conducted by Balaji 2017 that has been published in the report as report. So we see average age of a neem tree, 3 to 5 years, 6 to 10 years, 11 to 15 years, 16 to 20 years, and 21 to 25 years. Different age, at different age, the average fruit yield is increasing. So we cannot have any loss if we grow the neem trees, as the seed is main source which is used for so many things. The income is also increasing with the age of the tree. The next is, this is I think effect of neem trees on the yield attributes of gram. This is research conducted by Pandey et al. I think the Dean sir from Rani Lakshmibai sir, unka hi hai sir ye shayad. Distance from the tree base. found 50 centimeter, 100 centimeter, 150 centimeter, and 200 centimeter. And the experiment was conducted in 1998 and 99. The seed weight of gram was observed that when the distance was higher, then the seed was having its higher weight. These are the some examples of Vethian Thun et al. This is the allelopathic effect of the neem plant extracts on the germination and growth parameters of Belmustus esculentus. So this has been shown that the germination percent by leaf extract, bark extract, and root extract. So by leaf extract, there is very little amount of loss in the germination, and they are giving distilled water control as 100%, then only 14% inhibition in the germination. But due to root extract, it is very much higher. And the other parameters, shoot length, root length, fresh weight of seedlings, dry weight of seedlings, this has also been calculated for the same treatment. And these are the allelopathic effects of neem leaf extract on germination percentage of agricultural crops. In different crops, Pennisetum americanum, Raphanus sativa, Cicer eritinum, and Pisum sativum, different treatments have been conducted. Farmers case study as agroforestry. Per tree every year. Second, 14 acres, neem trees, fruit. And yield is half and a 1 bag respectively. That is half means 50.
I think we can conclude. I think he has covered major all the points of silviculture and agroforestry. So I think Professor Manmohan.
Okay, Dr. Rakesh.
Yes, sir. Yes, sir.
I think he's disconnected. So just Tomar Saab is there. So उनका जो थोड़ा सा बचा था वो पूछ लेते हैं.
Thank you, sir, for your nice presentation and sharing valuable information on the topic of silviculture and agroforestry of neem. And sir has nicely explained about the distribution of neem. He has also mentioned that neem is not found in a hilly region because of the neem is the fourth standard species. And he also explained about the silviculture characteristics, phenology, seed.
Rakesh,
Yes,
let
sir.
the Thomas sir present.
Okay, sir. Thank you. Sir, now I request Thomas sir to continue their topic on current status of neem, important research at AFRI and ICFRI. Sir, Thomas sir, please.
Is he available? I think then we can conclude. We can conclude. Come to the conclusion part.
Dr. Dobriyal sir. Dr. Dobriyal.
Yes, sir.
If Dr. Tomar could not complete this your today's presentations, then tomorrow we can request to Dr. Tomar.
So tomorrow will not be because we have the physical only, no, not Tamanna. So that I think most of part he presented, only few slides was remaining.
His presentation was very good, and he was having vast experience as a FRI. So we are missing some part.
Okay, over to Chairman, sir.
Okay,
so
thank you.
We request to Chairman of the session, Dr. Joseph Saab, for comment.
Thank you, sir. Thank you very much. And first of all, I would like to appreciate the organizers of this National Symposium, Dr. A.K. Pandey, Dean, as well as Organizing Secretary, Dr. Manmohan Dobriyal, and all the co-organizing secretaries, and all the faculty members of College of Forestry and Horticulture, Jhansi. So the this current thematic area, silviculture and agroforestry of Neem, the two papers were presented nicely. I appreciate again Dr. U.K. Tomar as well as Dr. S.B.S. Pandey for their elaborate lectures, which gave a very good information for all the participants. Even I also learned a lot of new things from their presentations. So let us be, and again, coming to the Dr. U.K. Tomar presentation, especially mainly which focused on the genetic improvement of neem. And I think this paper should have been in the third session, I think. It was totally focusing on the genetic improvement of neem. So anyhow, it was a good presentation. And he elaborated about the AFRI, role of AFRI, Jodhpur, in the genetic improvement of neem. They have done a very good work, especially Dr. U.K. Tomar, and they have done very good provenance trial, as well as the NOVAT board project, which they have explained development of neem in different agroclimatic zones. So it was very fantastic, and it has really very good information. A lot of information was been shared through his presentation, and especially the azadirachtin content of the samples, which were mainly Deccan Plateau, which had the maximum the azadirachtin content of 4,743, nearly about. So again, he reported the hot semi-arid condition with the mild winter. They showed a better performance in terms of azadirachtin content. That is also one of the important points to be noted. And temperature range, 28 to 34 degrees Celsius range, was found better for the better azadirachtin content in the plantations. That is also one of the things he reported. And he also reported the flowering and fruiting in the research trials, so related to the silvicultural aspects. And one thing I found, important one missing point, AFRI Jodhpur has conducted a very long research, as per his presentation, very long research they have conducted on the genetic improvement of neem. One of the, we can call it pioneer of genetic improvement in neem. And Dr. Dr. U.K. Tomar didn't mention about any improved varieties or any improved accessions which were registered. So I think he is not available also. Otherwise, he could answer this question, whether any kind of, so maybe Dr. Manmohan, you can collect these informations, then you can share it among the participants. So coming to the next second presentation, which is that directly concerned with the silviculture and agroforestry of the present National Symposium, presented by Dr. SBS Pandey. Again, it's a very good presentation, covered very elaborate coverage in terms of silviculture as well as agroforestry. And it has been mentioned that it is mainly used for it is in the afforestation programs for the reclamation of degraded lands and wasteland rehabilitation, one of the important species in India. And one more point, which is mentioned by him, organized neem plantations are very few and lack in scientific studies. So these things should be noted. So this focus should be given more on this particular aspect. So the scientific study in the organized neem plantations, maybe one or two institutions are conducting a very good long study, but still other institutions in India, whether it's a research institution or academic institutions like College of Forestries, they can also focus more research on this neem related to the silvicultural aspects. One more thing from my recommendations, I think other panelists also, they will be giving their recommendations. And from my point of view, the recommendations related to the silviculture aspect, the economic studies of neem plantations. So that I think today I didn't see much about the productivity as well as the economic studies of neem plantations, which will give the clear picture about the economic importance of this wonder tree, neem. And and also one table was presented related to the nutrient studies. Still more research is required for the nutrient cycling in the neem plantations, as well as in the neem neem-based agroforestry plantations. So that is also one of the recommendations which I want to forward. And roadside plantations and the scattered trees in farm, they are the major practice in India. These are the two major practice which are focusing the neem in India. And also it has been mentioned that indiscriminate cutting of roadside trees is a serious problem for the establishment and growth of neem. So again, there should be recommendations which should be given to all the farmers and all the neem cultivators regarding this particular practice. So which is it is retarding the growth and establishment of the plantations. Coming to the agroforestry point of neem cultivation, it is again the same thing. Economic studies of neem cultivation in agroforestry should be given priority, which this will give a clear picture, which can be used to promote the cultivation of neem in agroforestry and promote among the farmers. And wasteland, again, one more point, the presentation I saw it, 2-3 tables I saw it, related to the growing agroforestry in the wasteland reclamation. So that is also should be given high priority, because the agroforestry neem play a crucial role in the wasteland reclamation as well as in the degradation, eco-rehabilitation of degraded lands, and which can have a huge impact for enhancing moisture availability and biomass production in the wastelands. Because we all know that this particular neem tree is one of the hardy species, and which can establish very easily in the wastelands. And role of neem-based agroforestry for soil and water conservation, especially silvopastoral studies. So as reported by the presenter. So this is also the silvopastoral studies, their role in the soil and water conservation. That will give you a good impact for encouraging neem to be grown in the agroforestry systems. Again, neem leaf mulching, it has been proved. One more table was shown, which increases moisture and nutrient status of the soil. Again, elaborate studies are required to prove this point, because as we all know that neem, sometimes the allelopathic effect is also there. So we have the detailed study of neem leaf mulching that should be carried out by different organizations to strengthen this point. Okay, then one more point, the neem tree, the tree crop interaction studies. mainly there are many studies have been conducted in some institutes, especially the Forest College and Research Institute, Metropolitan. They have conducted many tree crop interaction studies. So like that, many institutions should carry out these tree crop interaction studies and crop compatibility studies with the neem. Again, this can remove the fear among the farmers that neem may hamper their productivity, as well as they may give a negative impact for the cultivation in their agricultural lands. So that is also one of the things. Again, one particular research focused on neem plus cowpea agroforestry practice. It was found better to other agroforestry models, but still more studies from different zones, agroeclimatic zones, are required in this aspect to strengthen this particular point. So these are all the observations from my point of view. I don't want to take much time. Already it's too late. It is very, very nice. All the presentations were very nice. It is very good. Once again, I congratulate the organizers, Dr. Ajay Pandey and Dr. Manmohan Dobriyal for this one. And if you allow, I think panelists also, they can give their comments here. What about Professor Manmohan?
Yes. Panelist, I think Dr. Salil Tiwari Sahab is not there, I think.
Or Dr. Arvind Bijilwan is there. Yes. We can hear from Dr. Arvind Bijilwan about his points on silviculture and agroforestry of neem. Dr. Arvind Bijilwan.
Yes, sir. A very good evening to one and all. Respected chair, am I audible?
Yes, audible, please.
Respected chairperson of the session, and first of all, I must congratulate to the university, that is Rani Lakshmi Bai University, particularly the Honorable Vice Chancellor of the university and the Dean College of Horticulture and Forestry, Professor Pandey, and especially to Professor Manmohan Dobriyal. You are continuously carrying out such type of webinars and seminars. You know, it gives us a platform to interact one another and to take some, very important and touching angles and aspects, particularly in forestry, agroforestry, and horticulture, I can say. So especially Pandey sir and Honorable Vice Chancellor and Dobriyal sir, I must thank you for inviting us, calling us, be a part of this, national webinar. Much of the things very nicely, point by point, have already narrated what the respected presenters have said. That is Dr. B.K. Tomar sir and Dr. S.B.S. Pandey. It was a nice presentation by both the presenters, particularly with respect to silviculture and agroforestry by Dr. S.B.S. Pandey, and more touching to genetic aspect by Dr. B.K. Tomar. There was some interruption in between, but it was a very nice presentation. Dr. S.B.S. Pandey, he has covered most of the aspects related to silviculture and agroforestry. And on the presentation, whatever done by both the presentation has nicely narrated and pointed out by Professor Joseph, the Dean College of Forestry, Swartz. I just would like to say, when I was at IIFM in Madhya Pradesh, I have written a paper that an insight to agroforestry in Madhya Pradesh. So at that time, we have visited different parts of the Madhya Pradesh, a big state having larger forest area. And when we have visited the farmland, we did not find any farmer, or we find hardly any farmer who has not neem on their farmland. But the fact of the matter is that, provided the fact that neem is present in abundance, in plenty in most of the farmlands, but they are knowing the value of that. As we say, neem from stick to storage. It has every use in our life, but it is not taking momentum as far as farmers' level is concerned. So my point is that things which I've already covered here, apart from this, there are certain things. Why not to have a consortia linking to market, to the farmers' level, such that, because everything, nowadays what is happening, this what we call the stick, the neem stick, when it has been marketed in a proper way, a stick which is just of 1 rupee can be sold even of 20, 30 or 50 rupees. And it will directly link to the income of the farmers. That is one way. Another is that that every part of the neem is being used. The leaves, if they collect leaves, farmer, they are trying to collect it, but no as such straightway market linkage is there. Thank for the previous and the present government and our Honorable Prime Minister that they have started this neem coated urea that has also taken momentum to to include economy with neem-based farmers. But these are certain things which are still lacking at the farmers level. So these things should also be taken into consideration, and some research and other things also be carried out in this aspect. This is my observation. Another thing is that, we just praise the neem and people and trees like this. So neem is a very good species in many ways, but the ecosystem services as such has been carried out in such that we can carry out the ecosystem services, what they are liberating in terms of underground and above ground atmosphere. If they are good study can be carried out on this angle. So definitely when these climate related treaty will be finalized, the farmer will be much more benefited planting trees on their farmland. As Professor Pandey, SBS Pandey has narrated, because the spacing of the tree, they are playing very important role. Definitely there are certain things where the genetic engineers are doing. Why we are not, farmers sometimes not willing to plant neem trees on their farmland? Because when it comes to economy, we need to have a straight, bold sort of tree. So there is a role of these genetic engineers, particularly Coimbatore, that FRI Institute can do a better way, develop some straight, bold, transgenic, some clones for neem, which may have similar quality, quantity, and everything related to that, which can minimize that shading effect on the crop. Then obviously farmer will prefer in majority. Another thing is that if you see, it is a frost hardy tree. I'm basically right now speaking from Rani Chauri, College of Forestry, Rani Chauri of Uttarakhand. If you see, people, they are very interested to plant this neem tree, even in this northern plant, where the Populus deltoides and eucalyptus has taken momentum in Haryana, Punjab. But if some hardy species can be developed by the geneticists and the plant scientists, definitely it can take momentum across the country, even in the northern part of the country and northwest and Uttar Pradesh and this region. Definitely it will help to the farmers. One more thing that already, as such, we are lacking, though this is a species, it is a traditional heritage to us, but we are lacking information as such that the majority of agriculture crop, one by one taken in combination with neem, such that means there is a need to find some most compatible, suitable neem and agriculture crop based compatible combination, such that farmer can plant it on their farmland. And this may be a win-win situation in terms of not reducing the agriculture produce up to a significant level, even may sometimes may be very good, because it has a very good potential to, sometimes works as an insecticide and pesticide also. Sometimes it's a positive allelopathic effect. So these combinations and studies have been documented, but it should be carried out in a larger scale, and then it should be lead to the policymakers. Another very important point which I would like to mention here, that is the wider adaptability of neem in silviculture and agroforestry, because it is there and we praise and we are very proud on this neem, but momentum it is not taking like this Populus and eucalyptus and casuarina and nowadays in Melia dubia. I think because we have got a species of the brother of this neem, we call it Melia as a direct, we locally call it Bacan. It has got a similar sort of character. But I think that we agroforester and silviculture in the time to come, there is a need to do some sort of such innovations and intervention. So this is something from my side. One more important, which I have noted down, we have studied something on this, but the nutrient dynamics. What is the dynamics as such? How much that study has been carried out, but not in plenty. So there are certain observations from my side. I once again thank you very much to the chairman of the session, and particularly the Honorable Vice Chancellor of the University. Special thanks to Dr. A.K. Pandey Sahab, the Dean College of Horticulture and Forestry, and to the Professor Manmohan Dobriyal and one and all those who are present here. So thank you very much, and thank you very much for granting and giving me this opportunity. Thank you, Pandey sir and one and all. Namaskar.
Thank you, Dr. Sir. Thank you. Excellent comments.
Thank you, sir. Now I would like to thank the chairman of this session, Dr. Anthony Joseph Das sir, for reviewing this session, and I also want to thank our eminent panelists and speaker who made this session informative with their presentation and knowledgeable discussion. With the permission of chairman of this session, I would like Dr. Ram Prakash Yadav sir for word of thanks. Yadav sir.
Sir, Din sir.
Ji. Summarize.
Should I go ahead, sir?
Dr. Dobrial.
Yes, sir.
We are too late, but some slight comments on to this, your all the sessions.
Yes, sir. 5-minute summarization, sir. Please.
Oh. So my colleague Dr. Dobrial and all chairman Dr. Anthony Sahab, Dr. Arvind Bijalwal Sahab, and others who are connected with this your webinar. Sir, we organized this webinar with a very holistic approach that in present era where there is so much commercialization is going to take place, where we can find a need, where we can find a product from a neem, as this or neem coated urea has made the revolutions. And in morning sessions, this or Patel was pointing out a number of these or products. We need more advertisement. We need more commercializations to need the programs more effective way, what I assume with this Dr. Patel's and others these your presentations. Side by side in most of these your sessions, very good comment has come. In first session where this your session was dedicated for this your plant health management, Dr. Satpathy and others gave a very good presentations, and certainly good comments we received. Further, I expect that such potential, such a this your it's whatever that is a potential up to this year as a pest management and other disease management, more research focus should be made, more some of these your fundamental research, some of to this your applied researches must be this your completed. So, and further they suggested in this your very first sessions, what there is the this your problem into this your licensing of different type of this your pesticide based on to this your name, that is also a very important aspect. And in many cases, good research can break this your change, and we can harness this your full of this your potentials. So only this part to this your first session after this your where plant health management was related. In soil health management, 2 points is the very important. Even Bijalwan's work, one comment that neem, where neems this your nutritions, neems nutrient dynamics, even how this your neem goes into this your rhizosphere, what is this your in different intercroppings, how this your allelochemicals react the things, and even we can go for a very out of the box ideas where we can go for to this your neem taking to the as a green manuring, like, and how this your neem once we are taking to this your green manuring, how it is controlling to this your nematodes and other this your pest and others, how these your changes are taking place. So such type of this your aspect must be covered, and that needs a more focus on to such type of this your researches. In that line, this your Dr. Pandey's this your presentations was also, and very means in different zones, number of the crops, once these your plants are to this your at early stage, that time the crop selections, when the canopy is a big canopy, when there is the setting conditions, that time crop selections and different this your physiological aspect should also be covered, how to this your in that conditions we are able to this your take this your crops. I could not attend to this your genetic resource and improvement sessions, but I listened to this your Dr. Biswas. He was giving a very right comment, and number of these your problems are related with this your genetic improvement, where one this your chemical constituents and if there is any variation in the chemical constituent, certainly that gives the good opportunity for collection of to this your good number of to this your germplasm, and further up to this your advanced breeding approaches. Even we scientists engage in that field, they can utilize to this your biotechnological tools for to this your enhancing the different biochemical constituents which are useful. And second point, he was pointing out that with the help of this your genetic engineering or even Jalwansa this your report, that if any this your genotypes of this your neem, if we are able to this your breed which are going to have to this your state bold, certainly that will be the one of this your most preferred to this your plant. We are not commercially utilizing to this your neem as a different up to this your where such commercial wood or such type of this your bold blocks or woods or logs are needed. So that is one up to this your trouble where this your genetics can play a very good role. And secondly, Dr. Biswas was pointing out that a such type of this your genotype which are having dwarf stature and very good to this your leafy, and even further we can go such type of this your where there is the very once we are going for to this your road type roadside of plantation or as a selecting a roadside avenues are very ornamental trees. So as a roadside avenues are taking as a neem as a roadside avenue, major problem happens to fruit falls during to this your rainy seasons, and that attract to this your flies, and many times that doesn't look to this your good. So if we are going to have such any genotypes where there is the less fruits and more canopy and more leaf and very feathery leaves, so that will be the one up to this your another idea of another aspect of to the developing to this your genotypes. That point should also be taken consider. In silviculture and agroforestry, almost very good comments has been given by to this your Anthony Joseph, and his sum up to this your recommendations should be finally be pointed out and finally to be this your placed into this your our proceedings. As one his comment was that we have conducted a number of research, number of the conclusions, number of the books and articles we have got, but a very systematic study is needed on the economic aspects of to this your neem. That is one of these your very important points we should consider. So by that recommendations we can attack to this your growers for that we can give recommendations that you can go with such up to this your profit and benefit. So I'm not taking much of time. Already we are very much late, but I appreciate to this your all our worthy chairman of this your different session. In first sessions, I am having very much appreciations to our very senior colleague, Dr. A.B. Rai Sahab, who gave a very good comments, and he's working into this your neem based up to this your different formulations right from the beginning, and his experience he spoke in the morning sessions. So in our chairman Dr. Anthony Sahab and Professor Biswas Sahab, all that gave very good comments and very nicely they chaired the sessions. I appreciate all to these panelists who gave a very, shared their experience for the further betterment and further extended to this your research program on the neem. Over to Dr. Dobria.
Sir, thank you, sir, for your concluding remarks and summarizing the whole day seminar, which already exceeded more than 2.5 hours. But it was good learning for all of us, and I hope all of the panelists and chairman and speakers contributed a lot for all the participants in the NEEM, and I thank for their patience also for such a long time. So over to now Dr. Dr. RP Yadav for vote of thanks.
A very good evening to one and all. This is Dr. Ram Prakash Yadav from RLBCAU, Associate Professor in College of Horticulture and Forestry. I feel privileged to propose the formal vote of thanks on this important national event that is the nurturing the neem for nature and livelihood security. It's my great pleasure to propose the vote of thanks. I express my sincere gratitude to all the chairmen, speaker, and panelists for holding different sessions and providing valuable comments during the whole day, and all the speakers for presenting a wonderful presentation on the different aspects that includes the on plant health management, soil health management, neem genetic resources, and silviculture and agroforestry. I also extend my sincere gratitude towards Dean College of Horticulture and Forestry for promoting us and giving suggestions for successful conduction of this webinar, and all the team members, different team members of this webinar. I thanks to all for conducting this, for making this event a successful. In last, I thanks to one and all for the for successful conduction of this event. Thank you, one and all. So, with the permission of Dean sir, I close this seminar.
So, go ahead.
Thank you.
Okay, thank you all. Thank you, sir. And we will share the recommendations after finalizing with all the chairman and panelists for the further. Thanks.
Thank you.
Dobriyal sir, one request is there. Kindly share Pandey sir's and Satpathy sir's presentations to us.
Oh, okay, sir. Okay.
Thank you, sir.
Thanks.
Thank you. So, Surli?
Dr. Tanuj, now you can.
Yes, sir.
Now it's over, so you can switch off now. Thank you.
Thank you.